क्या हो? कौन बोला? तुम कुछ कहे थे अजय? हाँ सर। प्रोभिसी को बोलने बोले थे। प्रोभिसी को। हाँ प्रणाम करते हैं। हाँ कह दीजिए। इसको प्रश्न करवा दीजिए ना। ये तो मरवा नहीं है। कमल जी प्रश्न आप लेके चलिए। है ना आपके पास? मार दीजिए। हाँ करी आपके 10:30 बजे किसी से आ गए।
एक होंगे प्रोफेसर एम के ठाकुर कीनोट स्पीकर, वो तो अभी आ जाएंगे। प्रोफेसर एम को, प्रोफेसर पारस मिश्रा, प्रोफेसर अनिल कुमार, डॉक्टर आकांक्षा, कोई ना कोई अरिजीत आ रहे हैं। ठीक है, फिर डॉक्टर प्रोफेसर पानीग्राही, डॉक्टर विजय, डॉक्टर सुमित निगम। आप इस लिस्ट को ले लीजिए, आपको समझ में आ जाएगा। बाहर
पूरा दुनिया अरे तुम लोग टाइम पर आओगी, तुम लोग का तो विचित्र है जी। अब फंक्शन स्टार्ट होगा, टीचरें लोग नहीं आ रहे हैं। सबको कहो तुम्हारे यहाँ के टीचर लोग कहाँ?
प्रणाम सर।
बुआ जी प्रणाम।
प्रणाम गोर लगे
बुआ जी हैं हमारी। उसको अब बंद कीजिए ना भाई, जरा ऐसे चल रहा है। राम कुमार उसको बंद कीजिए।
दिव्या
ले आया है
वो सब बुके और पॉट सब रेडी है।
गुलरुख जूलॉजी स्टूडेंट शुड नॉट बी
लगा
सिटिंग दैट वे।
खाली एटी बांध दें एक तो
करवा दो दिए हम कागज बनवा दिए। कौन चीज का करवा दिए? सुनो ना फ्रेश कर इसमें मार दो।
20 मिनट बच गया।
फ्रेशनर बनाइएगा, सैनिटाइज करा दीजिए।
सब करा दी सैनिटाइज पूरा करा है, एक चेयर वेयर सब कुछ।
अब आप आ गई हैं तो थोड़ा सा दिक्कत हो गया।
तुमको सैनिटाइज, इसको सैनिटाइज करते हैं।
आगे वाले दरवाजे से हैं।
नहीं
ये आगे वाले दरवाजे को आप सैनिटाइज नहीं करिएगा।
नहीं वो भी हो गया। तू जो मशीन लगा हुआ है पहले, जो पहले सैनिटाइज होगा।
आगे वाले दरवाजे से जो
सैनिटाइज होगा अभी ना।
सैनिटाइज नहीं करिएगा।
जो सैनिटाइजर मशीन से तो पहले सैनिटाइज होगा।
वो तो काम नहीं करता है सैनिटाइजर मशीन।
करता है तो
उस पर डस्ट बहुत है।
सब कुछ साफ। सैनिटाइजर मशीन से कहिए, वहाँ पर लोग आते हैं तो सैनिटाइज हो गया।
सैनिटाइजर है ना उसमें?
चेक कर लो।
कितने लोग ज्वाइन किए हैं?
अरे अभी तुम्हारे
स्पीकर भी सर ज्वाइन कर रहे हैं, वो विजय सब भी सर ज्वाइन।
हाँ विजय, अरिजीत तो आ गया, विजय भी ज्वाइन।
हाँ विजय को तो उसने देखे थे उसके।
अच्छा तब हाँ वंदना
वो भी कुछ देर के लिए ज्वाइन कराया था। अच्छा उस पर लिख के दिए टेलीग्राम पे दे दीजिए।
टेलीग्राम पे
ज्वाइन करने के लिए यूट्यूब पे ज्वाइन करना। यूट्यूब पे चला गया
यूट्यूब में बोल देता है।
दोनों भी तो
हाँ टेलीग्राम पे बोल दीजिए अपना यूट्यूब चैनल।
दोनों हो गया तो उसको लास्ट डिजीज को लास्ट
हाँ हेलो। यूट्यूब आपका लैब है हाँ बंद। हाँ ठीक है बंद करा। हाँ कर
लग रहा है ना।
हाँ नहीं तो लगता क्या है।
हेलो हेलो गुड मॉर्निंग।
गुड मॉर्निंग सर।
जी मैं डॉक्टर मनोज चंदन, एचओडी जूलॉजी, आरके कॉलेज। मेरी आवाज आ रही है?
यस सर।
ओके,
Yes,
थैंक
sir.
यू। आई हैव ज्वाइंड।
थैंक यू सर।
विश करते हैं नहीं बस उनका चेहरा
We are very close to begin our conference. Our Pro Vice Chancellor Ma'am has also arrived.
ठाकुर सर ज्वाइन कर यहाँ देख लीजिए सारा आप सबका देख देख के हाईलाइट कर दीजिएगा इधर ही बैठिएगा उनको हाईलाइट कर देंगे
It's a moment of pride and joy. Please be seated at alternate positions. A very warm good morning to everyone, respected delegates, dear students and our dignitaries. I am Mandavi, an enrolled PhD student of Department of Zoology, LNM.
Good morning, all of you. Zoology, I'm enrolled student for the University Department of Zoology, LNM.
This is a historic moment for University Department of Zoology, LNM, as it is organizing the Grand International E-Conference on the theme Recent Advances in Life Sciences with reference to diseases, disorders and adaptations. In the grand inaugural session of this International E-Conference, we are delighted to invite our dignitaries.
On this auspicious occasion, may I request the Dean Faculty of Science, the head of the Department of University Department of Zoology, and the President of the Organizing Committee of this International E-Conference, Professor B.S. Jha, to take our Honorable Vice Chancellor, Professor S.P. Singh Sir, to the dais and take their respective seats.
request Professor Mohammad Nihal sir to take our Honorable Pro Vice Chancellor Professor Dolly Sinha ma'am to the dais. Sir and ma'am, kindly take your respective seats.
May I request Professor Bhaveshwar Singh sir to take our financial advisor to the dais. And Mr. Kailash Ram sir. Sir, kindly take your seats.
May I request Dr. Parul Banerjee ma'am to take our Registrar of University. I request our Organizing Secretary of this event. Ma'am, kindly take your seat.
Our volunteers are requested to be ready for the floral welcome.
Volunteers are requested. Please be ready.
Taking a step ahead and maintaining cultural background, may I request the President of the Organizing Committee of this International Book Conference, Mr. B.S. Hassel, to kindly present the floral bouquet to our Honorable Vice Chancellor, Mr. S.P. Singh sir, and welcome him. Richard,
संजय भाई आगे मत जाइए, सुनिए सुनबा डांस करता हूँ मैं यहीं से फोटो खींचता हूँ सुनने के बाद
Thank you so much, sir. Now I would request our convener, Professor Mohammad Nihal sir, to present the floral bouquet and the medicinal plant to Honorable Pro Vice Chancellor, Professor Dolly Sinha ma'am. Give a round of applause.
Now may I request Dr. S.D. Verma sir to present the floral key to our financial advisor, Shri Kailash Ram sir, and welcome him. Shivangi.
Volunteers are requested to assist.
वो हम कर देंगे ठीक 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 ठीक
Now I take the honor to invite our Professor A.H. Sir to present the floral bouquet to our Honorable Head of the Department, Professor B.S. Sir. Thank you, sir.
I'd like to request Mr. Bhaveshwar Singh sir to present the floral bouquet to Honorable Professor Mohammad Nihal sir and welcome him.
Thank you so much, sir. Now I'll take this special opportunity to invite Mr. Kali Charan sir to present the floral bouquet
with the permission of the chair, owing dignity to the protocols, let us make the humble beginning of this event.
Light symbolizes knowledge and wisdom free will. May I request the chief guest, our Honorable Vice Chancellor, Professor S.P. Singh sir, and I request all the dignitaries present on the dais to accompany him for lighting up the diya. I request Ms. Divya to assist them. Please give a round of applause. The auspicious beginning of our conference.
सहनावतु शभुनक्तु शब्दकारवावहै तेजस्नावधीतमस्तु ओम शांति शांति शांति।
Our registrar sir has arrived. Please give a round of applause and request.
May I request Dr. Parul Banerjee ma'am to present the floral bouquet to our Honorable Registrar, Mr. Mustaq Ahmed sir, and welcome you. Now is the auspicious time for our Kulgeet. Our chorus group is requested. Please proceed and begin the Kulgeet.
Audience are requested not to clap. I repeat, not to clap after Kulgeet. moment of pride. Our dignitaries are releasing the abstract book. Please give a round of applause. Taking the fragrance of joy and pride ahead, I'll take the honor to invite the head of the Department of Zoology and the president of this grand event, Honorable Professor B.S. Jha sir, for the welcome address. Sir,
Honorable Vice Chancellor, Professor S.P. Singh Sahab, the Chief Guest and the Chairperson of this inaugural session of International Conference on Recent Advances in Life Sciences with reference to disease, disorders and adaptations. Honorable Pro-Vice Chancellor, Madam Professor Dolly Sinha, Honorable Financial Advisor of the University, Sri Kailash Ramji, our young, energetic and dynamic Registrar, Professor Mustaq Ahmad Sahab, my friends occupying the dais, Professor M. Nihal, and the young, energetic Organizing Secretary, Dr. Parul Banerjee, principals of the local colleges, heads of the various university departments, fellow colleagues, delegates, students, friends from both print and electronic media. It gives me immense pleasure to welcome you all. But then the Honorable Vice Chancellor has inspired us in such a way that I have no word to express obligation to him. I am serving this university right from its inception, both as a student and a teacher. So I have put in 42 years of my services and will be retiring now in 7 to 8 months. I remember that for first science conference was held late in 1983 or 84 by late Professor Akuri Balram Prashant, Satellite Symposium. That was the first symposium organized in this university. Thereafter, again, it took a long gap, and it was in 2001 that the Zoology Department organized another conference of national level on fish health and management. And this way the process initiated and other departments also took initiative. But the pace was very slow. So I am now confirmed that the old saying, Supreme Commander अगर अच्छा है तो विजय होगा. सेना तो यही था पहले भी. सेनापति जब आए तो हम लोगों में नया उत्साह भर दिया. और आप देख रहे हैं कि इस कोरोना पेंडेमिक में भी हमारे विश्वविद्यालय ने जितना अंतर्राष्ट्रीय और राष्ट्रीय सेमिनार कॉन्फ्रेंस किया है, शायद सभी बिहार के विश्वविद्यालय को मिला दे तो उतना नहीं हुआ होगा. So this credit goes to our Honorable Vice Chancellor and his team. So please give a big hand to our Honorable Vice Chancellor. Very dynamic and visionary person. How to welcome Professor Madam Dolly Sinha. Very soft spoken, very respectful, but firm in her determination. She would go on insisting you, requesting you, and will get the work done. This is her beauty of getting works done. Having a PhD with IIT Delhi and a vast experience as a principal and then professor, then Pro-Vice Chancellor of Patna University, and right now in our university. she is a rare stuff of her own kind, that without, she has a firm determination. She would go on, she repeatedly requesting you that and reminding you that this work is to be done, this work is to be done, and finally you will have to. So I welcome you, madam, from the core of my heart, on behalf of my department and on behalf of the Faculty of Science. I welcome you once again. Our financial advisor, Sri Kailas Ramji, a very, I say, gentleman in letter and spirit, and he finds out ways and means how to keep the university trouble-free within the ambit of the rules. So I welcome you also. My registrar, Professor Mustaq Ahmad, very energetic, dynamic, can work, put in labor from morning to evening at a stretch. You can see his dynamism and his positivity towards the teaching fraternity. And so I welcome you also, Honorable Registrar sir, and hope that under the leadership of this trial, our university will gain further heights and academic heights, or in all round of development, it will excel. So once again, I request you to give a big hand to our Honorable Vice Chancellor, Pro-Vice Chancellor and Registrar. Sir, within a very short time, we had to organize this international seminar. And I am telling you that this conference is in letter and spirit, has an international shape. We have speakers from Nepal, from states and other foreign countries, and altogether we are glad to inform that 745 delegates are registered right now, and they are stressing up to late in the night that we should be included. We told that now time is over and we can't include all of you. So 745 registrations are there, out of which 20 are from different outside the country, and one of these paper presenter will be from the city of Nabab, from where our Honorable Vice Chancellor belongs. Ankita Alexandra from States. She would be giving an oral presentation in the technical session. So, if there are any shortcomings, sir, I beg apology for that, being head of the department. But if you see anything good, the credit should be given to my team and my colleagues. So, without losing any time, since our Honorable Vice Chancellor has to join an very important meeting with Bihar government, so he would be leaving the venue after, so we will have some shift. So what we will, I would like to simply introduce the keynote addressee in short, so that the Honorable Vice Chancellor may bless and bless us. So our keynote addressee today is none other than the son of the soil, Dr. Mahendra Kumar Thakur. For PhD, FNASC, FIN, FAMS, he is the former professor and head Department of Zoology, Banaras Hindu University, Varanasi, and coordinator DBT Interdisciplinary School of Life Sciences, PHU Varanasi. Presently, he is UGC BSR Faculty Fellow at the Department of Zoology, BSU Varanasi. He has, his native place is Dharampur, some 30 kilometers away from here, and he studied in Saraswati School, a village school, and then moved with his father to Varanasi in 1968 after completing his matriculation. His father was a teacher in the municipal school, and so he is a self-made man, and he has excelled, and he is internationally acclaimed, much in demand in countries like States, Germany, Japan, where he has delivered lots of talks. And then Dr. Thakur is an example of simplicity and is deeply attached to his root. An internationally recognized, reputed, and acclaimed scientist in the field of neurobiology and biochemistry. and he is going to present his work in address today on aging brain and cognitive disorders. When the person ages, brain also ages, and then some biochemical events do occur, and the degenerative changes started. How to combat that? That will be a wonderful lecture. And he has published 150 research papers in not in Indian journals, all in reputed foreign journals. He has authored two very good book, which is even taught in the University of States, Molecular and Cellular Neurobiology and Brain Aging and Therapeutics Interventions. He is recipient of Dad Rockefeller Foundation Fellowship Award in Germany, USA and Japan. He was awarded with ENSA Medal for Young Scientist, ICMR Harvard Award, BHU Gold Medal, Professor M.S. Kamungo Lifetime Achievement Award, R.K. Reddy Memorial Prize, Young Awards. Out of many awards, these are the important awards. He has fellowships of Indian Academy of Science, National Academy of Medical Sciences, National Academy of Sciences. Presently, he is President of the Society of Neurobiology and is former President of INN and Council Member of International Association. Besides that, sir, I would like to let you know that 4 or 5 speakers are the alumni of our department, and they are excelling in states. I still remember Professor Muhammad Noor Alam, who is in the David Geffen School of Medicine, Los Angeles, sitting on a stool in our old building, quite calm boy, always listening and noting to our lectures. He taught here and from this department, and then he was selected at JNU for his MPhil and PhD, and his area is sleep physiology, sleep disorders. He is supposed to be the authority in that, and he is an alumni of our department. Next alumni of this department itself is Professor Anil Kumar, who is right now the nodal officer of all India Survey on Higher Education for the state of Chhattisgarh. He established the Department of Biotechnology and Clinical Pathology there. When he was a student here, he competed, cleared NET, and was selected for lectureship in Madhya Pradesh. And he is a very poor student. With doing private tuition, he somehow cleared his MSc examination, and he was a very bright student. When he was at Madhya Pradesh, he used to give me a call. Sir, I am performing an experiment, and blood is, what media do you use for blood clotting? And I used to tell him. Today I am proud of telling that he can teach every one of us in the department. He is our student. He has a project of 2.5 crores of DST, ICMR and CSIR. So he is also an alumni of this department. Another alumni, Paras Kumar Misra. He is Associate Professor of Department of Cellular and Integrative Physiology, University of Nebraska Medical Center. He did his BSc from LNMU and thereafter his MSc PhD from BHU. Presently he is President of Midland Society of Physiological Sciences, USA. And his interesting area is how diabetes induces heart failure and how that diabetic-induced heart attack can be prevented. He is working on that with the finance of National Institute of Health and American Heart Association. Another speaker is again the alumni of this university, Vijay Kumar Singh. He did his MSc from the Biotechnology Department of our LNMU, and then PhD from Central Salt and Marine Chemical Research Institute. And then presently he is PDF at Department of Microbiology and Surgery, Harvard Medical School, Boston, USA. Then we have another, Dr. Gopal Ji Gopal, a product of this university, did his intermediate from Marwari College, and then honors and MSc from here, then moved to for his MPhil and PhD to JNU, and he did his PhD from Special Center for Molecular Medicine at JNU. And then he is presently working. He has got a big project given by Gujarat government. He is presently working on production of recombinant plastic degrading enzymes financed by Gujarat State Biotechnological Mission. So out of the 12 speakers, we have these 5 speakers who are alumni of our department. And there are three very good, Snigdha Besra and Dr. Akanksha Singh. They are both from Lucknow and they studied from BHU. Right now they are excelling, and one, Dr. Akanksha Singh, she has received recently Fellows Award for Research Excellence from NIH USA. And then we have Arjit Chakrabarti and Sumit Murab and Manish Kumar, all of which I will be mentioning in the different technical sessions because of the paucity of time, as our Honorable Vice Chancellor has to join another meeting. So with this, I again welcome you, sir. If there are any lapses, kindly excuse us. And sir, we want the chairperson to bless us so that he may join the meeting, and then another address will follow. Sir.
Distinguished colleague, Vice Chancellor of the University, Professor Dali Sinha, Registrar of the University, Professor Mustaq Ahmad, Financial Advisor, Kailash Ram, Head of the Department, Dean of the Science, the President of this international seminar, Sir B.S. Jha, other colleagues sitting on the dais, faculty members of the university, head of the departments of the university, dear students sitting in this hall and joining us with the online mode. Really today, it gives me immense pleasure and contentment to note that University Department of Zoology at Mithila University Darbhanga has organized an international e-conference on an excellent, extensive and contemporary theme, recent advances in life sciences with reference to diseases, disorders and adaptations. I feel this conference will be very useful for the general mass, as the world is not only under the grip of the deadly coronavirus, but also of lifestyle disorders such as diabetes, hypertension, etc., which is plaguing the world. The neurodegenerative diseases such as Alzheimer's, Parkinson's are also proving to be maladies in today's times, afflicting the senior citizen. If understanding the etiology of diseases and disorders are crucial to plan proper treatment and devise ways of prevention. Peeking into adaptive strategies of living being, let us appreciate the exceptional ways of working of a living machine. The extremely intricate and elaborate metabolic pathway, interconnected like a spider web, have paved the way for the cybernetics in the field of life sciences. I am sure that the scientific deliberation during the 2 days gives all the glimpses of the latest researches in this field. Skimming through the brochures and going through the list of the invited speakers, I could not help feeling greatly excellent. The keynote speaker, Professor M.K. Thakur, is an erudite scholar and his qualities and academic achievements are vast. I am elated that Professor Panigrahi, Pro-Vice Chancellor, Bardhaman University, is participating in the conference in the capacity of an invited speaker. The fact that 4 invited speakers are alumni of the department and LNMU University, serving in prestigious universities and research institution of USA, is a matter of great satisfaction and fills me with the sense of pride. In the presence of such intellectual scholars during the 2 days conference, the university is certainly going to be on an academic high. The enthusiasm of the entire organizing team under the leadership of the Professor B.S. Jha at Department of Zoology and Dean Faculty of Science is praiseworthy, and I cannot remain untouched by their fervor and commitment. However, although well beginning is half done, the organizing team must also appreciate that half is not more. It is still left to be accomplished. I am confident that the path left to be covered without any glitches, culminating the huge success of the conference. I congratulate the entire team and wish them all the very best. I take this opportunity to welcome all the delegates comprising faculty, research scholars, postgraduate and undergraduate students from all around the world who would participate either physically or virtually in the convention. I hope that a great deal of knowledge sharing goes on during the 2 days and all the participants have a lot to take home. May this conference have an everlasting impact on their academic domain. I wish the conference great success. Thank you.
Thank you so much, sir. Thank you so much for your words of enlightenment.
Thank you, sir.
Now, let me take the opportunity to invite our convener of this international e-conference, Mr. Muhammad Rehan, sir, to share the theme of the conference. Sir, please.
हाँ दोनों में प्रोग्राम चल रहा है ना जो
Mandvi, this is happening for the History Department of Zoology. What was conceived as an idea is now being delivered. Why not then celebrate the moment? Let us join few claps. Thanks a lot. Honorable Vice Chancellor, Sir, Professor S.P. Singh, Honorable Vice Chancellor, Professor Dolly Sena, Financial Advisor, Shri Kailash Manji, Registrar, Professor Ajita Ahmed, Head of the Department and Dean Faculty of Science, Professor B.S. Jha, esteemed colleagues of the dais, participants and online guests, Organizing Secretary, Dr. Farul Banerjee, dear students and media friends, and today's keynote speaker and eminent scientist, Professor M.K. Thakur sir. Good morning, everybody. Acknowledging the hybrid board of the conference, let me begin on a lighter note. Language-wise, we all know we call those who are physically present as offline and those physically absent as online. Anyway, this is the beauty of the language and we go about the way it is. I am privileged to present the theme of the conference. Organizing academic conferences is usually considered a normal activity where ideas and findings are exchanged. But in recent past, a few terminologies have found space into our day-to-day conversations, such as masking, social distancing, new normal, etc. We did experience a global lockdown like never before. Life came to a grinding halt. Academic institutions were shut down. In short, a total disconnect. We lost dear ones, friends, and many more others. For quite some time, it was all disorientation, confusion, fear, loss of hope, groping in darkness. Yes, a tiny RNA virus, invisible, led us to zoonotic corona disease, creating complete disorder and throwing upon humanity a challenge to bring in adaptation. The theme of the conference. However, the show must go on and life has to continue. I hope we all are vaccinated. Let me submit with humility that the initiative and positive push from the Honorable Duo of the Honorable Vice Chancellor and the Honorable Pro Vice Chancellor to organize such academic events came as a rescuer. Grateful thanks to both of them, and we mean it sincerely. It is proving to be a connector as well as a way out to continue with academics against the ongoing restrictions. How we arrived at such a theme is a fine example of collective wisdom and camaraderie of all the colleagues. I am delighted to share that the idea soon became a super hit among all those whom we approached for participation, both nationally as well as internationally. Diseases are categorized as infectious, genetic, and lifestyle related.
Yeah. माननीय कुलपति प्रोफेसर साहब, माननीय कुलपति प्रोफेसर साहब, विद्यार्थी राम साहब, विश्वविद्यालय के विज्ञान संघ का अध्यक्ष, आपके अध्यक्ष मेरे बड़े भाई प्रोफेसर झा साहब, प्रोफेसर एम साहब, इस शोबे के दिगर असातिज़ा, इस हॉल में इस ऐतिहासिक कार्यक्रम के हिस्सा बने हुए सभी गणमान्य शिक्षक विभागाध्यक्ष, छात्र छात्राओं, मैं साहित्य का छात्र हूँ। साइंस को साहित्य के हवाले से समझने की कोशिश करता रहा। आज का जो विषय है वो विषय समसामयिक है। अब जानते हैं कि शब्द जिसे आप हिंदी में विश्वविद्यालय कहते हैं, अंग्रेजी में यूनिवर्सिटी कहते हैं, उर्दू में इसे जामिया कहा जाता है। जामिया का शब्द माने विश्वविद्यालय, ये अरबी का शब्द है और उसका जो लफ्जी माने वो ये है कि ऐसी जगह जहाँ बदलते हुए हालात और आने वाले ज़माने की शनासी का दर्ज दिया जाता है। यानी ऐसी जगह जहाँ ऐसा दर्ज पढ़ाया जाता हो, ऐसा निज़ाम तालीम हो जो बच्चों को वो इल्म दे जिससे वो जिस जगह बैठा हुआ है यानी वर्तमान को समझ रहा हो और जिसके पीछे तारीखी शऊर हो और आने वाले दिनों के बारे में वो अपनी राय रख सके। सच माने में इसलिए आप देखा होगा कि जामिया मिलिया इस्लामिया, उसके नाम यूनिवर्सिटी नहीं लगा हुआ। तो जामिया का जो अरबी का शब्द है वो आज माननीय कुलपति महोदया के आह्वान पर और कुलपति महोदय के निर्देशन में जो निर्देश प्राप्त हुआ हम लोगों को कि इस पैंडेमिक के समय में आखिर विश्वविद्यालय में जो जमूत तारी हो गया है, स्थिरता आ गई है, उसको गतिमान कैसे किया जाए। और एक कार्यक्रम तय हुआ और मुझे खुशी है, जैसा कि प्रोफेसर झा साहब ने आपको अपने संबोधन में कहा कि पूरे राज्य के विश्वविद्यालय में जितना सेमिनार, सिंपोजियम, चाहे वो राष्ट्रीय स्तर के हो, अंतर्राष्ट्रीय के नहीं हुए, उतने मिथिला विश्वविद्यालय में इस कोविड के काल में हुए। और सभी विभाग ने जिनको जो जिम्मेदारियां दी गई उसे जिस तरह निभाया है, वो वाकई वो सभी विभाग अध्यक्ष, उनके शिक्षक, यहाँ के क्रमिक धन्यवाद के पात्र हैं कि कि उन्होंने समसामयिक विषयों पर आयोजन किया और सबसे अच्छी बात ये हुई कि हर विभाग के सामने हर विभाग के लोगों ने अपने आप को प्रतिस्पर्धा के तौर पर इसको लिया कि हम दूसरे विभाग को पीछे कर देंगे। और इसमें जो विभाग, कोई ऐसा विभाग नहीं है जिसने कोई सेमिनार सिंपोजियम राष्ट्रीय स्तर न किया हो। आज मुझे खुशी है कि ये जो ई कॉन्फ्रेंस है जिस विषय पे हो रहा है, ये विषय बहुमंडलीय विषय है, पूरी दुनिया में इस पर चर्चा हो रही है और उस पर हम आज चर्चा कर रहे हैं। माननीय प्रतिकुलपति महोदया का तो ये विषय ही है साइंस। हम लोग तो ऐसे भी आम दिनों में भी उनसे इन सब विषयों पर गहन शिक्षा ग्रहण करते रहते हैं। आज कुलपति महोदय ने जो आपके सामने रखा विचार, मैं समझता हूं कि ये सेमिनार प्रोफेसर महेंद्र कुमार ठाकुर साहब जो हमारे वक्ता हैं, मिथिलांचल की धरती से उनका ताल्लुक है और आज इससे जितने जुड़े हुए हैं, ये मिथिला विश्वविद्यालय में जामिया का जो शब्द है उसको चरितार्थ कर रहा है कि विश्वविद्यालय में सिर्फ हम सिलेबस नहीं पढ़ा रहे, विश्वविद्यालय के छात्रों को हम सिर्फ पाठ्यक्रम पढ़ा के उनको डिग्री नहीं दे रहे, बल्कि आज दुनिया में बदलते हुए हालात के सामने, मानवीय समाज के सामने जो समस्या है, मानवीय समाज जिन दुखों से जूझ रहा है, जिन बीमारियों से जूझ रहा है, उसका मुकाबला कर रहा है और जो नई रास्ते निकाल रहे हैं उस पर चर्चा हो रही है और पूरे विश्व में ये सब विषय हो रहा है। और बिहार में ये पहला विश्वविद्यालय है जो आपने जूलॉजी विभाग के शिक्षकों ने और हमारे अध्यक्ष महोदय ने, हमारे प्रोफेसर बी झा साहब ने, उनकी टीम ने ये विषय को रखा है। और सबसे बड़ी खुशी की बात है कि एट ए टाइम 1000 का 750 के आसपास लोग इससे रजिस्टर्ड हुए हैं। ये मिथिला विश्वविद्यालय के लिए एक है। जानते हैं कि रजिस्ट्रार का काम कुलपति जी के आदेश पर चलना रहता है, कुलपति जी का आदेश से मीटिंग में जाना है तो आपके समक्ष, लेकिन आप सोवेनियर्स को देख करके या फिर बाद में जो इसका लिटरेचर छपेगा तो वो विश्वविद्यालय के लिए, हम लोग के लिए ज्ञानवर्धक होगा। आप सभी लोगों को एक बार बधाई देता हूँ और आपसे विदा चाहता हूँ। धन्यवाद।
Thank you so much, sir, for your expensive time and for the positive and enlightening words.
Thank you so much, sir. To be continued by our respected convener, Professor Muhammad Nihal, sir.
Thank you. It was a very positive intervention and interruption I would call, because the university must function as it is supposed to. But the very presence of our Honorable Pro Vice Chancellor Madam Donisina, along with the Financial Advisor Shri Kailash Ramji, certainly we continue under their guidance and gracious presence. Where I left the theme, I joined that point only. How we reached at such a theme is a fine example of collective wisdom and camaraderie of all the colleagues. I am delighted to share that the idea soon became a super hit among all those whom we approached for participation both nationally and internationally. Diseases are categorized as infectious, genetic, and lifestyle related. Over the period of time, we have triumphed over a number of infectious and communicable diseases, but new challenges are also emerging. Genetic engineering and gene therapy are slowly but surely growing for the otherwise incurable genetic problems. Likewise, modern times have gifted us many lifestyle diseases such as hypertension, type 2 diabetes, osteoarthritis, depression, insomnia, and age-related disorders, to name a few. A general survey reveals in no uncertain terms that lifestyle disorders are on the increasing stage. As regards adaptations, it is a biological dictum that adaptation is the soul and heart of evolution. Living beings do bring in changes within, inside, in response to changes outside. Negative changes are fatal and lead to extinction, whereas positive changes ensure survival and progression. We have received research papers covering a number of diseases like cardiovascular complications, diabetes, neurophysiological disorders, besides effects of pollutants, pesticides, and heavy metals, which will be presented during various technical sessions today and tomorrow. We are privileged to have a bunch of invited speakers from reputed institutions of United States of America and India and few more countries. That includes few alumni from our Department of Zoology as well. We take pride in them. The idea of the conference is all encompassing. That will surely enrich our understanding of life and its processes, of diseases and the complications, of disorders and the fallout, and finally, adaptations as the living armory of survival. Let me conclude in the form of a couplet. गम की अंधेरी रात में दिल को न बेकरार कर, सुबह जरूर आएगी, सुबह का इंतजार कर। थैंक यू।
सुबह जरूर आएगी और सुबह का इंतजार. We all are waiting for the sunrise. As Sir has said, the show must go on. I would like to invite our respected financial advisor, Mr. Kailash Ram Sir, to address us. Sir, please.
मंचासीन माननीय प्रतिकुलपति महोदय, ग्रीन साइंस, यहाँ उपस्थित प्रोफेसर, स्टूडेंट्स, ये मिथिला विश्वविद्यालय का एक बहुत ही गरिमामय कार्यक्रम शुरू हुआ, जिसमें कि विश्व के गिने चुने विद्वानों के द्वारा रिसेंट एडवांसेज इन लाइफ साइंस एंड विद रेफरेंस टू डिजीज डिसऑर्डर्स एंड एडप्टेशंस, इसके ऊपर एक ऐसी चर्चा होने जा रही है जिसमें लोगों के विचार और ज्ञान पब्लिश होकर के पूरी दुनिया के लोगों के हाथ में जाएंगे और इसका श्रेय होगा हमारे विश्वविद्यालय को। इसलिए हम विश्वविद्यालय को बहुत बहुत धन्यवाद देते हैं। इसमें जो चैप्टर जिस पे कि ये डिस्कशन होना है, रिसेंट एडवांसेज इन लाइफ साइंस, बड़ा अफसोस के साथ कहना पड़ता है कि हमारे देश की सरकार ने केवल एलोपैथी और केमिकल से बनी हुई चीज़ों के ऊपर ही सारा ध्यान केंद्रित किए हुए हैं, जबकि इस विज्ञान के युग में हमारी सबसे प्राचीनतम चिकित्सा प्रणाली आयुर्वेद, यूरोप से आई होम्योपैथी, यूरोप से आई इलेक्ट्रो होम्योपैथी, हमारे मुस्लिम कंट्री से आई यूनानी, सिद्धा, योगा, बहुत सारी चिकित्सा की प्रणालियाँ हैं, लेकिन इनको जो है हतोत्साहित करके केवल एलोपैथी के ऊपर ही सरकार का ध्यान केंद्रित है। ये जो हम बीमारियों का नाम पढ़ते हैं, इसके पीछे अगर हम जाएं तो मालूम पड़ेगा कि हमारे खान पान में जहर मिला हुआ है, खेती से लेकर के दूध तक, हवा में जहर है। ये जहरीले रसायन पदार्थ जो हमारे डाइजेस्टिव सिस्टम को डैमेज करने के साथ साथ हमारे ब्रेन को भी, हेल्थ सिस्टम को भी डैमेज करते हैं। इसके ऊपर अगर हमारे विद्वान लोग रिसर्च पेपर प्रस्तुत करें, उनसे बचने का उपाय दें तो बेहतर होगा। हमारी जीवन शैली बिल्कुल बर्बाद हो चुकी है। प्राकृतिक जीवन का जो ढंग है उससे हम बिल्कुल विमुख हो चुके हैं। रातों में जागना, दिन में सोना, फैटी चीज़ों का सेवन करके पेट की चर्बी बढ़ाकर ओबेसिटी बढ़ाना, अधिक से अधिक आराम करना, फिजिकल लेवल कम करना, ये हमारे बहुत सारी बीमारियों का कारण है। इसके ऊपर भी हमारे विद्वजन लोग जब चर्चा करेंगे तो उस पर भी ध्यान रखें, क्योंकि हम अगर प्रकृति से दूर जाएंगे तो प्रकृति ही हमारा नाश कर देगी। हमने विकास के नाम पर इतने सारे जंगलों को खत्म किया, जंगल के जीव जंतुओं को खत्म किया, हमें बिल्कुल दया नहीं आई। लेकिन अपने ऊपर जब प्रकृति का जब वार आया है तो हम कितने चिंतित हैं, कितने परेशान हैं, कितने घबराए हुए हैं। अभी कोविड पैंडेमिक का हमारे देश में अभी तीसरा फेज स्टार्ट हो रहा है। अमेरिका में चौथा फेज भी आ गया। एक इनक्योरेबल फंगस वहाँ पकड़ में आया है जिससे पूरा अमेरिका दहशत में है। इन सब के पीछे हमारी जो है बिल्कुल लापरवाही के साथ में प्रकृति के स्रोतों का दोहन और उपयोग करना है। हमारे विद्वजन बहुत सारी चीज़ों पर विचार करेंगे। हम केवल वायरस और बैक्टीरिया ही ना पकड़ें, अपने जीवन के उन पहलुओं को पकड़ें जिनके कारण ये वायरस बैक्टीरिया आए हैं। पहले नहीं थे, एलोपैथी के साथ आए हैं और एलोपैथी के अलावा और किसी के यहाँ वायरस है न बैक्टीरिया है, लेकिन उनके यहाँ संपूर्ण चिकित्सा है। हमारे विद्वजन लोग इस पर विचार करें। मैं चाहूँगा कि ये जो हमारा इंटरनेशनली कॉन्फ्रेंस हो रहा है अच्छी तरह सफल हो और यहाँ के द्वारा जो रिसर्च और ज्ञान की नई नई चीज़ें जो आएँ वो हमारे अधिक से अधिक आदमियों के पास पहुँचे, दुनिया के लोगों के पास पहुँचे और हमारे विश्वविद्यालय का नाम ऊँचा हो। धन्यवाद।
Thank you so much, sir. Now, I will take the honor to invite our organizing secretary of this event, Dr. Parul Banerjee ma'am, to present the report. Ma'am.
Thank you, Nidhi and Mandavi. Honorable Pro Vice Chancellor, Professor Dolly Sinha ma'am, our head Department of Zoology, who is also the Dean Faculty of Science and the President of this event, Professor B.S. Shah sir, the convener of the conference, Professor M. Nihal sir, the co-convener of the conference, Professor A.N. Shah sir, who is also the proctor of this university, Professor Bhaveshwar Singh sir, Dr. S.K. Verma sir, Professor D.N. Mishra sir, the heads of departments of the other departments of this university, my colleagues, my senior sirs and ma'ams, my sir from Banaras Hindu University, who is also the keynote speaker of this conference, Professor M.K. Thakur sir, and all the esteemed invited speakers of the conference, my dear students, all the delegates who are joining us, either physically or virtually, warm greetings of the day to you all. In less than 1 month's time, we have been able to come up with this e-conference on the theme Recent Advances in Life Sciences with reference to diseases, disorders and adaptations. And it is all because of the encouragement of our Honorable Vice Chancellor, Professor S.P. Singh sir, and Pro Vice Chancellor, Professor Dolly Sinha ma'am, that we could take the plunge. And once we were into it, there were very scary moments where we thought it would not be possible to make this event a success. And therein came in our head of the department, Professor B.S. Shah sir, who was always encouraging, and because of whom we have got the whooping 133 abstracts in the abstract book. And since there was a time when we were running short of abstracts and we were worried that the abstract book would look very empty. But then, after Sir intervened and he started talking to people from other universities, we started getting abstracts, and at one point it was raining abstracts. We were overwhelmed with the number of abstracts at hand, and it was a tough time, and we spent day and night together editing and formatting the abstracts. And there my colleagues, Dr. Kalicharan Mishra sir and Varun Prabhat sir, have helped us a lot in editing and bringing in front of you this abstract book. Briefing you about this 2 days program. This is going to be a very packed 2 days program for us, as you must have noticed in the schedule. And this entire thing is divided into several technical sessions. The first technical session today would be diseases, infectious and genetic. Now, diseases can be genetic because of some malfunctionings of the internal machinery of the body, or it can be because of some outside pathogen. So we have very esteemed 2 speakers who will be talking about diseases. In the next session, we have host pathogen dynamics. Now there is a continuous arms race that goes on between a host and pathogen, and they are continuously co-evolving. And this is also called an arms race. As Alice was said, in the famous Alice in Wonderland, Alice was said by the Red Queen, it takes all the running that you can do to keep in the same place. So we are running a race. We as host and the pathogens, our pathogens, they are running a race. The whole pathogens are devising strategies to attack us, and we as hosts are devising counter strategies to evade the pathogen. And this way, we are to maintain our existence, we have to continuously evolve and continuously run. This would be the second technical session, the host pathogen co-evolution or dynamics. The third technical session is about tissue engineering and in vitro disease models. Now this is very important. When you have to understand something, a condition, a disease condition, you will have to understand what happens. And more often it is very difficult to get real life models, or it is very difficult to perform experiments on these real life model. So for that, it is nowadays the researches on in vitro disease model is trending, and we have a very eminent speaker, Dr. Sumit Murad, who will be speaking on this. The final session of today would be oral presentation, where we have speakers not only from all around India but also from abroad. We have a speaker from Harvard Med School, Dr. Ankita Alexander. We have speakers from St. Joseph's College, Bangalore, from NCCS Pune, from RK College, Madhubani, and many eminent good workers who will be making their oral presentations. The next day, that is 27th, the first technical session is related to physiological disorders, where 2 eminent speakers, Professor Noor and Dr. Paras Mishra, would be talking on sleep disorders and type 2 diabetes and associated cardiovascular conditions, and how to intervene through miRNA in that session. The next session would be related to biological adaptations, and we have 2 very eminent speakers who would be talking on basically this session related to biological adaptation was devoted to the model organism Drosophila, and how that Drosophila can be used to study adaptations, biological adaptations, simple adaptations like the simple mating behavior or remating behavior, and how our body strategizes to maintain the equilibrium. And then we also have a speaker who would say that how the body adapts to toxic chemicals, and the model again would be Drosophila, right? Finally, the third technical session on 27th would be fish toxicology, where Professor Panigrahi, an eminent fish person working in the field of fish physiology, would be talking on the heavy metal toxicity and what it does to the fish. The final session on 27th would be again oral presentations, and during the last 2 sessions also we would have a few oral presentations, and the last session, not the second last session of 27th, would be devoted to the oral presentations, where again we have many eminent speakers. I have my own teacher from St. Xavier's College, Ranchi, who will be speaking on immunology, and there are many other big speakers who will be there, who will be giving an oral presentation. Even the oral presentations are very, very, very interesting. And so I urge all the participants, and I promise all the participants, that these 2 days are going to be academically, you will be charged. And let's hope that this is just the beginning and the stepping stone, and this conference would pave the path for a lot of future collaborations. And especially I tell my students, please listen to all the talks and try to gain maximum knowledge from those talks. And I'm hoping that you will learn a lot, and you will also work like this, and in future you will be called in such conferences to give invited talk or presentation, and you will be regularly participating in conferences. Thank you.
Thank you so much, ma'am. As ma'am has mentioned, we are having a bunch of speakers. Before that, I will take this special opportunity to invite the eminent physicist having doctoral degree from IIT Delhi with vast academic as well as administrative experience. My favorite, my inspiration, Professor Dolly Sinha ma'am. Ma'am, please come. And ma'am will also give the presidential address.
सभी को मेरा नमस्कार, वेरी गुड मॉर्निंग। प्रोफेसर S.P. Singh, Honorable Vice Chancellor of LNMU University and the Chief Patron of the conference, Professor B.S. Rao, Dean Faculty of Science and Head Zoology, also the President of this conference, Professor Mustaq Ahmad, Registrar, LNMU University and co-patron of the conference, Sri Kailash Ramji, Financial Advisor, LNMU University, respected delegates, participants, faculty members of other departments, and particularly the Department of Zoology, and also the zoology teachers of various units of LNMU University, all members of the organizing committee, Professor Muhammad Nehal, convener, Professor Ajay Nath, co-convener of this conference, Dr. Mark Banerjee, organizing secretary, Professor Bhavishwar Singh, coordinator, technical sessions and press and media, Dr. Sishir Kumar Verma, coordinator, technical sessions and press and media, representatives of press and electronic media, other distinguished academicians and personalities in the audience, ladies and gentlemen from all corners of the world who have joined us physically or through online
students. It is indeed a matter of great happiness that the Department of Zoology is organizing an international conference on advances in life sciences with respect to diseases, disorder, and adaptation. The topic of the conference is very interesting and relevant in the backdrop of COVID-19 pandemic scenario. I am delighted by looking at the list of distinguished speakers and resource persons. What a wonderful congregation. And I am confident that the deliberations during these 2 days would be of very high level. We are fortunate to have with us like Professor Mahendra Kumar Thakur, the former head Department of Zoology and Dean Faculty of Science, BHU Varanasi, and presently the UPCBSR Faculty Fellow. And he has it is particularly very happy moment that this distinguished scientist is having his root in the soil of Mithilanchal. Professor Mahendra Kumar Thakur, sir, on behalf of the university and on my personal note, I welcome you once again in this conference. We have with us a set of distinguished personalities like Professor M. Noor Alam of USA, Professor Anil Kumar from Durg, Chhattisgarh, India, Dr. Paras Kumar Mishra from USA, Dr. Snigdha Mishra from USA, Dr. Akanksha Singh from USA, Dr. Manish Kumar again from USA, Dr. Arijit Chakravarti again from USA, Dr. Vijay Kumar Singh, USA, and Dr. Sumit Mura, USA, and many other distinguished resource persons. On behalf of the university, I welcome you all and also thank you for giving your valuable time for this conference. The credit of organizing the conference of this level goes to Professor B.S. Jha, Dean Faculty of Science, and also the head of the Department of Zoology, and his team of faculty members and research scholars. I appreciate their effort wholeheartedly. I am very sure that a large number of teachers and student participants would be hugely benefited and academically enriched by the high quality lectures and deliberations by distinguished professors and researchers from various parts of the world, including many from highly reputed centers of research and learning from USA, India, and many other nations during these 2 days. I also appreciate the Department of Zoology because when we make any list of subjects, Z comes at the end. But when we started the present series of international conference, seminars, and symposium, after the locking period of second phase of COVID-19, Z became the first. So you all deserve appreciation for that. I have a chance to look at the abstract book which contains large number of excellent papers, and I hope after the conference we can publish a book of invited papers and contributed papers to any international publishing house. It is also doubly satisfying for me that the responsibility of organizing a conference at such level is shouldered by a young lady teacher of the department, Dr. Paru Banerjee, as coordinating secretary. On behalf of the university and also on my personal note, I extend my best wishes for the success of the conference. I don't have any hesitation in telling you or submitting that I have a very limited knowledge of biology, because when we were students, biology was taught in a different way.
And I am very happy that the way biology is evolving with the support of physics and other sciences and is being taught these days is a matter of great satisfaction. So biology, though I have a limited knowledge, but being a person of physics, I see there is large application of physics in all areas of biology these days. Physics, computer science, data analytics, machine learning, artificial intelligence, biological engineering are enabling and accelerating the change the biology used to be taught earlier and now it is being taught. These have revolutionized the world of life sciences. Of course, it has started earlier and took a few decades to come to this stage. The effort to map the human genome began in 1990 as a foundational building block. But the power of this map only began to materialize when it became cheaper and faster to sequence DNA. The cost of DNA sequencing has been decreasing at a rate faster than Moore's law now. We all are familiar with Moore's law transistors in the context of computer science. Advances in lower cost and high throughput screening HTS have helped lower the cost of entry, accelerated the pace of experimentation, and generate new forms of data to help us understand biology better in recent years. Advances in microscopy, again area of physics, reveal how the human genome organizes itself in three-dimensional space at much higher resolution than previously possible. A new study finds that transcription generates a force that moves across DNA strands like ripples through water. The discovery may have future implications for the understanding of genetic diseases such as Cornelia de Lange syndrome, a developmental disorder that affects many parts of the body. The features of this disorder vary widely among affected individuals and range from relatively mild to severe. It is linked to formation folding and open new avenues of research in genome fragility and cancer development. Biomedical advances have made it possible to identify and manipulate features of living organisms in useful ways, leading to improvements in public health and other related areas. The globalization of scientific and technical expertise also means that many scientists and other individuals around the world are generating breakthroughs in life sciences and related technologies. Advances in biological science could help in tackling global challenges from climate change to pandemics and thus transform economy and society in a positive way. Though plagued by the emergence of the novel coronavirus right from the start of 2020, scientific feats achieved in this year too are no mean achievement compared to the past years. Let us look at some great scientific breakthroughs in the area of biology even during the year 2020, the year marked by COVID-19 pandemic. Global responses to novel coronavirus SARS-CoV-2 illustrated substantial advances in biological science in just a few months. The speed with which scientists sequenced the virus's genome weeks rather than months bore witness to the new improved world of biology research. However, sequencing is just the start. Biological innovations are enabling the rapid introduction of clinical trials of vaccines, the search for effective therapies, and a deep investigation of both the origins and the transmission patterns of the virus. The lightning speed invention of the COVID-19 vaccine was another scientific breakthrough in 2020. The first vaccine for COVID-19 by Pfizer Biotech was developed, manufactured, approved, and being administered. Vaccines were developed at various laboratories in many other countries, including our own vaccine and Covishield. The effective mechanism for vaccine drive management was also developed by the medical and health worker fraternity. Drugs targeting coronavirus was discovered. This all happened in a short span of just over a year, which previously took many years to complete. We must salute the scientists, medical personnel, and all who contributed behind this exhaustive work. Going by the McKinsey Global Analysis Report, the innovations in biology may be grouped into 4 areas. First, the biomolecules. Second, the biosystems. Third, the biomachines. And fourth, biocomputing. Biomolecules relate to the mapping, measuring, and engineering of molecules. Biosystems involve engineering of cells, tissues, and organs. Biomachines is the interface between biology and machines. And biocomputing use of cells or molecules such as DNA for computation. Major breakthroughs in each of these 4 arenas are reinforcing one another. In biomolecules and biosystems, advances in genomics and molecular technologies are enhancing our understanding of biological processes. This also enabling us to engineer biological systems, increasingly including possibility to measure neural signals and fabricate precise neuroprosthetics. It is now possible to store the world's wealth of data using DNA. Here, I am delighted to mention that the storage density of DNA is about 1 million times that of semiconductor storage system. The capability of engineered and reprogrammed human and non-human organism is increasing. Gene therapies could offer complete cures of some diseases. A new generation of biomachine interfaces relies on close interaction between humans and computers. Such interfaces include neuroprosthetics that restore lost sensory functions or enable signals from the brain to control physical movements. Being a person of physics, I could not resist from sharing about the latest invention of a new optogenetic tool, a protein that can be controlled by light signal. Researchers at Bochum, Germany, used an opsin, a protein that occurs in the brain and eyes from zebrafish, and introduced it into the brain of mice and monkeys. Unlike other optogenetic tools, this opsin is not switched on, rather switched off by light. Experiment also showed that the tool could be suitable for investigating changes in the brain that are responsible for the development of epilepsy. A team led by Professor Nelline Mart from the Behavioral Neurological Research Group and Professor Stefan Hartlight from the Department of General Zoology and Neurobiology described the experiments and results in the journal Nature Communications latest issue published online on 23rd of July 2021. It is claimed that in human health, at least 50% of the current global disease burden could be addressed using available science today. New biological capabilities could bring about meaningful change across human health, economy, and lead to a happier society. At the same time, we cannot ignore the fact that biological innovation has profound and unique risks unless used with utmost care, caution, sensitivity, and sensibility. The rapid advances in genetic engineering and biotechnology could enable the production of biological weapons with lethal and unpredictable characteristics. The risks posed by bioterrorism and the proliferation of biological weapons capabilities are now a matter of increased concern. The ethical implication of biomedical progress is also an area of extreme concern. The biologists obviously have to play the right kind of role in this context. They must be careful to anticipate, identify, and find out ways to mitigate these dangers. I am very happy that the Department of Zoology is conducting this conference, and I am sure and confident that during the 2 day deliberations, all these aspects of biological science will be taken care of, and
privilege to welcome Professor M.K. Thakur sir to deliver his keynote address. He is also my teacher from Banaras Hindu University, and personally I always remember his classes, how he taught biological techniques, and it is his teachings that I apply now in my classes. And professionally he needs no introduction. As sir already mentioned, Dr. Thakur was awarded the Dad Rockefeller Foundation, MRC and JSPS Fellowship for postdoctoral research in Germany, USA, UK and Japan. His achievements have been recognized by conferring him with the INSA Medal for Young Scientists, ICMR Marwa Award, BHU Gold Medal, MS Kanungo Lifetime of Achievement Award in Biogerontology, R.K. Reddy Memorial Prize and Yen Academics Award. He is the Fellow of Indian Academy of Neurosciences, IAN, National Academy of Medical Sciences, NAMS, and National Academy of Sciences, India. And he is presently the President, Society of Neurochemistry, and former President of IAN and Association of Gerontology, and Council Member of International Association of Gerontology and Geriatrics. Today he will be talking on aging brain and cognitive disorders, and it's again my privilege to invite him. Over to Sir, Professor M.K. Thakur.
Thank you very much, Dr. Paru. Should I share my slide first? Then I can start. Am I audible?
Yes, yes.
Sir, as you feel, as
Can you see the slides?
स्लाइड नहीं आ रहा है सर, नो स्लाइड सर.
the slides are not visible.
Hello. Can you see the slides?
नो सर.
So just beside that there is a screen presenting options where you have the arrow facing upwards. Please click on that arrow.
Which icon?
So just beside the three dots, you have that screen presentation icon.
सीखे
Just
बोल सकते हैं वो
The arrow that is pointing upwards.
Just click on arrow button. Just click on arrow button. Select now. Beside CC. Beside CC. Just click on arrow button.
जैसे यहाँ से प्रेजेंट कर रहे हैं, यहाँ से करेंगे ना, उनको भेज दीजिए सीन तुरंत। व्हाट्सएप नंबर है, फोन यूज कर रहे हैं, बात कर रहे हैं फोन
सर बात करते हैं। सबका सब ट्राई कर सकते हैं।
Hello. Yes. Are you audible? Yeah, we can hear you. Okay, sir. Sir, very fine. Sir, kindly send your PPT. I'll provide you the mail ID. Meanwhile, I'll share your PPT. If you don't have any problem, so I'll provide you the mail ID.
कि स्लो चलेगा,
I'll provide you the mail ID.
दिक्कत करेगा।
Okay, okay. Thank you so much. I'm just getting you the text. I'm sending you the text.
वो भी स्टूडेंट सर के नहीं उनका पीपीटी और उनका पेपर प्रिंट कर व्हाट्सएप नंबर है सर का सेंड कर देंगे गूगल पे।
Or can you message this? Just send it.
There is a chat box, sir. In the Google Meeting, there is a chat box. You just open that, and there is a text message. Sir, it's in the lower part of your icon, taskbar. There is a three dot. When you open the three dot, you will find the text message, chat message option, and when you will open that, I have provided you my mail ID. It's 28abhinav@gmail.com. I think you will find, sir.
28 Abhinav.
Abhinav, yes, sir. ABHINAV.
NAV.
APHINAV. V for victory, sir. Yes, sir. @gmail.com, sir.
@28 Abhinav
@gmail.com.
@gmail.com.
Yes.
28 Abhinav, right? ABHINAV.
Yes, sir.
Okay,
Yes, sir.
I'm sending this. Just confirm that you.
Sure, sir. Sure, sir. Sir, I got your mail. Hello. Sir, I got your mail. Mahendra sir, I am audible. Mahendra sir.
Hello. Did anyone call him to reconnect? Did anyone call to Mahendra sir to reconnect? Good.
Hello.
हाँ भाई.
It's a request from to organizing committee. Hello. Parul ma'am, can you hear me? Hello. Okay, okay. Thank you so much, ma'am. Please.
Unmute chair.
Unmute sir.
Okay, thank you. I'm sorry for the technical glitches which has delayed a little bit. Thanks, Parul, for your nice introduction and appreciation. And I'm very happy that this conference has been inaugurated on a very special day. That is first Monday of Shravan month, which is very dear to Lord Shiva. So greetings from the city of Lord Vishwanath, Banaras. And I wish that Lord Shiva bless all of us with good health and happiness during this pandemic period. It's my great pleasure and honor to be part of this international conference, which is being organized by Zoology Department of LNMU University. For this organization, first I congratulate Honorable Vice Chancellor Professor S.P. Singh
Pro Vice Chancellor, Madam Dolly Sinha, Dean, Faculty of Science and Head of Zoology Department, Professor B.S. Jha, and other members, including Professor Bhaveshwar Singh, Professor M. Nehal, Professor A.N. Jha, Dr. Parul Banerjee. The theme of this conference is very pertinent to present time. Recent advances in life sciences with reference to diseases, disorders, and adaptations. In line with this theme, I have planned to speak on ABCD, that is basics of aging brain and cognitive disorders. For the benefit of students, I will try to make the talk less technical so that at the end, please wait for a moment, go back to the first slide. So at the end of the talk, you can realize that the old concept of cognitive loss in old age is not inevitable, and there is a scope that cognition can be maintained normal till late age. So I have, in the first part of my talk, I shall tell you about the importance of this aging study, the changes which occur in the brain. That is the focus of our laboratory. And in the middle, I will tell you about the cognitive impairment, which is a characteristic of old age, and some data from our research. And at the end, I will tell you about the strategies which can be followed to maintain this cognitive function in old age, even in old age. So should we go to the next slide, please? So this slide shows the population demography. If you see the histogram here, it represents the population in billion of elderly people, those who are 60 plus. And presently it is around 1 billion, which is expected to double to 2 billion by 2050. And majority of this elderly population will come from developing countries, which is highlighted in red color here. And this developing country, including India, if you see the scenario from India, you can see here that the people are living for longer age, and because of their higher life expectancy, we are more and more elderly people, and this will keep on increasing with advancing years. So with increase in the proportion of elderly people, next please, the question rises. Next please. Yeah, so the question, yeah. Is it okay?
Yes, sir. Sir, okay, sir. So you may continue. Completely fine, sir.
So with increasing age, it's quite common that there is high risk of cognitive impairment and brain diseases. So as you see in this slide, that as the people become more and more old, there is more risk to get the brain disease and the cognitive disorder. And this risk varies from individual to individual. So if there is any infection or any kind of injury at the beginning, say at the 30 years of age, then this aging rate becomes faster, and one will age our brain will age very fast. And because of this faster aging, there are more chances of getting cognitive decline and brain disorders. Next, please. So, next, please. Yeah, so this rising elderly population provokes us to ask questions that why do we experience changes in our mental abilities as we age? Most of us who are in the range of say 45 years and above will experience that daily we have some kind of forgetfulness, and when we start forgetting things, then there is we are afraid that one may get the cognitive disorder. So it's very important to understand that why this cognitive disorder occurs in old age particularly. So the next question is what happens to our brains to cause these changes, because this cognitive function is the vital function of the brain. And if we know the changes, then we can ask, what can we do to keep our brains active? So in today's talk, I will be telling you about these aspects. So next, please. Next, please. Yeah. So this brain is very important organ of the body. It's a master organ, which is linked to the spinal cord, and this forms the central nervous system. And through this spinal cord, it is connected to other organs of the body. So this controls almost all organs of the body, and that's why it's a master organ. And it's very complex. Its structure is very complex, and you can see that there is high heterogeneity in the brain consisting of several cells and regions. And each region of the brain is responsible for a particular function, vital function, like vision, speech, emotion, balance, and so on. And so it's very important to have coordination among different regions of the brain. So we let us see the structure of the brain in the next slide. Yeah. So this brain consists of billions of cells, which are known as neuronal cells. These are special cells of the brain. And 10 times more, there is another type of brain cells, glia cells, like astrocytes, oligodendrocytes, and so on. And this neuron is very unique. It has several branches, which are known as dendrites, that help in receiving the information. And this information is transmitted through a long branch, what is known as axon. And through these terminals, it is connected to another neuron. So this connection is known as synapse. So in the brain, when there are billions of neurons and trillions of connections between these cells, it forms a very complicated network. And this works in a coordinated manner. And the signal which passes from one cell to another is usually the chemical signal, most of the cases, and these are known as neurotransmitters, which are passed from one cell to another. Now with advancing age, or because of some pathological condition, if there is any change in the structure of the neuron or glia cells, or in the neuronal circuit, or in the signal transduction here, or in chemicals like neurotransmitters. So these changes with aging or under pathological conditions will cause some kind of brain disease and cognitive. So we'll study, next please, yeah. So when the techniques were not available, it was very difficult to study the changes in the human brain. And most of the information what we have is from animals. But when the radio imaging techniques became available, now it's very easy to see the live brain and also see its function. So there is a technique known as magnetic resonance imaging, or MRI, which can tell you about the structure of the brain. There is another sophisticated technique, functional MRI, which can tell you also about the functional aspect. And there is a technique known as PET, that is positron emission tomography, which can tell you about the functional aspect of the brain. So if you compare the structure and function of the brain between young and old, you will find several changes, like the brain mass. So with advancing age, there is shrinkage in the mass of the brain, and this shrinkage is not throughout the brain. In particular region, you'll find more shrinkage. For example, a region of the brain, cortex, hippocampus, they show more shrinkage as compared to other regions. And this shrinkage is mostly because of loss of synaptic connections. So the region of the brain which is not being used, they get lost. There are morphological changes. So if you see the outer surface of the brain, as you see in this PET scan here, it's convoluted. So usually the folds, they become, swellings become narrow, and the groups become more wide. Then there is decline in white matter and gray matter of the brain with advancing age. There are lots of studies on gene expression changes in the brain, and as we have done a survey with our studies, you can see in this graph, there are lots of genes, a number of genes which show down regulation, they are decreased. There are lots of genes which are increased, and there are few genes which show no change. So because of this, because of these in the gene expression, something else has come. So can you show my slide? Somebody else has loaded, I think. Or okay.
Yes, sir.
Somebody else has loaded. So somebody else has loaded the slide. So
Let me check, sir. Let me check. Just a minute, sir.
Okay. Okay. All right. You can see the slides there. Yeah, now it's coming. So you can find the change also in the neurotransmitters and the signaling. And because of all the changes in the structure of the brain or the neurotransmitter or other chemicals, you find change in the function of the brain. A very important aspect, which is a very hot topic these days, is gut microbiome. Our gut has billions of microorganisms of different types. And these microorganisms, they produce some metabolites, they produce some vitamins, which are very necessary for maintaining our health. And when there is change in this microbiome with age, so that brain function, right? A very recent studies has shown that when young children are exposed to antibiotics, say like penicillin or amoxicillin, which the doctors prescribe, so this exposure to antibiotic, it alters the gut microbiome. And because of this change in gut microbiome, there may be change in the brain development, and there may be neurodevelopmental disorders at later stage. So this gut microbiome is very important to study. And there are several factors, as you see in this slide, which influence this rate of aging of the brain, like education. So it is said that use it or lose it. So if you use your brain more and more, you have more learning, more exposures, then more challenge to the brain. So this kind of education will make the connections more strong, and your brain can remain more active. And if not, then it may age faster. And then exercise. It is said that exercise helps in the maintaining the brain more active and functional throughout the year. Then the rest, particularly sleep, is very important for maintaining the brain function. I shall tell you later about this. Stress, it's a very important factor. So when there is more stress, there is release of hormone cortisol, which can damage the brain and make aging more faster. Hypertension, that is high blood pressure, high sugar, high cholesterol. So these are all detrimental for aging of the brain and overall the general health. So which includes our lifestyle, diet, and so on. So I think there is a coordination, and yeah, you can move to the next slide. So with normal aging, there is slight change in the cognitive function, and these changes are preclinical. This is known as the silent phase. So there may not be measurable symptoms. So this is the stage when the patient or the individual will know that there is a loss of memory, decline of cognitive function, but the doctor doesn't know. So if one will be careful at this stage, this preclinical stage may not go to MCI, that is mild cognitive impairment. Otherwise, it will move to mild cognitive impairment, where the doctor can test and can say that there is mild cognitive decline in the person. But it doesn't affect the daily activities. So if it doesn't affect the daily activities, usually people will not take care of this and ignore, or will even hesitate to say that they are suffering from mild cognitive impairment. In that case, the person moves to dementia stage, which occurs in different stages, like mild dementia, moderate, moderately severe, and severe. And this is the stage which can be easily diagnosed by the doctor. And there is no cure for this dementia or dementia disorders, but there are ways that this can be delayed. And when this dementia is severe, dementia itself is a group of diseases which has which is caused because of death of cells, what is called as neurodegeneration, and it shows several symptoms. As you see here, loss of short-term memory in the beginning, then mistakes and confusion, what is also as brain fog, that one will be confused most of the time, will not have clear concept. And
sir, sorry to interrupt you in between. It's
yes.
a request to all participants, kindly do not share anything in between presentation. It's a
That
humble request to all participants, do not share any kind of screen during the presentation. Thank you, sir.
Yeah. So when
sir,
this
I'm just sharing your PPT. You may continue, sir. You may continue.
Yeah. When the dementia is severe, right, this leads to a very common disorder, what is known as Alzheimer's disease. So as you see at the bottom, you have three radio imaging scan of the brain from normal mild cognitive and Alzheimer's patients. And so this, when the person is suffering from the Alzheimer's or dementia, they will have this loss of memory, mistakes, confusion, difficulty in thinking, disorientation. They will not be able to take any decision. They will have mood swings, personality changes. So this entirely changes the personality of the person, and the person loses his or her identity. And there are large number of elderly people who suffer from this disease. And you know, our beloved Prime Minister earlier, Dr. Bajpayee, Atal Bihari Bajpayee, at the end of stage of his life, he was suffering from this Alzheimer's type dementia. So this Alzheimer's dementia, they are the problem of memory and other functions, cognitive functions loss. So we need to understand what is this memory. As I have told you, there are different regions of the brain which is responsible for this cognitive function. And there are different types of memory which are stored in these different regions of the brain. So whenever we get some stimulus, like we are hearing this lecture or we visualize something, so this information is perceived through sense organs, our 5 sense organs, and when this is perceived, this is known as or called as sensory memory. And it can stay just for millisecond. If it is not paid attention, it is lost. But if you can pay attention, this is converted into short-term memory, or what is also known as working memory, which can stay for about a minute or so. And if one doesn't repeat this, rehearse this, this may also be lost. So if you rehearse it or pay more attention to the short-term memory, it is converted into long-term memory, what is known as consolidation. So in short-term memory formation, there is no synthesis of new proteins, but only the existing proteins change. And but in long-term memory, there is synthesis of new proteins. So, so there is synthesis of new proteins, and synaptic changes or connections between the neurons, they change due to exposure to this memory. So this property of the brain is known as synaptic plasticity. This is amazing property that brain keeps on changing its neuronal circuit or neuronal connections based on the exposure to new kind of experience. And there are large number of genes which are involved. They are known as synaptic genes. And they all together make an orchestra or cascade of reactions to form short-term memory and long-term memory. I am not going in details of this cascade, but in brief, you should understand that there is external input in the form of sensory memory or sense information, and at the end we get the behavioral output, what is memory. And in between there are changes in the transcription, translation of this gene, there is change in the interaction of these proteins, there is change in the synaptic plasticity, excitability, and so on, about which we don't bother much. So to understand this synaptic plasticity and the changes which occur in the brain during aging, and particularly in the memory, our lab has been focusing on 2 types of model. One is physiological model of rodents, rats, and mice. So that is known as aging models. And in aging, we see that in old age, there is decline in memory function. So we have generated an amnesic mouse model where young or adult mice, they are injected with scopolamine, which is a derivative of the Thura plant family. And when they are injected with scopolamine, they behave or mimic like old age. So it's very easy to understand the process using amnesic mouse model without waiting for mice to become aged. And whatever results we get from these animal models, we try to check them with dementia patients, which is in collaboration with Institute of Medical Sciences at Banaras Hindu University. So we check the memory, different types of memory. We check different types of chemicals, and one of the very important chemicals which we have found during recent years is HDAC2, that is histone deacetylase 2, which increases in old age. And this affects the acetylation of histone proteins, which constitute the chromatin, that the part of which is a part of gene makeup. And in the memory, as I have told you in the previous slide, there are several genes which are activated. They are known as neuronal immediate early genes. And so these are the studies which are going on in our lab. And and we inhibit or silence this HDAC2, and there is also cofactor SYN3, neuropsin BDAC, and moreover we use herbal extract also to do this kind of reversal of memory in old age. So there are, besides this normal brain aging, there are several other factors which affect this memory decline, like pollution, environmental chemicals, stress, infection, traumatic brain injury, hypoxia, depression, neurodegenerative diseases. There is a bunch of neurodegenerative diseases which can affect this memory. So, and lifestyle more importantly. So I'll be touching this in coming few slides. So you can see this is an example of an American guy, Phineas Gage, which shows that if brain is damaged, then there is loss of memory. So he was a person working for the ministry and his job was to construct the railway track through mountains. So he was exploring mountains to get a way for construction. And one day it so happened that a rod, when he was doing explosion, a rod got through his brain, and frontal region of his brain was damaged. And because of this damage, his entire personality was changed and his memory was lost. So this showed that if brain is damaged, long ago, that if brain is damaged, it will cause mental changes. This is another story of a boy, Henry Molaison, who was learning bicycle riding at age 7 years. And when he met an accident, he got partial seizures after some time, or epilepsy. And to cure this epilepsy, temporal lobotomy was done. The part of the temporal lobe was removed. And during that time, 1926, not very advanced technology was available. So part of hippocampi and amygdala was also lost. And this led to anterograde amnesia, a kind of amnesia. So if brain is damaged, there will be loss of memory. And this shows that if there is pollution, and this is a study from Russia, where they have compared urban and rural population, and they showed that in urban population, if young ones are exposed to this air pollution, then the brain shows some characteristics of Alzheimer's disease later. So this is very serious thing, and it happens in our country also. You know that air pollution, air quality in the cities, this is the picture from Delhi, so it's very bad. And there is no study in our country, but I believe that if there will be a study, it will show that this affects the mental function, this air pollution. Then the radiation. Nowadays, we cannot work without these gadgets like mobile and laptop. And it has been shown that radiation from these gadgets, it prevents the neurogenesis, that is birth of new neurons, which is decrease the brain function or make the brain more vulnerable to get different kind of damage. Next, please. Next, please. So stress, as I have told, this is a very, very vital factor which can damage the brain through shrinkage. And as we know that stress is inevitable, but there are ways to manage this stress. And if it is not managed, it may lead to depression or addiction and other kinds of behavioral changes. Becomes more fast, mental functions including the cognition. Please. Then another problem which is more common in old age particularly is type 2 diabetes and depression. So when a person is suffering from this depression and diabetes, they are more vulnerable to get different diseases, particularly via inflammation. That is, there is swelling the risk to get is this COVID. So there has been lots of studies that how this COVID affects the brain. And you know, there are post-COVID particularly, there are several studies to show that, and this study is a survey from 13 countries, that about 80% of patients who were hospitalized, they show some kind of neurological complications. So earlier there were several studies to show that this coronavirus infects the brain directly, but when they checked the brain of dead COVID patients, they couldn't detect any virus. So it is believed that virus enters the brain through nasal mucosa, which is a part of the, which is connected with the brain. And as we know that most of these COVID patients, they also lose sensation, taste. So this is through nasal mucosa, which is the entry point for brain. So this COVID has great impact on the brain, and this increases the dementia. And so it is expected that in coming years, we may see more patients suffering from this dementia. Then there are ways to recover this memory loss in old age, or to prevent this memory loss in old age. For this, there are several strategies. So as I have explained to you, it's the brain training, yoga, meditation, exercise. So all these things, or all these practices, usually they increase the blood flow to the brain. They help in the birth of new neurons, what is known as neurogenesis. Then the sleep is very important for 6 to 8 hours sleeps. There are several herbs which have been used to boost memory or to boost brain functions. The social interaction is very important. Then, of course, brain boosting mind diet and calorie restricted food. So there are lots of studies, and our laboratory has also shown that young has normal cognition, in old there is cognitive dysfunction, and this is because of genetic mutations, adverse lifestyle, low education, socioeconomic status, and other lifestyle factors. But when these strategies are followed, like healthy lifestyle, exercise, yoga, meditation, training, enriched environment, positive thinking, so all these factors may keep the brain more healthy and cognitive functions more intact. So the take-home message is that brain does change with age. Accordingly, there are changes in the mental function. So this mental decline is common, but cognitive impairment is not inevitable. This can be delayed or it can be prevented. So studies have shown, including our studies on epigenetic modifications, that this memory is not erased, but it becomes inaccessible in old age because of some epigenetic modifications in memory genes. So animal model research shows that memory loss can be recovered. I have not shown you the data, but those who are interested can see the publications. And it is believed that when this animal data will be translated into human beings, this will improve the quality of life in elderly. So the present era with the available techniques is very exciting for research in aging, and I would suggest the young students to take up their career in aging, which is very challenging and rewarding. So the study which has been done on this brain aging from our laboratory has been conducted by large number of PhD students who are working in different institutions of India and abroad in different places. And part of the work was possible because of collaboration with different scientists from India and abroad, including Japan and USA. And I have been fortunate to get good funding from Department of Biotechnology, Department of Science and Technology, Council of Scientific and Industrial Research, Indian Council of Medical Research, UGC, DRDO, and some foreign funding also, which has helped to complete the research work. And thank you very much for listening. I'm sorry that there have been some technical issues because of which we couldn't present it so well, but I think the message has gone that if we are careful, we can keep our brain healthy and active. It is possible to have normal cognition even in old age. And if you have any questions, I'll be happy to take it. And thank you very much for your patience listening to the lecture.
Thank you, Professor Thakur, for such a lucid and enriching talk. And as you were quite visible and audible with each word and sentence that you spoke during your talk, it was really reaching every corner of this hall, and also I understand that those who are linked online, they benefited much from. The entire talk, which revolved around the structure and physiology of brain, especially during aging, and the effects of aging on the functioning of the brain. And when you were talking about the normal decline, and also when there is kind of pathology because of various kinds of diseases that may occur even before aging sets in or during aging. It was really very, very informative. It has enriched our knowledge. When you talked about the Alzheimer's disease, which is on the increasing side with advanced age, and you referred to many kinds of issues that were referred to in between. So I think it must have ignited many questions, many queries in the minds of those listeners. So, so just continuing those queries and questions that have come up in the minds of the listeners. So there is one question from the Honorable Pro Vice Chancellor, Madam Dolly Sinha. She is really interested to find out what kind of herbs that you would like to suggest to improve and maintain the memory in old age. Please.
There are several types of herbs, and we have worked particularly on Brahmi and Ashwagandha. So it has been shown in animal models that if we extract from Brahmi and Ashwagandha, they are fed to the animal, it boosts their memory and helps to retain the memory even in old age. So they help to some extent. And our country is full of such kind of herbal, what is known as Medya Rasayan in Ayurvedic products.
Yeah. Thank
So
you. Thank you.
scientifically it has been shown.
Thank you very much, Professor Thakur.
So, at the end, I thank the organizers for providing me this opportunity to share our research experiences and be part of this international conference. And I wish all the best for the conference. Yes.
Thank you very much. Thank you very much, Professor Thakur.
Yeah.
Any other question from the audience?
Yes, sir. I have a question. I'm Prashovat Malik from RP College, Madhubani. Sir, I am one of your students of the class of 2004. I am a batchmate of your research scholar, Dr. Vijay Barma. I wanted to ask you, sir, that can you brief about the mechanism by which steroid hormones interact with the dominant or cholinergic systems of the in relation to cognitive functions?
These steroid hormone receptors are present in the brain. So the hormone will bind to these receptors, and then there are several co-activators and co-repressors. So they make a complex, and these complex binds to the promoter region of the gene, and then it will inhibit or activate the gene expression. And if these are the genes linked to the memory, then it will affect the memory. And we know that estrogen particularly is neuroprotective. So there has been several studies to show that this can protect the brain, can protect the neuronal cells, it can help in the neurogenesis of the brain. So this is how estrogen protects the brain. And that's why particularly in women, it has been shown that in post-menopausal women, when there is decline in this steroid hormone, there are several neurological complications. So this hormone is very protective, and there have been several studies. You can go through the publications, you will see detailed studies. The brain has the receptor, and steroid hormones act through those receptors. In cholinergic, dopaminergic, all the types of neurons.
Thank you, sir. Thank you very much.
Okay. Thank you, Shashoban. There's a small query, Professor Thakur, which is based on my personal observation of my mother and father as well. Normally it happens in advancing age that people start losing memory of the current time, but old memories, they remain there. Sir, how this is?
How the loss of memory?
Memory which is of the present time, and the older memories, they normally they retain, they remember those things.
Usually, I have told broadly there are two types of memory, short-term memory and long-term memory. So short-term memory is working memory. So when this working memory is consolidated, it becomes long-term memory. So long-term memory is stored in brain cells. So usually, what people have learned in childhood, they will not forget this. So this is long-term memory. But short-term memory, for example, what one has taken in lunch or dinner or whom the person has met. So this working memory or short-term memory, one can easily forget because there is no consolidation, a very poor consolidation. But if, for example, forgetting the names, so this is known as retrieval. So even if you are not able to remember some familiar names, so this is because of the reason that you are not able to retrieve it from your old stock of names. And this is one of the reasons is that you are not practicing that name very frequently, so this has slipped from your mind. So this is very common in old age, but nowadays, because of stress and other lifestyle problems, even people of adult age, say 40, 50 years, they face such kind of memory lapses?
Thank you, Professor Thakur, for your so informative and engaging talk.
Give him a big hand.
And naturally, we give him a big hand for such a basic talk. Thank you very much.
Thank you. All the best for your conference.
Thank you. So before we conclude this inaugural session, this is time for national anthem. I request all the participants over here to please stand.
उधर कट गया ना, उधर कट गया, पीछे कर दो ना कैमरा को।
For the technical session first, we are late for the first technical session. There would be a tea break of just 10 minutes. After all, we assemble here and the first technical session would start.
We have two speakers in this session, Dr. Arijit Chakrabarty and Professor Anil Kumar, respectively. We now like to introduce our first speaker from technical session, Dr. Arijit Chakrabarty, who is postdoctoral fellow from Harvard Medical School, Boston, USA. Dr. Arijit's interest in post-pathogen interaction was developed during his undergraduate years in Banaras Hindu University, when he was introduced to a world of pathogenic disease by his professors.
During the course of his doctoral study, he encountered an enigmatic pathogen, Leishmania donavani. He tried to understand and uncover its intelligent ability to destabilize the host immune system. His work with his colleagues to understand the role of WH signaling in innate immune system led to publication in Frontiers in Immunology and GI Abstract. As a postdoctoral fellow in Dr. Prashant's lab, he is elucidating the molecular mechanism by which an opportunistic pathogen like Pseudomonas or Rhinolosa tries to
यहाँ कैसे लिखा हुआ फेसबुक श्री राम को बोल कहाँ रहे हैं
Okay, can you people hear me now? Hi. Am I audible?
Yes, yes, you are audible, sir.
Okay, okay, okay. Um, Sorry for the technical glitch. Today I'm going to talk about something like a very different kind of story that you in generally hear. Going to talk about Leishmania, a tale of a Trojan horse. Before even I go into the topic, I want to thank all the dignitaries and especially all the people who have put on a very hard work to bring forward this meeting, and especially in these trying times. And it's really an honor for me to talk before you, and it's a privilege to present some of my work that I had done during my PhD times, and that is in India. And this is a very topic, very close to my heart, because it started from BHU, where I tried to think about how wind signaling was working in different diseases, and then slowly I moved forward to a little bit of immunology, and started working on Leishmania. When I started my work, I never had any inkling about what Leishmania was and how Leishmaniasis, what the Leishmaniasis was a disease was like. But as days progressed, I found out that Leishmaniasis is one of the very important but still neglected diseases that has been affecting India, and especially the region of West Bengal and Bihar, which still sees a number of new cases growing every day. And this disease is something which is affecting most of the underdeveloped parts of the world, not only in India, but some places in Brazil. And then we had we had seen diseases occurring in the war-striped Syria and all these places where the economic conditions are very bad. But interestingly, this disease is caused by a zoonotic parasite, which is a unicellular parasite, and it starts into the it gets into the body without showing much much effect. So, but when it goes in, it starts it ravages the body, and it then changes most of the metabolism within the body. So it looks like more like a Trojan horse. And if you people might know about the Trojan horse, uh, it was the story of a war in ancient Greece, and the Greeks tried to overpower Troy. And when they tried to overpower Troy, they had the big breach they had to do was on the walls of Troy, because they were big and they were invincible. So what they did was build up a horse and left without attacking the city. And in that wooden horse, they put in some soldiers. And when they came inside, when the people thought that okay, this horse was um, gift for them, they brought them into the city. And at the mid of the night, the attackers from inside the horse ravaged the city. I have given a pictorial representation of the Trojan horse, and beside it is one of my EM slides which I got during my PhD. And in the electron microscope slide, if you can see here, you can see that just beside the nucleus, there is another small vacuole-like structure. And actually, this is the leishmania. You can see its nucleus and its kinetochore or the kinetoplastids, and there are the flagellar things that you see in general you see on leishmania. So what this leishmania is doing is that, if you can see, it is trying to take over the host metabolism. It's trying to take over the different lipid moieties inside the cell, and trying to kill that cell. So that's why I have said that leishmania is like a Trojan horse. It invades the system, invades the body, and takes over the metabolism. So before I go into the how it takes over the metabolism, why should we even study leishmania Donovani infection? We should study because there is, as I told you, there is emergence of new cases. There is emergence of drug-resistant strains of parasite. So in this case, I should also tell you that leishmania can only be treated by like 2 or 3 drugs, like sodium antibody antimony gluconate. These are the antimonials, and then there is amphotericin B, and basically that is it. And if nowadays whoever is working with leishmania, they have seen that there is emergence of the drug resistant parasite. You give that drug, still there is the parasite growing somewhere in the body. And leishmania has a very beautiful way to evade immune response. So the immune response are the gateways of Troy, and it has enough system because it has evolved throughout ages and tried to build up system where it can bridge these walls, and that's how they can evade the molecular. They have molecules which by which they can like break open the system. And we actually do not know what the susceptibility and how the resistance comes up, and there is unavailability. Unlike COVID, that you had seen that there were proper vaccines that could have been produced, but leishmania has a very big repertoire of membrane proteins which cannot be breached. And even if vaccines are produced, they are mostly not usable. They do not form good antibodies, and there are very few moieties on the leishmania parasite that can actually make proper vaccines. Now, coming to the world perspective, you can see that we are India is the most endemic for visceral leishmaniasis. Leishmaniasis, as you all know, has 3 different forms: the cutaneous form, and then the visceral form, and then the post-Kalajar dermal leishmaniasis form. So of the most difficult one is the visceral leishmaniasis. It's very hard to cure, and it causes a plethora of problems, and it is very hard to be it's not only hard to cure, it's also very hard to detect. It is detected at a very later stage of the disease, and therefore it's kind of a big problem for the to think about in the worldwide perspective. So India is an endemic region where visceral leishmaniasis is seen. Brazil is also another one. Now this is a data which is from 2016, but in 2021, there are endemic regions in Turkey as well as Syria where most of these patients have been found. Now, I have mostly worked within West Bengal and a little bit parts of Uttar Pradesh, Jharkhand, and Bihar to get some of my samples and then see how what are the changes that we see. These are some of my images which I got from different while we were taking blood samples. This is one of the patient Here you can see she's very young, and it was very difficult for us to see her in a very bad condition, although she had been treated with sodium antibody gluconate, and later it was like she recovered from the disease, and we were constantly having backup. We are constantly seeing her so that she doesn't get a relapse of that kind of infection. These are some images from our collaborators from different parts of the world, where you can see that this child is a very young child, and he shows symptoms like hepatomegaly and splenomegaly. And this is a cutaneous form of leishmaniasis, where you can see that the nasal part of of this guy has been totally devastated. So these are big problems with leishmaniasis, and the story of suffering, if you do not go into the into their places, and if you do not see the patients, it would be you won't realize how difficult they are, and how much suffering they see. So this also inspired me to work on the story, and I started thinking about what are the places where I can get into and understand the disease. So first of all, it's a very simple thing that Leishmania donovani is a parasite which is ingested, which has a digenetic life cycle. So it has the procyclic chromastigotes, where there is there are small parasites with flagella, and they remain in the sandfly salivary gland. And when these sandflies come and bite human, it transfers these chromastigotes into the human cells, and from in there they get attached to the phagocyte, where they change themselves into the more virulent form, which are the amastigotes. These amastigotes divide, and after they divide, they burst these cells and come out, and again start infecting other humans through sandflies. These chromastigotes which come in secretes a lot of virulence factors, and these virulence factors are might be in the form of membrane moieties, or called the lipophosphogycans, and they can alter actin cytoskeleton, or they can inhibit the lysosomal fusion of phagosomes. So these are very technical terms. Basically, they are inhibiting the killing potential of the macrophages that we have. So to make it simple, Leishmania donovani has all the virulence factors by which it can suppress the immune system. So taking these into perspective, we still thought that, okay, what are these virulence factors and what are the molecules they are affecting? So somehow in my interest towards Wnt signaling actually made me think that might be this is a molecule which might be affected. So when I went into our PhD lab at that point of time, very few things were known about Wnt signaling, and specifically Wnt5a, which is a non-canonical mediator of Wnt signaling, was being studied in that lab. And we had just seen that when Wnt5a is given to macrophages, it can phagocytose a lot of bacteria. And you can see here in 2 hours in PBS treated macrophages, there is very less bacteria, but with Wnt5a treated macrophages, there is a lot of macrophages that has been engulfed. So, and also we had also worked with the Chadipura virus with Wnt5a, and we could see that Chadipura virus can also be cleared by Wnt5a. Technically speaking, Wnt5a is not a molecule which was specifically seen in immune system, but it was mostly seen in systems where regeneration and cancer and all the other topics except for the immune system. But these were the first inklings that we had, that we could say that, okay, Wnt5a had some role in macrophage phagocytosis. So we thought that might be Leishmania is also affecting Wnt signaling, and thereby it could change the outcome of Leishmania donovani infection. So specifically, Wnt signaling is a very simple thing. It has a number of ligands, and it has its cognate receptors, which are like G-coupled protein, like G-coupled protein receptors. They are not G-coupled, but like G-coupled protein receptors. And when these ligands come and bind to the receptor, they bring about different changes like differentiation, morphogenesis. I'm going a little bit fast in this slide because it's a very we do not need to delve deep into it. Just we need to know that Wnt signaling are actually can signal in form of two signals, like canonical and the non-canonical signals. In the canonical signal, they actually do not degrade beta catenin, and this beta catenin is a nuclear protein which actually helps in transcription. So it goes, the beta catenin does not get degraded, and it goes into the nucleus and starts transcribing. And in the non-canonical part, there are two pathways, which is, one is calcium mediated pathway, which calcium is like a second messenger, and the other pathway is the CDC42 and Rho and RAC pathway, which actually changes the cytoskeleton of the system. So Wnt5a specifically is a non-canonical mediator, and it changes the cytoskeleton milieu of the cells. So I thought that might be this Wnt5a signaling is being affected, and it is like trying to change the macrophage cytoskeleton. Now, let's go a little bit fast with the objectives. We try to see how Leishmania pneumoniae infection was affecting Wnt5a signaling and what are the mechanisms behind it. So before, the first thing what I did was I took sensitive parasites, which are drug sensitive and drug resistant parasites, and infected them with macrophages. And these are like small Western blot images, and you can see that in uninfected there is a huge amount of Wnt5a, but with infected you cannot see that amount of Wnt5a, whereas the beta actin remains same. But when we check the RNA, we do not actually see this amount of differences. So we could say that the Wnt5a that is being degraded is degraded by the parasite, and it's not by the transcription process. We tried to check it with axenic parasite or egressed amastigotes. Axenic parasite means which are axenic amastigotes, which are like forced to be amastigotes in normal culture, and we could see that still it was showing degraded Wnt5a. And when parasites were coming out of the macrophages and we are infecting again the macrophages with these parasites, we could see again there was a degradation of Wnt5a. And this was true, and this degradation was prevented when we used a specific inhibitor for degradation, which is phenanthrolene. And when phenanthrolene was used, this is actually a metalloprotease inhibitor, and we could say specifically that Leishmania pneumoniae was producing some kind of metalloprotease, which were actually degrading the protein. And what was the metalloprotease? The very well-known GP63. GP63 is a small metalloprotease which is produced abundantly by promastigotes and amastigotes, and that were actually changing the Wnt5a milieu in the cells. We got donors from human, and these were like PBMC donors, where we infected the macrophages with Leishmania, and we could see that Wnt5a levels were still going down in these donors. Mind it that I have given a second donor which showed a less amount of degradation, and actually this donor had even less amount of Wnt5a, which also gives us a preview that all human beings are not the same, and the amount of Wnt5a could differ from human to human, and that can lead to the susceptibility towards the disease. Now, when we added exogenously Wnt5a and tried to check the amount of parasite by a parasite specific gene called the amastine, we could see that there is, with 10 nanogram, 25 nanogram, or 100 nanogram, you can see there is complete abolition of Wnt5a. So what was it? The Wnt5a was exogenously added in form of recombinant protein, and that actually led to complete abolition of Wnt5a in both like with different concentrations as well as at different time points. You can see here in Win5A there is less amount of amostin. We did a post treatment also, and we could also again see that the amostin levels were going down. Now comes the age-old images from like we saw it in RNA. Do we get, is it happening? So okay, fine. Let's get some Giemsa micrographs, which is a classical leishmania people's love. And you can see that in Win5A there is very less amount of parasites, as you can see that there are no Giemsa bodies out here in the macrophages, but in PBS there are a lot of these kind of parasites, and through different techniques like confocal microscopy and video micrography, I had seen that this is going down. I would like to just go forward and show this image. This is a video micrography of previous infected macrophages and Win5A infected macrophages. So, and the red thing that you see is the lysosomes of the macrophages, and green are the parasites. So in previous, you can see it doesn't change much, but with Win5A, if you see this parasite, it starts becoming yellow. The lysosome is coming and binding to the phagosomes, and it's starting to degrade. So this was one of the video micrographs which actually gave us enough amount of confidence that actually Win5A is degrading the parasites. Now let's go back to the mechanism behind it. So if we want to go to the mechanism, I would say sorry. So if we start to see the mechanism, so we had to look at the electron micrographs, and if you see the electron micrographs in Win5A treated macrophages, you can see that the parasites are very badly degraded, and these, because of this bad degradation, we could say that the lysosome was coming, and we hypothesized that the cytoskeletal dynamics was a major player in it. And so we started treating the macrophages with Win5A and check the cytoskeletal integrity. And this integrity we were doing with phalloidin macrophages. And this is a flow cytometric measurement. And you can see this red one is the Win5A treated cells, and the PBS is the untreated or the PBS treated macrophages. And you can see there is a distinct shift of the Win5A treated macrophages, suggesting that there was increase in phalloidin staining. One thing is of very importance: this increase is not more than 20%, but it was so significant that it was we could not neglect it. And Win5A was like a small amount of Win5A could bring about big changes. We also did some TARF microscopy. TARF microscopy is a microscopy which actually helps in seeing the basal part of the cells where the actin reorganization complexes are made. And you can see in Win5A treated macrophages there were more these kind of structures like the actin bundles, and these actin bundles were helping the macrophages to kill the parasite. Now when we used an inhibitor of these things, like the inhibitor of cytoskeletal dynamics, RAC1, a RAC1 inhibitor, we could see that these structures were vanishing. So that's why we thought that might be the cytoskeletal dynamics was being changed by Win5A, and that was actually helping the macrophages to play when it was helping the macrophages to kill the parasite. The alteration in actin was also seen to change the NOx activity or the NADPH oxidase activity. we were checking with the help of DCFDA, which is a specific ROS ROS stain, and we could see with Win5A there was an increase. You can see with this red curve is basically there was an increase in ROS, and that was like decreasing the parasite load. And this was inhibitable by DPI, N-acetylcysteine, or RAC1 inhibitor. Now, if we had to check, okay, whether it was really killing the it was actually really inhibiting the killing of bacteria. And when we used 5 macromolar of RAC inhibitor or 5 macromolar of ROC inhibitor, you can see again the parasites were not killed. You can see by the micrographs the parasites were remaining even with the treatment of Win5A. So we could tell in a very certain way that these parasites were actually inhibiting the Win5A, and Win5A being secreted by other macrophages was actually being degraded by the parasite, and it was not allowing the cytoskeletal dynamics. And this was actually changing the killing potential of the macrophages. Basically, the innate immune function of the cells were altered, and the parasites were not being killed. So this was my conclusion from that paper. And then further on, I tried to check it in Win5A like heterozygous mouse. So Win5A heterozygous mouse is our mouse which has one copy of that gene, and it does not have the other copy. It's a part of the copy is deleted. So you can see I had like wild type and heterozygous mouse, and you can see that in heterozygous mouse, where the Win5A levels were much lower than the wild type mouse, the number of internalized parasites were much higher, suggesting that this was a story which we had previously told was right, and like less amount of Win5A was actually helping the parasite to survive within the macrophages. And then I took those macrophages and stained for different lipid distribution with CTB and other dyes like Philippine. And you can see in PBS and Win5A there is more amount of cholesterol stainable lipid rafts, and this might be also playing a major role in like killing of the parasite. So taking in consideration in with all these things, we could say that Win5A was actually plays a very important role in like killing of the parasites. And it was like if Win5A is not there, or if it is in less amount, it was actually helping the parasites to survive within the host. And this is how Win5A actually prevents our Trojan horse, that is the Leishmania, to build a niche within ourselves. And there are further work that is being done by my juniors in the lab, and they have found out that there are more aspects like B cell signaling and T cell signaling which are also being affected because of Win5A. And although the parasites have a very unique ability to degrade this protein, we are also trying to find out newer therapeutics or newer strategies so that we can defeat this parasite. And to end, I should acknowledge my PhD supervisor, Dr. Malini Sen, and there was a big help from Dr. Sumit Chattopadhyay and Dr. Shamal Roy. Our collaborators were like Anjan Das and Dr. Aditya Konar were very instrumental in helping us give different mouse strains like the Win5A mouse strain. Kosta Panda was one of the person who actually motivated me and helped me to get my PhD, and my wife also who was a PhD in from BSU, Dr. Rashmi Richa. She was also a very big help in my during my PhD time. And also Shreyoshi, who is a junior and who is soon to be become a doctorate, had helped me in a lot of experiments in the later part of my study. And I got, at that point of time, I was getting funded from UGC. We had projects from DBT, ICMR, and some institutional funding and instrument support. And this is how I got to the story. And thank you all for being patiently listening to me. And I am open for questions.
I hope the Trojan horse is never able to enter Troy and wreak the havoc that it causes. Now the audience is requested to unmute and ask questions.
Hi Arijit, this is Manish. Thank you for the nice talk. I have a very quick question. So you are telling that there is some cholesterol that is involved in the killing of the parasite. So can you tell me, how this cholesterol is involved in the killing of the parasite? What is the mechanism?
Yeah. So this was like a topic that I started when just before leaving. So this is something we are still investigating, and we were getting some good results from that. So cholesterol actually forms lipid rafts, and these lipid rafts, you can see that with Win5A there is more amount of lipid rafts, and that was actually the results that we got was that the different TLRs were coming and binding in these lipid rafts, and this was helping the macrophages to get activated through its receptors. So somehow the cholesterol was actually helping different receptors and its downstream molecules like MID88 and all these things to get activated. And when these were getting activated, Leishmania parasite was going down. So this was what I saw. I don't know whether they have still
Actually, the reason we are asking this question is, to my surprise, I know that most of the illness and the pathogens use cholesterol as a nutrient source for survival.
Exactly.
Yeah.
Just cholesterol is like a molecule which actually can go from membranes to inside. So there are a lot of transport, retrograde transport, and all these transport that are going on. And I think, but I'm not very sure about it, but in case of Leishmania, the amount of cholesterol goes up because parasite tries to get more cholesterol. But Win5A tries to get the cholesterol out onto the membrane so that it does not get accumulated on the lysosome. If it doesn't get accumulated in the lysosome can function properly. So that's why.
Okay, thank you.
Any other questions?
Back on the
Can you throw more light on how this Leishmania is?
No. How Pseudomonas evade the immune response.
This is evading the immune response. You talked about it, but
That particular molecule.
How particularly it evades the immune response?
Papa.
ठीक है, छोड़ दो, कंफ्यूज।
मैं माइक की तरफ होकर बोलिए ना एक बार, बोलिए ना।
Arijit.
Parul, I cannot hear you.
Okay, Arijit, we would like to know, you throw more light on how it evades the immune response, a little bit more about how it evades the immune response.
How leishmania evades the immune response?
Yes.
Okay, so till now I have been talking only about the cell biology. But if you want to hear about the immune perspective, so basically Leishmania pneumoniae produces more amount of, triggers the macrophages to produce more amount of IL-10 than IL-6. And because of this anti-inflammatory response, Leishmania pneumoniae can build a niche within the macrophages or within the tissue that they grow in. So that's how they try to evade the immune response. These are like very basic findings that have been shown previously, but now new things are also coming up. So new things means there are studies where it has been shown that it is actually affecting different metal proteases that are present within the host cells, and because of that degradation, sometimes you see a lot of parasites growing within the host.
You can just repeat the question. It is that, like malaria, is there any proneness of a particular blood group as regards to infection of this one?
Okay, there is one more question from Nihal sir. He wants to know that like malaria, there is more proneness towards malaria in a particular blood group. So a particular blood group is more affected by malaria. So is same for Leishmania?
To answer these questions, I really do not know, but the things that I have seen, the number of patient samples that I have seen, there is no direct linkage towards those towards different blood groups.
In Leishmania,
it's more
blood group has nothing to do. In malaria, we have that much of specificity. In Leishmania, in Kala-Azar, it is not so.
Leishmania is affecting the white blood cells, and malaria is affecting the red blood cells. So obviously, I do not think that there is any specificity. But truly speaking, I won't be very surprised if someone comes up with that kind of idea. Like, if someone gets something from that.
Thank you, Arijit, for such insightful talk. And we would like to have more of your talks in future. And I request you to please guide our students here so that they will get encouraged and try like we already taught. So thank you very much for joining us.
Thank you, Parul.
जल्दी से, अरे वो अजय चला गया ना, अजय को जाना पड़ा ना। नहीं भाई, अजय had to move. There is some development in university. तो घर गाँव क्या होगा? पता नहीं.
Thank you, sir. Moving further, now we would like to introduce our next speaker, Professor Anil Kumar from Government BYT PG Autonomous College, Durg, Chhattisgarh. He established postgraduate diploma in clinical pathology and diagnostic technology and acted as course coordinator for 5 years.
He is acting as program coordinator for Star College program of Department of Biotechnology and also acting as nodal officer for All India Survey and Higher Education at Government BYTPG Autonomous College, Durg, India. Besides, he is also nodal officer for Chhatra Jeevan Chhattisgarh program of Department of Higher Education and CHIPS Chhattisgarh, India. In addition, he is acting as chairman of studies for Clinton Yadav University, Durg, India.
Professor Anil is acting as member of Academic Council of Clinton Yadav University He is member of National Resource Center for Education under Pandit Madan Mohan Malviya National Mission on Teachers and Teaching. He is presently working on problems related to human health, especially on monogenetic disease. Over to Dr. Anil sir to give detailed information on the topic sickle cell anemia and G6PD deficiency, a societal problem for genetic alteration.
बहुत अच्छा टॉक है.
म्यूट में है, अनम्यूट चले जा. म्यूट में है सर.
Good morning, good afternoon, everybody. Sir, I want to know that my slide is visible or not.
Yeah, you are audible. You are audible.
Yes, the slide.
And my slide is visible or not, sir?
The slide is yeah, now it is visible. Your slides are visible. Anil.
Is it moving?
No.
Not moving.
Yeah.
It is the second slide, sir. It is moving or not?
No, it's moving now.
Yes.
It's moving now. Go ahead.
Okay. So, good afternoon, everybody. I'm very happy to participate in this E-International Conference on the topic, the recent advances in life sciences with references to the disease, disorders, and adaptations, which is being organized by my parental department, the Department of Zoology of LNMU University Darbhanga. Respected Chief Patron, Honorable Vice Chancellor, Professor S.P. Singh, Patron, Honorable Pro Vice Chancellor, Professor Dolly Sinha, Co-Patron, and Registrar, Professor M. Ahmad, President and HOD, my teacher, Professor B.S. Shah, Convener, Professor M. Nehal, Co-Convener, Professor A.N. Shah, Organizing Secretary, Dr. Parul Banerjee, and Technical Session members, Professor B. Singh and Dr. S.K. Verma, and all other members of the organizing committee. First of all, I would like to express my sincere thanks to my all teachers of the department. I'm happy that I am alumni of the organizing department, and today my own department has given me opportunity to participate in this conference. Even after long gap, I am deeply associated with the department, and my every teachers are in my mind. So again, I express my gratitude to my all teachers. Sir, today I'm going to describe about one very major problem of our society, the sickle cell anemia and glucose 6-phosphate deficiency. So the topic of my lecture is the sickle cell anemia and glucose 6-phosphate deficiency, a societal problem of genetic alterations. Actually, what is sickle cell anemia? In normal condition, our RBC is almost oval and flexible. So its movement is very smooth inside the blood vessels, and a lot of other factors are there which are helping the frequent movement of hemoglobin inside the RBC inside the blood vessels. But in this case, in the sickle cell anemia, the normal RBC get torted and it becomes sickle-like structure due to some genetic alterations, which creates a lot of problem. And under sickle conditions, it becomes very rigid and sticky, get attached with the internal wall of the blood vessels, coagulate there, and interrupt the flow of the blood inside the vessels, causes hypoxia and other problems, and lot of strokes and other diseases, other pathogenic effects. There is a very long history. First of all, in 1904, the Walter Noel, he was a student of the dental surgery. In Walter Noel, first of all, one symptoms was reported of the sickle cell anemia, like the respiratory distress and ulcer in the leg. And his professor, Iron, he first time recognized that there is one elongated sickle blood cells, sickle-like RBC cells. In 1910, Dr. James Herrick has credit to document this problem first time, and he has recorded this pathogenic problem first time in the Western medicine. Professor Dr. Noel finally returned back after completion of his study to original country, to his native country. And just on the age of the 32, he passed away, died due to this problem. In 1922, the Mason, Dr. V.R. Mason, first time he recognized and named
this problem as a sickle cell anemia disease. And thereafter, a lot of problem again appeared. But one major study is in 1915, the Meyer reported the same problem in 21-year-old lady, and they found that there is an inheritance pattern of the disease in the sibling of the sufferer women. And after that, it was established that this is a genetic problem. What is today's condition of the sickle cell anemia in the world? Mostly it is reported from the African continent, but also from the Indian continent. And in African continent, sickle cell anemia, particularly in the West and East Africa, it has been reported in large numbers. In India also, and but in the Nigeria, near about 25% populations are suffering from the same problem. Here, one important point is near about 5% of the world populations carry gene responsible for hemoglobinopathies. And each year about 3 lakhs infants are born with major hemoglobin disorders, including more than 2 lakhs cases of sickle cell anemia in Africa alone. In our India, sickle cell anemia is found in particular pocket, but thalassemia is more common than the sickle cell anemia, which is also a similar genetic abnormality of our blood cells. In some area of the sub-Sahara Africa, up to 2% of the children are born with the condition. In broad term, the prevalence of the sickle cell trait ranges between the 10% and 40% across equatorial Africa, and decreases to between the 1% and 2% in the North African coast. So in Western African countries such as the Ghana and Nigeria, the frequency of the trait is 15% to 30%, whereas in Uganda, it shows marked tribal variations reaching up to the 45% among the Bamba tribes in the west of the country. In our India, this is the most prevalent zone for the sickle cell anemia, which covers some part of the Rajasthan, Gujarat, Madhya Pradesh, Maharashtra, and Chhattisgarh, some parts of the Telangana and Odisha. And still it is not reported from our Bihar and other northern part of the country. But another similar disorder, the HBE disorder, has been reported from the northeastern part of the country. Actually, this is the normal shape of the RBC, and inside one RBC, near about 2.5 to 4.5 million hemoglobin molecules are there. The normal hemoglobins are HbA, HbA2, HbA, but abnormal hemoglobins are referred as the HbS, HbC, and HbE. This is the genetic pattern of our hemoglobin. Under normal beta hemoglobin, the glutamic acid and glutamic acid, and in the HbS conditions, the valine and glutamic acid, one changes takes place here. Here it is visible. In this slide, you can see that in normal case, there is a glutamic acid, but at chromosome number 11, at 6th amino acid level of the beta chain of the globulin proteins, one mutation takes place. And due to this mutations, the glutamic acid is replaced by the valine. And due to this single mutations, the normal hemoglobin gets torted, and a clump of the hemoglobin appears, which causes a lot of pathogenicity. Here, this is the configuration of the normal hemoglobin, and this is the torted hemoglobin. Hemoglobin is meaning the sickle cell anemia hemoglobin. And this is normal inheritance pattern because it is genetic, so it follows the Mendelian inheritance pattern. And in our society, in India, there is some sorts of the sanguinity due to caste-based society. In our India, it is practiced that normally people prefer to get married within the caste system. And in some of the populations, it is very confined. So sanguinity level is very high. In that particular populations, the sickle cell anemia-like problem get accumulated. And what are the symptoms? There are some major symptoms. As a result of the sickling and the premature aging of the red blood cells takes place. Under normal conditions, the life of the RBC is near about 120 days, 100 to 120 days. But in the case of the sickle cell anemia, it varies 40 to 50 or maximum 60 days. So this is one major cause of the anemia. Thereafter, it increases the risk of the severe infection, like bacterial infections and other related problems. Splenic sequestration is a bigger problem, and sometimes it become life-threatening. Spleen swelled up very large, in very large in size, and sometimes it become life-threatening. And due to interrupted flow of the RBC inside the blood vessels, generally it coagulates with the internal wall of the blood vessels, causes interruption in the free flow of the blood, and finally results in the stroke, seizure, weakness, ulcer in the leg, and other problems, respiratory problem also. In the children, the sickle cell anemia experiences slowed growth and delayed maturations, including the puberty as a result of the anemia and infections. There are repeated painful episodes called vaso-occlusive crisis, a big problem for the sickle cell patients. They are generally facing these problems, and due to this, the life quality of the sickle cell patients is very poor. And finally, inflammations appear in the extremities of the body. And complications and disassociate the sickle cell anemia, like the splenic infarctions at high altitude with exercise or with hypoxia due to poor supply of the oxygen, and the isothenuria with the loss of maximal renal concentrating ability, hematuria, secondary or renal papillary necrosis, fatal exertional heat illness with exercise, and sudden idiopathic death and exercise, glaucoma, recurrent hyphema following a first episode of the hyphema, and bacteria in the omen, and bacteria or polynephritis associated with the pregnancy, renal medullary carcinoma, and early onset of the end-stage renal disorder from the autosomal dominant polycystic kidney problem. There is one study that why the sickle cell anemia, like mutation occurred in the populations. And the study is probably, the reason is the malarial parasite, especially the Plasmodium falciparum. In our Chhattisgarh, the Plasmodium falciparum is a serious problem. I do not know what is condition of Bihar at present, but when I was there, the Plasmodium vivax was the problem. Plasmodium falciparum was not major problem, but it is very long gap now. And Plasmodium falciparum is especially found responsible for these mutations. The story is probably the parasite when try to enter inside the RBC in erythrocytic cycle stage of the malaria. For the protections from the parasite, the mutations occur as a natural selection process, and that's why in the sickle cell patients, the Plasmodium falciparum or any malarial parasites do not survive. And but the result is the another problem, the sickle cell like conditions and poor quality of the hemoglobin and poor supply of the oxygen and other related problems. But there are some contradictions also. But still, the large number of researchers believes that the invasion of the Plasmodium falciparum is one of the major reasons for this mutation and this problem in the society. There are three hypotheses, three evidence behind this hypothesis. One is the geographical correlations, second one is the epidemiological association, and third is the biochemical studies. So these three parameters proves and gives some evidence that probably malarial parasite is responsible for this mutation and this genetic problem. Here the malarial belt extends across the southern Mediterranean, sub-Saharan Africa, and Middle East India, Southwest and island of the Southeast Asia and northern Australia. And in all areas, sickle cell anemia prevailing up to the 10% of the population, up to the 10% of the populations. In the Mediterranean, thalassemia and G6PD are elevated, while in India and Southeast Asia, hemoglobin E, S, and thalassemia all are predominant. In epidemiological support are also here, and similarly, the biochemical evidences are also in support of this theory. Here, there is another one of the major theory, the nitric oxide, role of the nitric oxide in the sickling. Actually, in our blood vessels, normally nitric oxide is being produced by the internal cell of the blood vessels. And this nitric oxide ensures the smooth movement of the RBC inside the blood vessels. It also ensures the prevent the aggregations of the platelets inside the blood vessels. So for the normal smooth flow of the blood inside the blood vessels, nitric oxide is being produced by internal wall of the blood vessels. But in the case of sickle cell anemia, due to reduced life cycle, a large quantity of hemoglobin comes out from the ruptured RBC. And this large quantity of the hemoglobin absorbs RBC. absorbs nitric oxide produced by the internal wall of the blood vessels. And due to lack of the nitric oxides, the blood vessels fails to ensure the smooth flow of the blood inside the vessels, and finally it creates a lot of problems. Some of the researchers have detected one particular gene, identified one particular gene, the NOS2 gene, and nitric oxide synthetase 2 genes, and found that in the case of the sickle cell anemia, both genes have impaired. So this figure indicates that this is the RBC, and when it breaks down the RBC, it releases large quantity of the hemoglobin inside the blood vessels, which absorbs nitric oxide secreted from the blood vessels. So nitric oxide level decreases, and finally it creates blood vessels susceptible to the damage, and a lot of problems, related pathogenic problems appears here. Now, our medical fraternity treats the sickle cell anemia up to some extent, although there is no perfect treatment for the sickle cell anemia, but they are using the hydroxyurea. And because there are some evidence that hydroxyurea is helpful in the case of the anemic monkey and the sickle patients of the human also, which produces the HbF, the fetal hemoglobin, and fetal hemoglobin having the high quantity of oxygen absorptions. So it is helpful, definitely in our society also. Doctors are frequently using the hydroxyurea. It reduces some problem, and up to some extent it increases the quality of life, but this is not remedy. The genetic alterations cannot be reversed. To reduce the pain, cannabis is being used by a lot of physicians, but the vaso-occlusive crisis is very problematic for the sickle patients. The pain sometimes appear up to very high extent, and patients suffers badly. So under such conditions, the doctors prefer to use the cannabis also to reduce the vaso-occlusive pain. Platelet inhibition is another problem. Because there is a tendency in the sickle patients that platelet cells accumulates together, and finally it causes the development of the clot inside the blood vessels. So salicylic acid and salicylic acid derivatives like compounds are being used to reduce the platelet accumulations. Very recently, arginine and glutamines are also being used to help the sickle patients, and it is evident that the arginine and glutamine like compounds are helpful to reduce the pain, and it increases up to some extent the flow of the blood inside the blood vessels. Yes, CRISPR-Cas9 technology, definitely I am hopeful that CRISPR-Cas9 technology may be helpful for the gene editing, and there is only one report from the USA that in one patient, he has voluntarily offered the CRISPR-Cas9 applications, and after applying the CRISPR-Cas9 applications, this CRISPR-Cas9 technology in a mass of the populations, because it is genetical, it flows by the following the Mendelian inheritance, so it is not possible to apply on the mass of the populations. Blood transfusion is one of the factor. There are two types of the sickle cell anemia: the homozygous condition and heterozygous condition. This heterozygous condition is known as HbAs. A is the normal hemoglobin, S is the sickle hemoglobin, and homozygous is the HbSS. So in heterozygous conditions, we can give some relief. We can help patients to improve the quality of life, and we can elongate the life of the patients up to the 50-60 years also, with some pain and some problem. But in homozygous condition, it is very difficult to protect the patients, and normally up to the age of the 35, normally the patients do not survive. But the blood transfusion is one of the major helping factor. After 15 days, if you can transfuse the blood, then the longevity of the patients may be increased. Bone marrow transplantation is also possible after 16 years of the age, but again, this is not feasible for mass of the populations, and it's very specific and very costly and very difficult to apply all the patients. So this is not the remedy. Still, we do not have any remedy to control the sickle cell anemia. We have only one thing to check the Mendelian inheritance, and by checking the Mendelian inheritance in the society, can curb this problem. We have also performed some work. Now I am going to present what we have contributed, our lab have contributed to this problem, to reduce this problem. First, what is the hemoglobinopathies and why we have selected this problem, the justification of our work. In Chhattisgarh state, the Chhattisgarh state is malaria-prone region, and a large number of the communities are found with the sickle cell anemia. It is believed that HBS is an adaptation against the malaria and thus selected in the malaria-prone zone. It has also been reported that the malaria parasite has tempted RBC for protection with the sickling is one of them. So in our region, in our Chhattisgarh state, the Plasmodium falciparum is very serious problem, particularly in the Bastar region, in the Naxalite belt. And in the Lancet, it was already published that within 1 year in the forest of the Bastar, where about 2 lakhs people have died due to the Plasmodium falciparum infections. And similarly, in this zone, the sickle cell anemia is also prevailing in very large quantity. That's why we have considered this problem. And here I'm going to present the result of work which we have carried out for the 2 districts, the Durg district and the Rajnandgaon district. And for that we have considered 3 populations, the tribal population, Kurmi population and Sahu population of our society. And in the case of the sickle cell anemia, the free radicals generation is very common due to oxidative stress and due to hypoxia, and due to large quantity of production of the free radicals, free radical oxygens, the atomic oxygen, which attacks on the DNA and the protein and the carbohydrate of the cell and causes degeneration of the cells and denaturation of the nucleic acid also, and which results the early deformity of the life and early death of the life. So for the protection of the free radicals, there are one gene, the GST gene family in our body. So in this experiment, in this work, we have tried to identify the GST gene polymorphism, frequency of the GST gene polymorphism in our society. So this is the objectives, the study of sickle cell anemia among a target population, the study of oxidative stress in terms of the various enzymatic parameters of the DNA damage among the sickle cell patients, polymorphic study of the GST gene in sickle cell patient, and to study association between the all selected parameters. Here, we have selected population from the Durg and Rajnandgaon, considered tribal Kurmi and Sahu, followed the ICMR institutional ethics guideline. Firstly, we have prepared the slide to detect the sickle cell anemia and used 2% sodium metabisulfite slide process, and thereafter in the confirmed cases we performed the electrophoresis also, and we have analyzed the oxidative stress among the sickle patients considering the GSH, MDA, SOD, catalase, and GPX. These are some photographs. We are moving inside the village, going out inside the village and mobilizing the populations, taking help of the panchayat and collecting samples. So this one figure is from my department. The rest all are from the villages. From the village, we are collecting the samples here. Our research scholars, and we are also involving the MSc students, MSc final year students. They all are moving inside the village. For that previous, we are taking pre-consent of the panchayat and local government, and thereafter we are collecting the samples. This is also photographs from the school. We are collecting samples from the students, also young generation and old generation from every corner. And this is the primer of the GST gene analysis for the GST M1 and GST. There are 8 families of the GST gene, of which we have considered GST M1, GST1, and beta globin gene. This is the PCR condition. We performed RT-PCR, and this is the PCR conditions. And the finding, this is the cell, sir. This is RBC cells, but it looks like sickle shaped. This is not normal. This is not normal RBC. This is the RBC of the sickle patients. You can see that how much the RBC cells have changed here in these slides. How it is possible to deliver oxygen by such type of cells. So we have practically seen that the such type of RBC very frequently releases oxygen and do not carry oxygen up to the target cells. And after conformation, we perform the electrophoresis. And in the electrophoresis, you can see that there is one SS. This is the number, 47 number is the SS, meaning the homozygous conditions of the hemoglobin sickle problem, and 48 is the AS, meaning partially the sickled and partially the normals, and the rest are the normal. This is one the normal hemoglobin. So by after confirmation in the slide test, we are performing the electrophoresis, and only after confirmation by the electrophoresis, we are keeping sample report to the panchayat, and directly we are going to the village and giving counseling to the concerned patients that what type of care they have to take. And in the sickle cell patients, from the Durg district, we have tested 983 individuals. This is presentation of one very small case. Up to till now, we have detected 15,000 patients from our society. We have database for the 15,000 patients. And out of 983, total 92 individuals have been reported with the sickle, of which 28% from the tribe, particularly two tribe, Halwa and Gond, 22% from the Kurmi, and 42% from the Teli. This percent has been calculated from the 92 individuals, which was reported sickle positive from 983. From Rajnandgaon district again, out of 767 samples, 52 individuals were found sickle positive, of which 19% positive from the tribes, 8% from the Kurmi, and 25% the Sahu community. And the result of oxidative stress is we consider 4 or 5 parameters: the MDA, GSH, SOD, catalase, and GPS. And for that, from all populations, we categorize the populations into 3 age groups: 10 to 20, 20 to 30, and 30 to 40 years of the age group. And more or less, the finding was same. In almost all cases, MDA was found increased, and rest all parameters, GSH, SOD, catalase, and GPX were found decreased, which confirms the oxidative stress, serious oxidative stress in the patients. So this is for the data for the 20 to 30 year of age group. This is for 30 to 40 year age group. And now from the Kurmi community, 10 to 20 year, 20 to 30 year, 30 to 40 year, and again for the Sahu population, 10 to 20, 20 to 30, 30 to 40 years. So definitely the sickle populations are suffering from the oxidative stress. The free radical oxygen generation is very high, which attacks on the DNA and the lipid and protein of the cell and causes damage, serious damage to the cellular system of the body, which is responsible for the gradual degradation, gradual dysfunction of the organ, and finally reduction of the life and death. And result of oxidative stress are being examined, verified by the COMET assay. We have also performed the COMET assay to examine the genetic distortions, and definitely we found that in the sickle patients, the comet-like the tail was clearly visible. This is the broken DNA, and this part is the intact DNA. So after COMET assay, this is single cell electrophoresis in high pH, 13 pH. And thereafter, by the help of the software, we have analyzed the image. And this is from the Kurmi populations. This is the COMET assay verifications. And this is from the Sahu populations, the COMET assay. By the COMET assay, from all 3 populations, we confirmed that there is a DNA damage due to oxidative stress among the populations. Then, gene frequency, yes, we have performed HBAA, HBAS, and HBSS gene analysis. And here, in this slide, you can see that all genes, the GST-M1 gene, GSTT1 gene, and beta-globin gene, all genes have expressed very nicely. So gene expressions, the GST gene family expressions have been also performed. And here we performed the gel electrophoresis of the PCR product, and we confirmed that the GSTT1 gene, beta-globin gene, and GST-M1 gene here. Again, in this next slide also, the GSTT1 gene, beta-globin gene, and GST-M1 gene. And finally, this chart shows that in the normal populations, the GST-M1 gene was found up to the 93%, expressive, and in HbA's populations, the heterozygous conditions, it is 40%, and HbS it's reduced to the 36.66%. So under normal conditions, the GST-M1 gene was expressive up to the 93%. In heterozygous, it reduced to the 40%. And in homozygous, it reduced to the 36%. So how they can counter the free radicals? It is not possible for the HbSS and HbA if the gene fails to express itself properly. Here GSTT1, first one was the GST-M1 and second is the GSTT1 gene. The condition is almost same. In the normal condition, it was 86.6%, HbA is 53.3%, and in HbSS 23.33%, very reduced. So finally, we are giving this horoscope to the society. We are moving inside the society and providing this horoscope and giving counseling to the populations that before taking decision about the marriage, what they should consider. Because in societies, in particularly in some caste, in some populations, although we have reported from diversified populations, we have also reported it, the case from the Brahman populations. But normally in our place, in our Chhattisgarh, it is most prevalent in the tribe, Sahu community, and Chandrakar community. So we are giving this, providing this horoscope to the populations that before taking decision of the marriage, you have to consider this horoscope. Otherwise, the lineage of the family may be adversely affected. And I'm happy that in our Chhattisgarh state, the populations are very much supporting. When we are moving inside the society, the panchayat is cooperating us. Even people, after some counseling, they are very cooperative. An unfortunate point is, in our Chhattisgarh state, as per the state government report, near about 17%, 15% populations are suffering from the such type of disease. But in our experiment, we found no, not up to 17 or 18%. Near about 7 to 8% populations are suffering from this disease. This is my database. This is based on our 15,000 analysis. And we have reported maximum prevalence from the area of our present chief minister, and also very high prevalence from the area of our ex-chief minister. So from both area of the both chief ministers, the prevalence rate is very high. Now I want to draw attention of the audience regarding another problem, the glucose 6-phosphate dehydrogenase deficiency. This is also one of the serious problem. The glucose 6-phosphate dehydrogenase deficiency. This glucose 6-phosphate dehydrogenase is very essential for maintenance of different cellular process. Particularly, this is responsible for the first step of the pentose phosphate pathway and essential for the production of NADPH. And this NADPH is required to prevent and regenerate the reduced form of the glutathione. And this glutathione plays an important role to prevent oxidative damage of the RBC by H2O2. So definitely the glucose 6-phosphate is one of the important enzyme, glucose 6-phosphate dehydrogenase for proper maintenance of the metabolic process. But unfortunately, some mutations has been reported from for this enzyme also. This is actually enzymopathies, but for this on the X chromosomes, there are one mutations. The gene responsible for this enzyme is of 18 kb, and we have reported mutation from our society for this gene also. Here, due to deficiency of this enzyme, the early hemolysis takes place inside blood vessels. The hemolysis takes place, which creates serious problem. And there are so many drugs which can stimulate the hemolysis in absence of the glucose 6-phosphate dehydrogenase deficiency. If persons are unknown that they are suffering from the glucose 6-phosphate dehydrogenase deficiency, then under influence of several drugs, they may suffer. So according to severity of disease, the glucose 6-phosphate dehydrogenase deficiency, there are 3 variants. The class 1 mutation associated with the chronic non-spherocytic anemia. Class 2 mutation is Mediterranean, more severe, and in that case J6PD enzymes shows normal stability but very low activity in all RBC. And class 3 mutations, the J6PD group A moderate, it creates moderate problems, but some non-hemolytic abnormalities have been also reported in association of the G6PD deficiency, like the cataract, reduced mental ability, and coronary artery disease. Actually, the gene is found on the X chromosome. In the male, only one X chromosome is found. In the female, two X chromosomes are found. So in the male, such type of problem is more prevalent. And you can associate that cataract and reduced coronary artery disease. These two particular diseases are more prevalent in the male also. So there is some correlations, although I'm not claiming because I have not performed a specific study on this line. But it is correct that the gene is found on the X chromosomes. So due to only one X chromosomes, male are worse sufferer than the female. For that, again, we have collected sample from the society and performed the methemoglobin reduction test. In methemoglobin reduction test, this part and this part, which are decolorized part, are confirmed cases of the G6PD, glucose 6-phosphatidylinositol deficiency. Thereafter, we have analyzed several genes for the glucose 6-phosphatidylinositol deficiency from our society, and we detected three important genes from our society: the Mahidol, Union, and Chinese variant from Chhattisgarh society. We have submitted this paper to the hemoglobin of the Taylor and Francis, and reviewer finally declined to accept our result that the Mahidol, Union, and Chinese variants may be reported from the Chhattisgarh. They have suggested me that in Chhattisgarh there may be Keralian variant, not the Mahidol, Union, and Chinese, and they have suggested that repeat the experiment. We have repeated our experiment several times, and every time we found these three new variants of the gene Mahidol, Indian, and Chinese from our society. So first time we have reported these three variants of the gene from our society for the G6PD. And thereafter, this is the PCR product, electrophoresis, the single band for the normal, for the Mahidol variant, and two or more bands for the mutated variants. And in this case, for the Union variants, again, 3 bands the normal and 1 or 2 bands are the mutated. And again, then for the Chinese variant, single bands are the normal and 2 or more bands are the mutated variants. Then we submitted our result to the Gene Elsevier journal, and here this paper we got overwhelming response from different parts of the world after publication of this paper in the Gene. And very recently, we have signed one MOU for the whole genome analysis with the South Korea for the populations. This is another population, and this is under consideration at present in the mutation research. This paper is under considerations. So this is all about regarding the genetic problem of the society, the glucose 6-phosphate dehydrogenase deficiency and the sickle cell anemia. This is my final submission before the audience. And I'm again happy to present my work before the dais of the my parental department. And definitely I want to extend my work to Bihar also. I'm in search of the partner, and I'm sure that there is possible that there is no sickle cell anemia in the Bihar, but I'm confident that glucose 6-phosphate dehydrogenase deficiency like problem we may detect from the population of the Bihar. So I request one platform from our parental department for some for collaborative work of this genetic problem to the society. So this is my final submission, sir. Thank you.
In the real sense of the word, and we would really like to collaborate, and we have got many appreciation for you in the YouTube. Many people have written very good and informative sessions, sir. Very informative session. We have gained a lot of knowledge from this session, and if there is any question, I would ask the audience to unmute and then put forth their question.
So, any question?
So, sir, I would like to ask you that this sickle cell anemia and G6PD deficiency both have a relationship with malaria. Like sickle cell anemia is known to be protective against malaria because the malarial cannot survive. So what is the trade-off? Means is sickle cell is malaria so dangerous that this sickle cell anemia is selected in a population to protect the population where malaria is prevalent? Like, maybe you please throw light on this.
Dr. Parul, your voice is not very clear, but I got some points. And there is one hypothesis that Plasmodium falciparum, particularly the malaria, has induced this mutation from the sickle cell anemia. But some contradictions are also coming from different parts of the world. So we are not sure, but there are some evidence also. Plasmodium falciparum definitely very dangerous, because it directly invades our cerebrum. It goes to the in our brain, and it causes very fast damage to our brain cells, and certain in instruction of days, 10 days or 15 days, which is evidence from our society of the tradition, particularly from the West. Sickle cell anemia. In the case of the sickle cell anemia, the problem is lifelong problem. If the patient is suffering from HBSS, meaning homozygous condition, then in adult active life it is very difficult to cope up with the problem of the life for such patients. If he is getting blood transfusion frequently, then he may continue for a long period, but it is not possible for everyone to receive the blood transfusion. But in the case of heterozygous, by supportive medicine, you can prolong the life. We are working just to improve the life conditions of the life, quality of the life of the patients. Even we are also giving support by the help of the scientific research to the homozygous patients also. Still, we do not have any particular scientific device to protect the life of the homozygous condition, but in heterozygous condition it is supportive. The problem is that if the patient is suffering from heterozygous or the homozygous, after active life, he is unable to exercise more and more. He is unable to do work more and more. So finally life becomes futile. So medical science is just helping the populations, but we can't cure the populations. We do not have any medical science, we do not have any medical knowledge to cure the populations. We have only one thing, that is the counseling. If patients are suffering from the HBSS, before taking decision of the marriage, they should first go for testing of the sickling, and if the partner, the would-be partner, is suffering from the sickling, they should take decision properly. This is only way we have to check the contagion inheritance from our society. Otherwise, due to sanguinity problem, the caste system problem in our India, still we are very inclined to the marriage inside the caste. There are some exceptions, but by and large, we are very inclined to be married inside the caste. So it is very difficult to protect the problem, and the particular pocket or particular community are going to be suffer very much. So this is final result.
Thank you, sir. Let's hope that in future we have the CRISPR gene editing way of we could do that gene editing, and we could improve the lives of people suffering from this sickle cell anemia. It was a very, very informative lecture. I thank both the speakers of this session for enlightening us, and it was a very, very interesting session, and we have got many, many comments in YouTube. I am just, yes sir? Okay sir.
That is regarding to small question. Basic, right? Yes, Dr. Anil, this is Nehal.
Yes, it is, sir. I know you, sir.
It's a very basic thing that came to my mind, and since we have worked on our disease, as we already know that there is something called erythropoietin released from the kidney cortex, and that actually does play a role in erythropoiesis of the lot of what we call stem cell, hematopoietic cell. So in case of those who are going to suffer from sickle cell anemia, what role does it play? I mean, this effect of erythropoietin on cellular differentiation that leads to formation of RBC.
Sir, can you repeat your question because voice is not
Sir.
clear.
Oh,
Sir.
okay. This is actually a straight, my query is a straight, any possible link that you can throw light on, that role of erythropoietin that plays in erythropoiesis while acting on the hematopoietic cells towards formation of RBCs. So in case of sickle cell anemia, the release of erythropoietin from the kidney cortex and its action on hematopoietic cells only on those which are going to become RBCs. So is there any effect in such conditions? Means production of RBCs?
Sir, the problem lies with the gene. So erythropoiesis is the result of the genetic expression. So I do not feel that any interference with the erythropoiesis process may be helpful. But I have not read any such type of publications from any part of the world.
Anil, just small.
The consideration for
Anil,
further work.
yeah, Anil, actually a small interruption is that it means that what you are saying, there is no change in the total production of RBCs, meaning thereby as many RBCs were being cellular differentiated, there is no difference in normal condition, in anemic conditions.
No, there will be difference.
Yes or no?
No. For the RBC production, if there is mutation in the gene, so definitely the result will be the same.
No, Anil, I interrupt. What happens in sickle cell anemia, the production of RBC goes down.
Yes.
It goes down. That is what you wanted to know.
Sir, just 1 minute. The production of RBC is not going down. The life cycle of the RBC has been reduced. The normal life cycle of the RBC is 100 to 120 days, but in the sickle cell anemia it survives only up to the 40 to 60 days. But the rate of productions has not been affected due to this genetic mutation.
Anyway. Thank you.
Thank you.
Okay, sir. Thank
you. You have a lot of appreciation on YouTube. Speak various delegates. They have appreciated it like anything. So once again, thanks and blessings.
Thank you, sir. Thank you.
Next
Arun, this section.
अरे इनको असल में कोरिलेट अभी नहीं आई ही है ना मालूम नहीं जाओ
So this was a very fruitful session. I thank both the speakers, Dr. Arijit and Professor Anil Kumar, for enlightening us with their talks. And we are hoping for future collaborations. And let's hope that we have more chances of calling them physically for conferences. And there is a great exchange of ideas amongst us and their university. We are also willing to collaborate. This session closes with Professor Anil Kumar's talk. We'll break for lunch and we'll join again after an hour. That is, we'll be meeting again.
Get it for just 30 minutes because why to make late?
Okay, so we break for lunch for 30 minutes. We'll be joining again at 3:45.
Thank you.
Session. I give the mic to my students, Nidhi and Natasha, who will be comparing this session.
So this is the second technical session and the theme of this session is a host-pathogen dynamics, which is going to be a very interesting and exciting one. The chairperson of this session is Professor S.K. Verma sir, who is a renowned fish biologist, and I would like to request sir to please have his chair. The session would be reported by Mr. Kalicharan Mishra sir, so I would like to request sir to please have his seat. In this session, we have two invited speakers with us. Dr. Manish Kumar from National Institute of Health, NIH, from Maryland, USA, and Dr. Vijay Singh from Harvard Medical School.
Hello.
So now, our first speaker of this second technical session is Dr. Manish Kumar, who is working in the National Institute of Health, USA since 2019 as a postdoctoral fellow with Dr. Nihal Alton Bonnet on enteric viruses to identify their mode of transmission, replication, and survival strategies. He obtained PhD from the University of Calcutta in 2019, for which he worked in the Bose Institute with Dr. Jyoti Basu. During his PhD, he studied host Mycobacterium tuberculosis interaction and identified the role of microRNA and transcription factors in the survival of a bacteria inside the host. He has a 14 publication in a high-impact journals like Cell, Host and Microbes, Molecular Cell, and PLOS Pathogen with an average citation of 70. Now, I would like to invite Dr. Manish Kumar to put some light on theme, molecular and cellular basis of a pathogen survival inside host.
Thank you for the nice introduction. Now what is going on? I cannot see any PowerPoint option here. How to do that? I mean, there is option of window entire screen.
Both are window entire screen and window दोनों where से
अच्छा window okay, yes, perfect. Yes, I think. So can you see my screen?
Yes.
And it's moving as well, right? Okay, perfect. So good evening, everybody. So first of all, I'd like to congratulate every member of this organizing committee to put their all the efforts from last several months, I know, to bring this conference into the reality. And I also thank you for giving me the opportunity to share my work on this platform. So the topic of my talk is, as you can see, the molecular and cellular basis of pathogen survival inside the host. So here I'm going to discuss some of the molecular events and some cellular events. And I will cover two aspects. One will be with the bacteria, specifically with the Mycobacterium tuberculosis, and in the latter half I will discuss some of the important discoveries that we recently made on enteric viruses. So if you see how a pathogen survives inside a host, the pathogen must able to meet the several criteria, including the colonize the host. find a nutritionally compatible niche in the host body, avoid, subvert, or circumvent the host innate and adaptive immune response. It must be able to replicate using the host resources, and it should be able to exit and spread to a new host. So the question is how you can control them. So to understand the controlling strategy, we must learn about the pathogens, like what are the signs and symptoms of infection that is caused by the pathogen or that may be appeared due to the host response, the diversity of pathogens and their complex life cycle, identify the virulence factors. So this may give you a pathogen-directed therapy. Another is the dissect molecular and cellular events during infection, and it will lead to the foundation of host-directed therapy. And finally, identify the cellular tropism, which is really important for the viruses. So I'd like to start first with the Mycobacterium tuberculosis and how it interacts with the host. And I'm not going much more detail into the Mycobacterium tuberculosis, as I'm hoping you all are aware with the basic biology of the Mycobacterium tuberculosis, which is an intracellular pathogen that causes the tuberculosis. So in this part, my broad aim will be to understand what are the role of microRNAs during the host pathogen interaction, specifically with this MTB, and how it regulates the MTB survival. Okay, so we started with a very simple strategy. We infected the raw macrophages with the Mycobacterium tuberculosis at an MY10, and we isolated RNA at different time point post-infection, like 4-hour and 24-hour. Isolated RNA, and we use the microarray technology to do the global profiling of the microRNA and the gene. So here you can see in the figure A, there are heat map of few down-regulated microRNAs, and in figure B, this is the differentially regulated transcription factors, and the boxes shown here for microRNA like LET7F and MIR26A, and the transcription factors including KLF4, ATF3 is the boxes by interest that I'm going to discuss here. So first we started to looking the role of LET7F down regulation, and we wanted to understand what is the role of this down regulation during the infection. So before going to next level, we first validated the down regulation of LET7F in MTB infected macrophages, in the mouse macrophage raw cells in figure A, in DMDM in figure B, and in human macrophages C through the northern blot. So you can see with the control, LET7F is clearly downregulated at 4 and 24-hour post-infection. We further confirmed the downregulation of LET7F in lungs of infected mice at 9 weeks and 12 weeks post-infection. So here in figure D and E, the lane 1, 2, 3 is a control samples, where the 4, 5, 6, 7, and 8 is the infected samples. And this data in figure E, see, it clearly indicates that the downregulation of LET7F is more prominent as the time progresses. Next, we identified the mechanism behind downregulation of LET7F. We identify the secretory antigen of MTB called ESAT6, which is an important antigen that modulates several immune responses. So here, this data is from the northern blot analysis. So in figure A, you can see the downregulation of LET7F at 4 and 24-hour post-infection with control to the uninfected. When we use the delta ESAT6 mutant of Mycobacterium tuberculosis, then you cannot, here in figure B, you can't see the downregulation. So the expression of LET7F is unaltered. If you go to the figure C, when we infected the raw cells with BCG, which is a vaccine strain of Mycobacteria, again, there is no change in the LET7F expression. But when we restore the region of, so basically the ESAT6 is encoded by the region of deletion 1. So when we incorporate the region of deletion 1 into the BCG, so we call that strain 2F9. when we infected the raw cells with 2F9, you can see there is a very clear downregulation of LET7F expression. Again, we further confirmed the downregulation when we purified the ESAT6 protein and treated the raw cells. Then we also observed the downregulation of LET7F. So this clearly indicates that ESAT6 is involved in the downregulation of microRNA LET7F. Next, we ask the question, what is the physiological significance of the downregulation of LET7F? So for this, we did a very simple experiment. We quantified the colony forming unit in the presence and absence of LET7F mimic. So basically, we overexpress the LET7F mimic in the raw cells, and then we infected MTB followed by the CFU. So you can see the dotted line is for the LET7F mimic, which shows the reduction in the CFU number. That indicates that the LET7F overexpression attenuated the survival of MTB inside the macrophages. As you all are aware that the microRNA itself don't do any function.
No screen is visible.
Sorry? There is any question?
Slide is visual. Sir,
slide is
continue,
visible?
sir. Yes.
Okay, thank you. Thank you so much. So the microRNA function through their target genes. So we identified what are the target of LET7F. So we use our global gene profiling microarray data, and we identified more than 100 target gene of LET7F. But we focus on the A20 or TNF-AIP3, which is a well-known NF-kappa B inhibitor, since it has an obvious role in the innate immune response. So we validated that A20 is the target of LET7F. So for this, we clone the A23' UTR, and as well as we deleted the seed region of LET7F that is complementary into the A23' UTR. And we transfected these two constructs along with the LET7F mimic in the HEK cell line, and we measured the luciferase activity. So you can see in the presence of LET7F mimic, the luciferase activity was almost 50% reduced, whereas in the mutant plasmid, the deleted seed region don't have any effect of the LET7F mimic, which indicates that A20 is a target of LET7F. Further in figure B, we did the MIC-IP by transfecting the AGO2 construct. As you know, the RISC complex incorporates the AGO and the AGO protein that selectively enrich the passenger strand of microRNA that pull down its cognate mRNA for the degradation or stop the translation. So after the AGO2 precipitation with LET7F, you can see there is a very clear increase in the A20 mRNA that further confirms that A20 is a target of LET7F. We studied the A20 expression in the presence and absence of LET7F to understand whether it modulates the protein level after MTB infection or not. So in figure A, you can see we transfected raw cells with the control mimic as well as the LET7F mimic, followed by the MTB infection at the different time point. So you can see in the control mimic, A20 expression gradually increases with the time, but in the presence of LET7F mimic, the A20 expression attenuated, which indicates that LET7F inhibits the expression of A20. Further, this observation was true for the BMDM in figure B. Conversely, when we transfected the raw cells with LET7F inhibitor, the A20 expression was increased. That indicates that LET7F really targets the A20. As I mentioned before, that A20 is an NF-kappa B inhibitor. So we asked the next question, that if LET7F targets the A20, does it regulates the NF-kappa B activities by modulating the A20 expression during the MTB infection? For this, we measure the P65 translocation from cytosol to the nucleus for ELISA. Here you can see there is a the black bar is for the LET7F mimic and the white one is the control mimic. So you can see at the latter time point of infection like 12-hour, 20-hour and 28-hour post-infection, there is a very significant increase in the NF-kappa B activity after LET7F mimic transfection, which indicates that LET7F regulates the NF-kappa B translocation from cytosol to nucleus during the infection. Next, we measured the different cytokines and chemokines from A20 knockout mice isolated macrophages. So here you can see in the A20 deficient macrophages, the cytokines like TNF-alpha, IL-beta, IL-6, and the chemokines like KC, MCP1, and MIP2 all were upregulated compared to the wild-type macrophages. Next, we asked, what is the physiological significance of A20 in the survival of MTB? Since the nitrite is directly correlated with the survival of MTB and the killing, so we measure the nitrite as well as the CFU from the same well. So you can see the nitrite level is increased in the presence of, in the absence of A20, as well as the CFU is also attenuated. So meaning there is a reduced survival of MTB. So which is well correlated with the high level of nitric oxide kills the mycobacteria inside the macrophages. So briefly going for this part, the summary is LET7F and A20 regulate the NF-kappa B signaling to modulate the production of cytokines and the survival of MTB in macrophages during the early stage of infection. Going into the next part, here I would like to discuss briefly about the role of macrophage polarization in the survival of Mycobacterium tuberculosis. And briefly, I'll go for the M1 macrophages and the M2 macrophages. So M1 macrophages is the characteristic feature of antimicrobial defense. Basically, the pro-inflammatory region, which has the high level of nitric oxide and the ROS. Whereas for the M2 macrophages, they are the anti-inflammatory with the high level of IL-10, arginase 1, and of course the KLF4 that I'm going to discuss here. So once we were checking the expression of KLF4, and to our surprise, when we did the QRT-PCR from the MTB infected raw cells, the KLF4 expression decreases with the time. But when we did the Western blot to see the protein level, the KLF4 expression was increased and it was modulated. So this was to our first knowledge. We were always thinking that mRNA and protein is well correlated. If your protein is going down, it should have a low amount of protein at the same time. But actually, this is not the case. So we try to understand what are the mechanism behind it and how it can happen. So we identified one microRNA that is called the MIR26A to explain this phenomenon, which was downregulated after MTB infection. So downregulation of MIR26A was very well correlated with the KLF4 expression at the transcription and the translational level. So first we validated the downregulation of MIR26A in the macrophages and the infected mice organs. And this is a northern blot analysis, and you can see the downregulation with the time points. Next, we studied the expression of KLF4 by overexpressing the MIR26A mimic or the inhibitor. So figure A and B shows the KLF4 expression under the influence of MIR26A mimic. So you can see it's very obvious that the mimic inhibited or attenuated or reduces the expression of KLF4 level. Whereas when we transfected the raw cells with MIR26A inhibitor, there is an increased expression of KLF4 as compared to the control inhibitor, which suggests that the MIR26A really targets the KLF4. Next, we ask the question how KLF4 influences the survival of MTB in macrophages. So we transfected the raw cells with control siRNA and KLF4 siRNA and quantified the colony forming units at the various time points. So you can see here under the influence of KLF4 siRNA, the survival of Mycobacterium tuberculosis was attenuated in the raw cells as well as in the primary macrophages BMDM as well, which suggests the overexpression of KLF4 actually favoring the survival of mycobacteria. Next, we try to understand the co-localization of or the trafficking of mycobacteria to the lysosome, which is the major killing mechanism of the macrophage utilized to clear out the pathogen, the mycobacteria during the infection. So you can see here we transfected the raw cells with Mir26A mimic and the control mimic, and then we infected actually the MTB, which was labeled with the FITC, that is, you can see in the green, and we use the LAMP one, which is a marker of lysosome. And in the right-hand side, you can see the quantification. So in the presence of Mir26A mimic, almost 30% increased co-localization of MTB with the lysosome was observed, meaning the overexpression of Mir26A promote the trafficking of MTB to lysosome, means it is involved in the direct killing. And we observe the similar result when we silence the KLF4 siRNA in the raw cells. So in summary of this part, Mir26A and KLF4 shifts macrophages towards the M1 phenotype by augmenting the production of pro-inflammatory chemokines. Downregulation of Mir26A and upregulation of KLF4 favor the survival of the pathogen in macrophages. And the Mir26A and KLF4 regulate trafficking of MTB to the lysosomes. Now going into the third part, I will take a very small time for this, and here I'm going to discuss how a transcription factor can act as an activator and as a repressor at the same time, and how it regulates the MTB survival. So generally our conception is either a transcription factor can be activated or can be a repressor, but this is not true. A transcription factor actually depends on its cognate partners, whether it is getting dimerized or it is acting alone, and what are the abigenetic modifications. So there are several factors actually that influences the activity of the transcription factor, and we have shown here beautifully how ATF3 has a dual mode of action at the same time during MTB infection. So ATF3 was upregulated during the MTB infection of macrophages, as you can see in figure A and B, as well as in the infected lungs of mice. ATF3 was also upregulated at the protein level, as you can see in figure D. And we also checked the survival of MTB in the presence of, by silencing the ATF3. And the silencing of ATF3 promote or augmented the survival of MTB during the infection. When we try to understand what are the cytokine profile by silencing the ATF3 during silencing of ATF3 during the infection, you can see here the cytokines TNF-alpha, IL-6, and IL-12, as well as the chemokine MCP1, both were reduced, which shows that ATF3 positively regulates the cytokine and chemokines during MTB infection. On the other hand, we quantified the lipid body formation by silencing ATF3. So here, I will go just briefly for the lipid bodies. So lipid bodies are the cholesterol metabolites that is actually sensed by the mycobacteria as a nutrient source, and it helps for the long-term survival of MTB during the granuloma formation in the lungs. So you can see after ATF3 silencing, there is an increased number of cell populations were positive for the lipid body, as was quantified in figure B. You can see the red box where there is a very high population of cells having more than 15 punctate structure that were positive for the lipid body. So there are two things. ATF3 positively regulates the cytokine production and negatively regulates the lipid body formation. So what does it mean? So we are trying to understand what are the mechanisms behind this dual role of ATF3. So we identified one molecule called the BRG1. This is a protein chromatin modifier that actually binds to the DNA, opens the chromatin structure, accessible for very easily for the transcription factor and some associates molecule. And hence it promotes the transcription of the gene. And this BRG1 was upregulated during the MTB infection as compared to the control, as you can see in figure A. So we wanted to see whether ATF3 interacts with BRG or not. So for that, we overexpress the flag ATF3 construct in raw cells and infected with MTB, and then we pull down using the flag. And we did the Western blot. You can see the BRG1 expression was increased at 4 and 8 hour post-infection compared to the control. That indicates that BRG1 interact with the ATF3. Next, we did the re-chip analysis, which involves the first IP using the anti-ATF3 antibody, and the second IP involves the anti-BRG1. You can see, and then we did the qRT-PCR to understand the relative enrichment. So you can see, sorry, at the TNF alpha promoter, you can see the association of both ATF3 and BRG1 that was very similar for the IL-12 promoter and the NOS2 promoter that is responsible for the nitric oxide production. And the level was almost very similar for the first IP and the second IP as well, which indicates that ATF3 and BRG1 sit together on the promoter of these genes and activates the expression. So basically the mechanism for the positive regulation is when ATF3 interacts with the BRG1, it promotes the cytokine or the NOS2 production. Next, we try to understand the mechanism, how it acts as a negative or as a repression. So here I'm giving you one example of the CH25H gene, that is the 25-hydroxycholesterol, which is an intermediate metabolite of the cholesterol metabolism. So here you can see when it silences the ATF3, there is an increase in the expression of CH25H gene as shown in the figure A. And we did the IP experiments and we did the chip by the qPCR, where you can see the ATF3 enrichment on the promoter of CH25H gene, which is increased in a time-dependent manner at the 8, 16, and 24 hour. So meaning that the silencing of ATF3 is going, so it's like a transcription factor is going and binding to the promoter of some gene upstream. And if you are silencing a gene, there is an increase in the association, decrease in association, but there is an increase in gene. So how it can be happen? So we identified another histone modification that is called the H4K8 acetylation that was being affected by the silencing of ATF3. So basically ATF3 silencing increases the H4K8 acetylation at the promoter of CH25H gene, and that was the reason that the silencing of ATF3 increases the expression of CH25H. So basically this mechanism involves the ATF3 inhibits the H4K8 acetylation, and that inhibits the CH25H expression. So in summary, there is the opposing role of ATF3 in regulation of distinct sets of macrophage gene during infection, and the ATF3 function converging on a host protective response. So now I'm moving from the bacteria to the viruses, and this is really an amazing story that we have challenges the previous hypothesis that people were believing. And here I'm going to talk about some enteric viruses replication in the salivary glands and how they bypass the fecal-oral route of transmission. So enteric viruses is basically the viruses which has a route of the oral fecal route. They transmit and transfect with the oral fecal route. That was the previous conception. And the best known example of the enteric virus is the norovirus and rotavirus. And these viruses causes diarrhea. And these both are the positive sense RNA viruses. And for norovirus, there is no vaccine till date. Whereas for the rotavirus, there is a vaccine like Rotatrac and the Rotatrix that are being used. So going briefly, enteric viruses basically known to be replicate in the small intestine. So we did a very simple experiment. We just orally gavas the norovirus strain of the murine strain of the norovirus and rotavirus. So for norovirus, it is called the MNB1, and for the rotavirus, it is called the EDIM. That is, I'm going to throughout the lecture. So when we orally gavage the MNB1 and EDIM into the 10 days old pups, and we just isolated the small intestine and RNA, and we did the qPCR, you can see there is an increase in replication, and it gradually going down after 5-day post-infection, which is very obvious. Next, we ask the question, if you are giving the orally gavaging the pups with the virus, either MNB1 or ADM, during the suckling, that is transfer the virus to the mammary gland of the dam mother. To understand this, we simply isolated the mammary gland of the dam at the 3-day post-infection, and we did the qPCR for the MNB1 and EDIMs. And you can see in figure B, there is a very huge copy number of the virus, like even in the order of 10 to the power 6 to 10 to the power 7 copies per ml in the mammary gland for MNB1 and EDIM respectively. So these all are the unpublished data that is already in communication, and it is under review in Nature. So that's interesting story I'm sharing here, and it will be more interesting later on. We further confirm the mammary gland infection with this noro and rotavirus by the confocal imaging. So here you can see for the EDIM, we use the NSP5, which is a non-structural protein for the EDIM. And we use an epithelial cell marker called the alpha spoon muscle actin, which is in red. And for MNB1, we use a non-structural protein 4 and the alpha spoon muscle actin for the staining. And it clearly indicates that the epithelial cells of the mammary gland got infected with this virus, which is previously unknown. I mean, nobody was thinking that it can be happen, that the virus can be retroductively transferred to the mothers as well from the pups. But here we ask one question, there may be some possibility that if you orally gavage the pups with the virus, it may be possible that in mouth of the pups, there may be some virus remains and they can transfer into the mammary gland during the suckling of the milk. To rule out the possibility of this, what we did a very simple experiment. We infected the pups with the EDIM, and after 1 day post-infection, we transferred the natural dam with the foster dam. So meaning is, the natural dam, if it got infection through the saliva, that we were hypothesizing that some part of the liquid of the virus may remain in the mouth and may be transferred to the mammary gland. After 1 day post-infection, we transferred the pups in direct inoculated pups with the foster dam that has no infection, no orally gavage virus. And then we use the uninfected pups and put into the case of natural dam. So basically we just swap uninfected with the infected one to understand what is going on. And then we quantified the genome copies of the ADIM in the foster dam and in the natural dam. So here you can see the foster dam and natural dam both were positive for the EDIM replication in the mammary gland, as well as the direct inoculated pups that were, the direct inoculated pups is really positive for the virus, but the uninfected pups that we keep into the case of the natural dam were also got infection. So meaning the mammary gland was infected, and therefore during the suckling of milk, some viruses were coming back to the pups and they were getting infection. So we ask the next question, how is MNB1 and EDIM is transmitting to the mom? Is it through saliva? So for this purpose, we infected the adult mice with MNB1 or EDIM. After 2 and 3 day post-infection, we injected the pilocarpine. So it activates actually the autonomic nervous system, and it activates all the secretory pathway to eject the liquid outside. And then we put some cotton swab inside the mouth of the mouse, and we collected the saliva. And that saliva was used to feed the 10 days old pups. Oh, sorry. So here you can see the result. We first confirm the presence of virus capsid protein, the BP1 for MNB1 in the saliva, and it was positive for 2, 3, and 4 day post-infection. And we also confirmed for EDIM the BP6, which is positive for the 2, 3, and 4-day post-infection. We also confirmed the TCID50 for the MNB1. And when we orally gavage this saliva into the pups, you can see, and we measure the genome copies per ml in the small intestine of the pups, it has a very high copy number for the MNB1 and EDIM as well. In fact, more than 10 to the power 6 genome copies per ml. So meaning these salivas are really infectious and able to transmit the virus from one individual to another individual. Next, we measured the replication kinetics of these viruses into the salivary gland. So basically for mice, we isolated the submandibular gland because they are very easy to extract out. The other glands are very small, it's very difficult to take out. So basically we worked with the submandibular glands only here. So we infected the mice and we isolated the submandibular glands at the different post-infection and measured the viral copies through the qPCR. So here you can see for the MNB1, the peak of the replication in submandibular gland was a 5-day post-infection and later and gradually decreases. Whereas for the EDIM, the peak was at day 3 post-infection and later it gradually decreases. We also confirmed another enteric virus called the astrovirus that also infects the submandibular glands of mice and positive for the 8 and 12 days post-infection. So if you just see the anatomy of the salivary gland, how it looks like. So in figure 1, it is a salivary gland anatomy. The figure in the right hand side, you can see if you cut the longitudinal section. So basically I'm interested in the figure 2 and 3, where you can see the anterior cut and the posterior cut. So anterior cut is ductal cell for which we are going to show you the infection, and we use the marker syndican-1 for that. And in figure 3, you can see the acinar cells for which the marker is NKCC1. So we did the imaging after isolating the pup's salivary gland, submandibular gland, and we stained with the non-structural protein and the corresponding marker SDC syndican-1. And for the norovirus, it's NKCC1, and it was positive. So basically, the EDIM infects ductal cells of the salivary gland, whereas the norovirus infects the acinar cells of salivary gland. Further, to understand what is the replication dynamics of the virus inside the intestine of norovirus, if we remove the salivary gland. So to understand this, we did the salivectomy. We surgically removed the salivary gland from mice, and we infected the wild type. Wild type means the mice having the salivary gland, and one set which was without salivary gland, infected with the murine norovirus, collected the small intestine and checked the virus genome through the qPCR. So you can see it's very obvious, the pink one, the blue one is for the wild type. After day 5, the clearance of the virus was much more faster compared to the wild type, which suggests that if you are having a infection in the salivary gland, they are secreted into the saliva, and through saliva, they are going again into the small intestine, and that virus again promoting the infection in the small intestine. So in summary of this part, this salivary gland can replicate enteric viruses, and these enteric viruses are secreted into saliva, and enteric viruses in saliva can reproductively transmit via suckling. So this is the whole story that I shared here, and I'd like to acknowledge my PhD mentor Jati Basu, my postdoc mentor here, and my lab members, and my collaborators in the NIDCR and HLBI core facilities, and some NIAID people who help with the antibodies and all those. Thank you so much for your patience and hearing, and I'm happy to take questions. Mhm.
I would ask the audience to unmute and ask questions.
Parul, there is one general worry about what we call MDR, multi-drug resistance, है ना? Especially in case of tuberculosis. इस पर थोड़ा कुछ कहने की अगर बोल सकते
So Manish, Professor Nihal is asking, would you be able to throw light on the multi-drug resistant strains of tuberculosis?
Well, so multi-drug resistance is the actually that is the resistance for at least one or two drugs of the first line, like the rifampicin or isoniazid. So that is.
Would you get the question, Manish?
Can you repeat again, please?
We want to know about the multi-drug resistant strains of tuberculosis, a very general perspective.
Yes, yes. So that's what I'm coming to the point. So the multi-drug resistance strain means the strain which is there is no effect of the first line drugs like isoniazid, rifampicin as I mentioned. And unfortunately, I have to say, in India, we are not clear about taking medicines. Whatever, everyone in India is a doctor actually. So whenever we are something, we are taking the medicine of the class, or we are asking this to anyone, and we are taking the medicines. And that antibiotic patient is actually converting its tuberculosis to the multi-drug resistance. And there is a very little studies about the multi-drug resistance that is continuously going and really serious, and we should consider how we can tackle with that. But very little studies is currently going on in India, unfortunately. But we have to focus mostly on multi-drug resistance strain compared to mycobacteria itself. And until unless if we don't find the mechanism, how it's going beyond just the antibiotic patient is more wanted. So in my opinion, we should understand the mechanism behind the NDR strain and what are the complexity of the organism that is developing during the selected patient, and how it is evolutionarily evolved that maybe give a more drug targets, and we can develop some new antibiotics against them to kill them.
Thank you, Manish.
Professor ji.
Thank you, sir. Now I invite our second speaker of second technical session, Dr. Vijay Singh. He obtained his master's in biotechnology from Department of Biotechnology, Lalit Narayan Mithila University, India, and PhD in biotechnology from CSIR Central Salt and Marine Chemical Research Institute, India. During his doctoral research, he worked on isolation of antiquorum sensing compounds and inhibition of biofilm formation of Pseudomonas aeruginosa, and actively contributed to the plant molecular biology. He joined CSIR Institute of Microbial Technology, India as National Postdoctoral Fellow and worked on Mycobacterium tuberculosis biofilm before joining Professor Lawrence Rahim at Harvard. His PhD thesis is on isolation and characterization of antiquorum sensing compound and rhizospheric bacteria of coastal saline ecosystem. He is currently working as postdoctoral fellow in the Department of Microbiology at Harvard Medical School, Department of Surgery, Massachusetts General Hospital, and Schreiner Hospital for Children, and working on the drug discovery against antibiotic tolerant persistent bacterial infection to tackle the emerging threat of antibiotic tolerance and multi-drug resistance. His focus is on multi-host pathogen interaction and anti-infective therapeutic to prevent intestinal barrier rearrangement following burns, injuries, and Pseudomonas aeruginosa superinfection. And now I request Dr. Vijay Singh sir to put some light on the theme bacterial communication and crosstalk, disease treatment and beyond.
Appreciable.
Can you hear me?
Yes, sir.
Yes. Namaste. Good morning, everyone attending this conference, and I am very delighted to present my work where I started my education and research, and I am very thankful to the president of this conference, Dr. B.S. Jha, and Professor M. Nehal, and organizing secretary Dr. Parul, and my friend Kali Charan. This is an before I go further, I wish to share my slides. Give me one moment. Yes, can you see my slides? Yeah. So I'm going to talk about today what I'm doing with the bacterial infection, how bacteria talk to each other, how they cause a disease, and how we can do the treatment of these infections, which is very hard to eradicate, and there is a multi-drug resistant bacteria which is increasing day by day. So, and I'm very delighted to present my work in the online mode, and just wanted to remind Professor M. Nehal and Kali Charan that this e-conference is very unique opportunity for me. We started this online classes very long ago, where there was no 4G internet or concept of online classes, and we used to have these online classes like 13, 14 years ago. Now I'm very happy that I have opportunity to present my work is online mode. So now basically I'm going to talk 3, 4 things today. What I'm doing, why I am doing, what people are going to get benefited of this, and what the people listening today is going to get the take home message. So first I would like to start with bacteria. Once you heard the term bacteria, immediately you think about, okay, so person have infection. If you are resistant and if you are healthy, you are walking, you are working, you have no problem. If you are a tolerant host, you have a bacterial infection, you get little bit sick, but you are still healthy, you are walking, you are doing daily life. But if you are susceptible and bacterial infection is severe, you have a more bacterial infection, you get sick, and there is a different kind of bacterial infection. You have a cholera, typhoid, tuberculosis, diphtheria, tetanus, many bacterial infection you have. Then very simply, you go to the doctor, they check you, they prescribe you antibiotic, and you get treatment. You are healthy, you come home, and you are fine. But the question is that so easy, or bacteria is all about all this, or bacteria is only causing this infection, or is there any other thing? So before I talk more about bacteria, just to put some comparison, we have a we consider ourselves as human, and everyone knows that we have a human body. The human body is consists of multi cells, and we have organized cells. At the same time, bacteria have a single cellular organism, and they have very simple cellular makeup. They have the outer cover, the single DNA molecule, and then some flagella, and then so just to put it more forward, then how many number of cell and gene in our body have? So if I ask this question or anyone or myself, we simply think that okay, so we have a human body, you have a human cells, and we are working, and what we are doing is because of our cell. So if I say in the number of cells, so human body is almost having like 30 trillions of human cells. So just put in comparison, if you accumulate all the cells, and if you have a marker, like if you see that the accumulation of human cell, so just to put a comparison, it's not number exact number, but just to put a number, so you have 30 trillion, about 30 trillion cells. Now the question is, when you put the number of bacterial cell in human body is 10 times more than animal cells. So what does it mean? The human body all the time have 10 times more bacterial load in your body all the time, in you, within you, or inside you. And we are scientists, or the people are the science backgrounds. Of course, gene matters. And if you put forward with the perspective of genes, so human body have almost 30,000 genes. But on the human body, we have this much, like 100 times more bacterial gene within the body or outside of the body. And now we are not alone. The simple people are saying that we are the now it's like very complex. We have a human body, the only disease causing bacteria. Now if we have 10 times more bacteria than our cells, and 100 times more bacterial gene than bacteria, unlike human, then why we are not sick? And what is the other perspective of these bacteria? These bacteria is keeping us alive. They are forming a protective layer around us, which is the invisible jacket, and they protect us from the environmental stress, different kind of pollution, and the different pathogens. The bacteria digest our food. They give us vitamins. They train our immune system to fight with the pathogenic bacteria. They educate them that how bacteria will come and how they attack the immune cell and how to hijack the immune cell. So immune cells is trained before, and once the pathogenic bacteria comes in, they fight and they eliminate the pathogenic bacteria, and then the 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 beneficiary bacteria which is in our body is kills the pathogenic bacteria. Now the question is, what do we remember? The good bacteria which is helping us with all these things, but no one knows, or people don't focus on this. If the people know, they don't focus on that we have lots of beneficial bacteria which is beneficial for our daily life, but we do remember that there is some bacteria which cause disease and we get sick. So it's kind of uh, the bad thing done by some people is like affecting the other. And so what happened most of the case, when we have the now come to the point where once you go to the doctor prescribes you antibiotics, and what happened, bacteria get killed. But at the same time, now we have huge number of bacteria which is working for us. And when we are eliminating and killing those bacteria, whenever we are taking antibiotic, we are killing those bacteria too. And our digestive system is affected, our vitamin intake is infected, affected. We have we have no protective coverage, so other bacteria can infect you, and you will get multi-bacterial infection. And the worst thing is, in case of India, we have like no control on antibiotics. So we can go to the antibiotic shop, we can have antibiotic for anything. You have a viral fever, you have a flu, you have a headache, you went to the shop and you said, oh, I have a fever, can I have antibiotic? And they gave you. Even for any kind of infection, you don't know whether this is a bacterial infection or viral infection or some other. So people have to be very careful. And one thing I'm sorry to say, but in India, doctors are also without checking, without confirming that this infection is bacterial or viral or whatsoever, they just write the medicine like antibiotic, everything antibiotic, everything antibiotics. No, it's very harmful, and this is leads to the antibiotic tolerance or multi-drug resistance. So people have to be very careful, and most of the country had this ban this antibiotic selling. So in here in US, you can't get any antibiotic by your own. You have to have doctor's prescription, and doctors are hardly rating this prescription to have antibiotics. So for short term, you are taking antibiotic without knowing. You think you are going to get cured, or you are in hurry, but it's causing like very long term effect. So once you have a high level of antibiotic, you don't know the dose, or most of the people don't know the dose. So if you have a high antibiotic, bacteria have the pathogenic bacteria already have many machinery, and once you have a continuous antibiotic, they get adapted to that, and they gradually developing the antibiotic resistance with the most of the antibiotic. What happened? The large number of persistent bacteria, multi-drug resistant bacteria worldwide is developing, and this hepatitis use of antibiotic is not good at all. So if there is no action taken, by the end of 2050, we have millions of people suffering from this multi-drug resistant, and which is very, very impossible to cure. So the other thing is, once you put the antibiotic, bacteria also have to fight back. So what bacteria is doing, they develop a outer sheath or mucilage kind of layer outside them, so drug cannot enter. Or some of the cell develop themselves as a persister formation. So what happens when the antibiotic concentration is very high? So here is the one thing you have to differentiate between the multi-drug resistant and persister cells. So multi-drug resistant bacteria is they permanently change themselves to the tolerant to that particular antibiotic. But persister cells is when there is an antibiotic concentration is high, they put themselves in susceptible state, and once the concentration is goes off, your body you wash off the antibiotic, the bacteria is start growing again, and then this persistent then recurrent infection is very impossible to eradicate. And once they develop the biofilm, it's very impossible to eradicate. And the bacteria also have the machinery to protect themselves from desiccation, ultraviolet lots of things is happening, and bacteria protect themselves. So now question is how we can overcome with this situation, and how we can avoid the antibiotic, and how we can fight with these bacteria without using antibiotic. So to understand that, we need to understand the bacterial mechanism, and that mechanism is bacteria. Now once you understand the bacteria, how they are living, so the bacteria have a very simple life. What they are doing, they are getting nutrients from environment, they keep growing, they grow twice of their size, they divide in middle, and they make two. And then again they grow, they divide and become two. So on, so on, so on. So their population is increased. But bacteria, as you know, bacteria is very tiny organism. And if you remember the previous question, so bacteria is a very tiny, and if you think bacteria cannot do anything alone, and in comparison to our human body cells and our human body is huge, and bacteria is very small. We can't see even by naked eye. We can only see in microscope when we zoom in 100x. Then how they are infecting us, how they are making us sick. So they have the very specific communication system, and which is called quorum sensing. So this quorum sensing started almost 50 years ago. So what happens once bacteria is low density, they start sending some signals to their neighbor cells. So these signals is a chemical signals. What happens is they send some chemical, the neighbor cells sense these signals, and they start producing signals. So what happens with these signals, they also recognize the number of cells. So they use the signal in a two way. One, they send the signal, make the other cell to send that signal, and they also counting the number of population. Once they have the certain number of population, they accumulate together, and at the certain density, that population, they switch on their virulence gene and they attack us together. So you have a large number of virulence, you have on and on the virulence gene, and they invade the cells. And once they invade the cells, they keep increasing and they so on. So to understand this mechanism, I just wanted to put some pathways how bacterial column sensing works and how they send the signals. So what happens once the bacteria have low density? So this signal is very unique. This is kind of very secret. They talk to each other secretly. And now you know that there's a large number of bacteria. So there is a millions of bacteria around. How they can sense one signal, the bacteria, one bacteria can send a signal to their only own siblings or their group. There is thousands of bacteria around. So this signal is very specific. They synthesize a particular synthetic gene, and they bind to the particular receptor. They cannot bind to the other bacteria, like other bacteria other than this group or other bacteria. So they are very specific, very secret, very encrypted, and like other bacteria cannot decipher. So they only make their siblings to understand the signal, and they produce more and more virulence. So here the like survival of fittest kind of thing. So when you are in minority, you are sending signals secretly. Once you have a majority population, you attack. But the thing is, the bacteria is very clever. This is tiny organism, but they are very clever. So they also have signals. So this case, the gram positive bacteria, gram negative bacteria, what they are doing, they are synthesizing one signal molecule. If I start my pointer, yeah. So they are synthesizing signal, and this signal go outside for the other cell. The other cell also sends the signal. They start producing the signaling molecule. And once this concentration keep increasing, increasing, and increasing. In the case of gram-positive bacteria, they have some cyclic peptides. They synthesize the cyclic peptides, they bind to the channel, they go outside and send signal. They keep producing the signals. In the case of the third thing is autoinducer. So this kind of inducer is for the interspecies. Once they have to interact with other bacteria or they have to interact with other hosts or other plants. So they also have the chemical language to interact others. So what happened once they have a less density, they synthesize this signal. And next, what happened when their population is increased and the signal concentration is also increased at the same time, they start getting into the cell and they bind to the specific receptors and they turn on the gene. And then at the same time, there is a millions of bacteria. The same phenomena happens. So all bacteria have bind to the receptor, like signal bind to their receptor. They start synthesizing their gene at a once. And at a once, they have production of huge amount of virulence vector, and that is sufficient enough to make you sick. So to understand little more, the quorum sensing and this phenomenon is involved in many things. So this quorum sensing is responsible for virulence vector production, biofilm formation, the secondary metabolite like formation, the bacterial crosstalk, the bioluminescence, the motility of bacteria. Then also they can modulate the immune system of the host. They can arrest the cardiovascular system of the eukaryotic host. So this quorum sensing is very, very unique and very dangerous weapon for the bacteria. So now this was a general overview of pathogenic bacteria. So now if I focus a little bit more about the pathogenic bacteria I am working on, which is Pseudomonas aeruginosa. So Pseudomonas aeruginosa is a opportunistic human pathogen, and The bad thing about this bacteria is it can infect any host, whether it's a human, plant, animal, fat, bacteria. It can infect, sorry, like not bacteria, but it can infect anything, and it can kill the bacteria. So these pathogenic bacteria have wide range of hosts. And this cause a persistent secondary infection because this has a very strong weapon of bioinformation. Once they goes in, they turn on their gene and they start synthesizing their virulence vector, they establish in your cells and they form a biofilm. The biofilm is thick covering of the group of bacteria. And once they cover all bacteria together, they work as a multicellular organism. They don't allow any antibiotic to get in. They don't allow any other bacteria to attack. They don't allow human immune cell to attack, and they are very persistent. And this pseudomonas infection is even more worse if it's infect the immunocompromised patients, such as like someone have HIV, someone have the cystic fibrosis, someone have the burn injury, or someone have a trauma. So this bacteria is like once your immunity goes down, this is more dangerous. And what happens, this bacteria is very ubiquitous, and the worst part of this bacteria is they are like nosocomial. What nosocomial means, the 57% of this bacterial infection happen in hospital setting. You are healthy or you are sick, you have a trauma, you have a burn, or you have any other infection, you went to hospital, and then there is a spread of this pseudomonas from anywhere. It can come from flower, it can come from food, it can come from any other person, clothes, or any plant, and it can infect you. And once you get infected, it's very hard to kill by antibiotics. So how can we get rid of this? How can we kill this bacteria, or how we can eliminate this bacteria? So we have to understand a little bit more about their mechanism, how they are using their quorum sensing machinery, what is their quorum sensing signals, and how they start signal and when they stop signal, and what we can do. So what they are doing, they have three interconnected systems. One is LAS, one is RAL, and third is MBFR. This is a main quorum sensing system of Pseudomonas. So once they enter into cells, or if they are in low density, what they are doing, they are synthesizing the signal which is called homoserine lectone. So homoserine lectone goes outside the cells. At the same time, the homoserine lectone also influence the synthesis of RAL-I and RAL-R. So they are not synthesizing their own signal molecule, they also influence the synthesis of other signal molecule. And this goes on. They also induce a third quantum sensing system, and all are started synthesizing their molecules. So happened very soon, they accumulated large number of chemical signal, or all three type of signal outside. So, and once they have the enough number of quantum sensing molecule, again, the same procedure, they form a biofilm and everything. So what now, what we can do, we can stop this quantum sensing signal, and we don't allow so bacteria to synthesize their virulence vector or send a signal to other bacteria. So here there is a three way we can stop. Whether we can stop the synthesis of signal. So if we have something and we stop the synthesis, they will be not able to produce their quantum sensing molecule, and they are not able to influence the upstream genes. Or if we have something and we stop the binding of these molecules to their cognate receptor, even they synthesize, and if they don't bind to the receptor, they will not able to turn on their virulence factor gene, and they will not be able to produce this virulence factor or virulence the toxin at once. So if you block their receptor, they synthesize their signals, but they will be not able to attack you at once. So you will be like, and then if we have something, if you can't bind to the, if we can't stop the synthesis, we can't stop the binding. If we have something, we can degrade the signal molecule as well. So now the question we had during my PhD, so how can we stop these quorum sensing molecules and how we can get rid of these? So first thing come into mind that why don't, as we are saying, लोहे को लोहे काटता है, so why don't we ask something from bacteria, other bacteria, beneficial bacteria, which is naturally inhibiting these pathogenic bacteria or destroying their signal, and then they're killing pathogenic bacteria. So for that, we chosen to have the rhizospheric bacteria. So why we chosen to have rhizospheric bacteria? So we were working and the when I was doing PhD, the Bhavnagar regional coastal area, and there is a very large coastal line, and the coastal line have like high salt concentration. So the in the kilometers and kilometers, you don't have any plants. But there is some plant which can grow in that such a high salt concentration. So normally, if you put some salt in the plant, they will die. But even this like high salt concentration where there is a crystal of salt, how can this plant survive? So there is something happen with this plant. So here, the rhizosphere and the rhizospheric bacteria play a role. And there's a long been known that the rhizospheric bacteria is influencing plant for their growth promotion. So what happened, this is a kind of mutual combination in the root of the bacteria. What is the rhizosphere? Rhizosphere is a small area around the roots. So what happened, plant excrete some nutrients and some secondary metabolite and something. So the bacteria around this utilize this food for their survival and the shelter. So there is a lack of nutrients. So definitely there is a millions of bacteria. So the bacteria fight to each other for their survival, for their food, for their shelter, and then in this process, they know how to kill other bacteria and how they can enzymatically degrade the other bacteria and how can they kill the pathogenic bacteria. And what plant get from these bacteria, they are not getting affected by pathogenic bacteria because these beneficial bacteria kill the pathogenic bacteria. At the same time, these bacteria induce the systemic resistance in plant. So plant can also fight with the pathogenic bacteria, which is not from soil, which is from the leaf. So they train them in the root, and once the pathogenic bacteria come on the leaf, they can also fight with them. So we try to get benefit of this mechanism, and then we isolated several bacteria from the two locations in the Bhavnagar, and one from Dwarika, and one is Newport. And we isolated many hundreds of bacteria, and then we screened up these bacteria for their anti-quorum sensing behavior. So, so for the next slide, now I'm showing like how we came to know this bacteria is anti-quorum sensing or not, because there is hundreds and thousands of bacteria, and we have to screen. So how can we know this which bacteria we should go further, and we can go further isolate the compound which is useful to kill the pathogenic bacteria. So we had a very unique biosensor plate. What the biosensor plate had, we had a Chromobacter villosium, and in that bacteria, what we had, we mutated one gene, which is again responsible for quorum sensing, and they were responsible for AHL production. So for the mechanism we had, we removed the gene, so they are no longer uh, able to synthesize the quorum sensing molecule. So if you look at this plate, it's not exactly what I like. I put the picture, but this is the same bacteria, Chromobacter villosium. So this bacteria is not able to produce any AHL molecule. And so if there is a no AHL production, they don't able to produce the pigment, which is called villosine. A villosine is very unique molecule of this bacteria, which is the result of quorum sensing. So what we did, we removed that quorum sensing gene and provide the exogenous AHL. So they are not able to produce AHL, but once you provide an extra AHL, they are going to bind to the receptor and they are going to produce the villosine. So what we did, we add the bacteria on the plate, w- and before that, we add the AHL molecule. We grow the bacteria without AHL. We plate them. We add the AHL. Now they are able to produce the violacin. What we did, we put the bacteria culture here, and we put the positive control, which is a known quorum sensing inhibitor here. If you can look carefully, this area where there was a bacteria, there was no violacin production. What does it mean? That these bacteria not allow this quorum sensing molecule, which is AHL, around the bacteria present here to bind to their receptor, or they destroy, we don't know yet, but either they destroy the signal or they don't allow them to bind to the receptor. And if just for reference, there's a difference between this white zone and this is clear zone. So if you see here, there's a no production. So there is a no transparent but it's a milky white, which is a reflection of the in quorum sensing inhibitor. If you see the negative control, there is no inhibition of the quorum sensing. And so what we did next, we try to extract the compound, which is, so the whole bacteria, this was a whole bacteria. So we try to isolate the bacterial extract and try to find the compound which is responsible for the inhibition of that. So what we did, we extracted whole cell bacterial extract in the methanol, which have the all compound or metabolite bacteria have, and we directly exported the extract here, and this extract also inhibiting the quorum sensing. Then now we know we have the compound in our solvent which is responsible for quorum sensing inhibition. So to further confirm, as we discussed before, the antibiotic is killing bacteria, and this extract or this quorum sensing inhibition is not going to kill the bacteria. They are going to disarm the bacteria, or they are going to stop the virulence factor of this bacteria. So what we checked here, that we put the antibiotic in one side and put the extract on the other side. As you can see, the antibiotic is able to kill the bacteria, but our compound don't have any effect on the growth of the bacteria, but they have the inhibition of quorum sensing. So before going further, we also quantified the amount of production of the violacin in presence of this extract, and we found almost, once we use just 0.6 mg per ml of the extract, there was almost zero violacin production. So it's completely eradicate the quorum sensing system. So the now question we had, which compound is responsible for that, and how can we isolate this single compound, because we can't treat bacteria with the bacteria because there's a lots of things. So we have to find the compound which is responsible for the inhibition. So what we did the fractionation of this extract. For that, we use the solid phase extraction, where there is a lots of fraction. We fractionate this compound in hundreds of fraction. We checked all the fraction, which fraction contain the active compound, and at the end, we found the one fraction which has the active compound. Then when we went to the GCMS analysis, in the GCMS analysis, we found the retention time at 16 minutes. Then further, we also want to, and they also matched with the known library. So this compound was like 3-benzyl hexahedrous pyramide 1,4-dione. So we wanted to further confirm. Then we also go ahead and check the LCMS analysis, which is also called the ESIMS analysis. We find the mass of the compound, we match the mass of the compound with the library, and we confirm that the compound we have in the active fraction is this compound. So now, since we established this compound is inhibiting quorum sensing, now we have to functionally validate this compound. Is this compound working in the real life or real bacterial infection? So what we did, we got the two Pseudomonas aeruginosa. One, the commercially available PaO1 strain, and one PaH strain, which we currently find from the Harvard Medical College. They give us they isolated these bacteria directly from infected patients, and we collected from them and we checked them. So what we checked their virulence factor production and the ability to biofilm formation. So first, we check the how this compound is affecting the biofilm formation of this bacteria. So we use the different concentration of bacteria, sorry, different concentration of this compound, and we found that the maximum like 50% biofilm inhibition once we choose this compound. And in a similar result, we also found the clinical isolate. So this is very sure that this compound is able to inhibit the biofilm formation. So again, we also check this effect of this compound, the growth of bacteria. So this compound have no growth at all at any concentration in compared to control. So one thing here was very clear that this compound is not killing bacteria. This compound is disarming bacteria. Means the bacteria is no longer pathogenic or virulent. So once we treat this bacteria with this compound, they will be not able to form a biofilm. To confirm more about this biofilm, we also check, like visualize this biofilm, how it's going, and we also confirm the live and dead cell present in this biofilm. So what we did, we used two kind of dye. So one was propydium iodide and one is cytonine. So what these dye have, this dye bind to the live bacteria and this dye can bind to the dead bacteria. So what happens once they bind to the live bacteria, they fluorescent gray. And once they bind to the dead bacteria, they fluoresce red. So as you can see, this upper panel is controlled. So there is a huge number of biofilm formation. Even 24, we checked the 3 time point, 24 hour, 48 hour, up until 72 hour. So the biofilm formation is increase and increase. And once we treated these bacteria with the compound, if you can see the comparison, the number of biofilm cells present in biofilm is very less. Even longer duration, even after 72 hours. Sorry. And there is a no dead bacterial cell here. You cannot see much the dead cells. So we also further wanted to see the physical appearance of biofilm and their architecture. So what we did next, we checked the scanning electron microscopy and atomic force microscopy. As you can see in electron microscopy, these cells are controlled. This is PO1 and this is PAH. And as you can see, the bacterial population and their biofilm and the covered in, they are compactly packed. Once you treat with this bacteria, they are dispersed. And there is a normal biofilm. Bacterial cell is individually spread and they have no biofilm, so it will be easy to eradicate. We also check the thickness of the biofilm in atomic force microscopy, and as you can see, the thickness of the biofilm in atomic force microscopy is very much in comparison to treated one. So now, as we see there and like effect on the biofilm formation, we also wanted to check whether this virulence factor, which is responsible for this production of biofilm and their pathogenicity, how this affect. So we check the different virulence factor production. We check the pyrocyanin production, rhamnolipid production, elastase, and protease activity of this compound. And what we found in both bacteria, once we treated, the production of virulence factor was reduced. So next, we also wanted to check whether this compound have any effect on bacterial motility or not. So what we did, we put the low agar medium, we put the bacteria in the center of middle of the like the plate and allow this bacteria to grow 24 hours. As you can see, this is called swarming motility. So these pseudomonas have very unique property. So how they swarm, they have a very big large flagella. So they swim like a fish and they move all around. So once we treat this bacteria with the compound, their motility is also reduced. What does it mean? That they cannot move from one part to another, or they cannot, their motility is reduced. So other is like ciliary motility, that is also reduced. So once we found that, oh, the virulence factor is reduced, the biofilm formation ability is reduced, and then we wanted to find how this compound is working and where it's acting, whether they are a stopping signal, they are destroying signal and what the effect of the bacteria have. For that, what we did the microarray analysis. So what the microarray analysis is, this is a very unique kind of chip. You have the gene of the control and you have the bacteria. You isolated the whole RNA from the bacteria which is not treated with the compound. You isolated the RNA from the bacteria which is treated with the compound. And you convert them into cDNA, you bind to the chip, you hybridize. I'm taking it short, but it's a long process. But once you hybridize, so this chip has like 5,886 genes. So this is the total number of Pseudomonas gene they have. So we have the all gene for Pseudomonas. So we treated with the compound and hybridized this RNA or the transcript into the chip, and we read and then we analyze the data. What we found, the large number of gene is differentially expressed when we treated this bacteria with compound. So almost 1237 out of like almost 6,000, 1237 gene was differentially expressed, out of which 868 gene was downregulated. That means they affect the, the expression of large number of gene, and this gene is responsible for, and we annotate all the gene which is differentially expressed, and we found the large number of gene is responsible for quorum sensing. As you can see, the number of upregulation and downregulation genes. So then once we have all this data for this gene which is differentially expressed or downregulated or upregulated, then we try to find that which gene is affected in quorum sensing system. So if I again put the map of the quorum sensing system of the Pseudomonas, as I saw before, this compound entered into cell, so they have the quorum sensing synthesis. They were able to synthesize the quorum sensing molecule, and then once we treat this compound, they enter into cell and they bind to the receptors. So they bind to the starting receptor of MBFR. They directly bind to the MBFR, and as you can see, the inhibition of quorum sensing regulated gene was reduced, and that's how the biofilm formation was reduced. And we will successfully reduce their virulence factor, although this compound need further investigation before going to the clinical trials, but this was very encouraging leads. So now we are working on the natural compound as well as synthetic compound for the quorum sensing inhibition in here, and I'm really sorry that we couldn't so the data before we can publish or get patented, but we have very lead molecule which can inhibit the biofilm formation, can reduce their virulence factor, and we will be hopefully we will be able to have some compound which can treat this pathogenic bacterial infection. So thank you very much. And this research was funded by CSIR, Ministry of Earth and Environmental Science, and all this research was done at Central Salt and Marine Chemical Research Institute. I am very thankful to my PhD supervisor, Professor Bhavna Jha, and my very intelligent scientist, Dr. Avinash Mishra, which was there always for the all research and help. I'm also thankful to my co-authors of the mini paper and my wife, Dr. Ankita Alexander, for all her help. And I'm also very thankful to my parent lab, Professor Lawrence Rahme, and all my colleagues, especially Dr. Arunava and Dr. Mariana, who was Dr. Mariana actually I was working on this drug molecule, and we had a very good lead, and we are going to publish very soon. And I'm very thankful to RSD for their all help in the lab.
Thank you very much. Thank you very much, Dr. Vijay Singh. We feel proud.
Thank you.
We feel proud since you are the student of, you have been the student of biotechnology of this university. It was a really very good presentation on bacterial communication and crosstalk, disease treatment and view.
Thank you, sir.
It was a nice story on relationship between host and parasites. Though this story was very agnostic, but very informative and lucid. You presented it very nicely. We came to know about good and bads of the bacteria, covering many aspects like unjudicious use of antibiotics with a special reference to our country, India, development of multi-drug resistance and quorum sensing etc. You deserve all appreciation. In future, we hope to have more cooperations. Our students and researchers have been highly benefited. Now I open this seminar for discussion if at all there is any question from any corner.
Hi, sir. Thank you for the nice talk. I have a very nice question.
Yes, sir.
So can the effect of this biofilm formation be reversed by the quorum sensing?
Can you put it again, sir?
My question is, can you reverse the effect of biofilm formation through quorum sensing?
Yes, of course we can reverse. So what basically now we are doing, so we now know the gene responsible for the quorum, and we know the target. We actually basically now we are working on the target, etc. So we have the target receptor. So once you block the receptor, so what happens? Once you mutate the receptor, like there is a three main receptor in the body. One is that, one is that, and one is that. So once you remove the one of the system, like mutate the gene from the system, there will be no able to pass information. But once you have the external gene by a plasmid or the expression into the cell, or you add the exogenous signal, you can reverse the particle signal.
Another thing, I'm just curious to know, I know you cannot actually, do you believe there is any role of extracellular vesicles that can package the signal molecules that is being secreted from any bacteria, like your Adaptations and all those, and then the intracellular communication and all those?
Yes. Actually, we are working, and there is some, we have some paper long ago from our lab from here. So there is some signal molecule which is packed in the extracellular vesicle, and once they travel to the full host cell, they choose this vesicle as the packed quorum-sensing molecule to release on the surface of the host cell, and it is easier for them to invade. They don't have to wait longer to reach their quorum or attack at the same time. They store this quorum-sensing molecule, and they went to the cell, and they release there, and they can invade the cell. But we are not very much working on that right now. Our main focus is on the inhibition of these targets so that we can stop the virus. But yeah, definitely they are doing like this. But we don't have much detail on that, but yeah.
Just extending to this question, do you know any protein that express on the membrane of extracellular vesicles from bacteria?
No, I don't have any about that, because the expression of the protein on the extracellular vesicles, it says no direct link on these, but I'm not aware much about this.
Okay.
Thank you so much. Thank you.
Okay, thank you, Mr. Vijay. Thank you to all. So it's now over this session.
It must be night in his place. Late night in his place.
late night. अब देखो दूसरा पागल नहीं है, दूसरा सेशन है और क्या? देखो जस्ट वहाँ तो हो गया होगा 6:15 3 बज रहा होगा रात का।
वही तो कह रहे हैं सुबह। तुम लोग जो बड़ा जिनका इंजीनियर हो,
अमेरिकन अभी वो लोग का रात का 2:45 होता होगा।
तुम्हारा अच्छा कर
Due to time constraints, we'll immediately begin the technical session 3. The theme of this session is disease models.
Others?
And the chairperson for this session is Professor P. Singh. So
निहाल को ही कर दो
चलो I will take it.
Okay, Professor Nihal will be the chairperson of this session, and this session will be reported by Mr. Rajesh. We have one invited speaker for this session. He is Dr. Sumit Murad from University of Cincinnati.
बहुत जगह से अप्लॉज आ रहा है वेल ऑर्गेनाइज्ड जो बहुत इंफॉर्मेटिव है.
We now welcome our speaker of third technical session, Dr. Sumit Mura. Dr. Mura completed his master's research from National Environmental Engineering Research Institute, CSIR, Nagpur. He then moved to pursue his PhD in biomedical engineering from Center of Biomedical Engineering at Indian Institute of Technology, that is IIT Delhi, and All Indian Institute of Medical Sciences, AIMS Delhi, under the supervision of Professor Saurabh Ghosh and Professor Alok Roy. He completed part of his doctoral work at Center for Medical Devices in Ireland under the supervision of Professor Abhay Kuram at NUI Galway, Bandit.
He then moved to Tufts University in Boston, USA for his postdoctoral research with Professor David Kaplan, and is currently working as an instructor in the Division of Orthopedic Surgery at Cincinnati Children's Hospital Medical Center and University of Cincinnati College of Medicine. Over to Dr. Sumit sir for giving us detailed information on the topic use of tissue engineering strategies for development of in vitro disease models and regeneration.
Hello. Am I audible?
Yes.
Okay. So thank you for the introduction. Very informative talks before me. So a lot of talks from fundamental sciences, and I'll be presenting something that is towards translational medicine. So the topic of my talk is use of tissue engineering strategies for the development of in vitro disease models and regeneration. So we have been looking at different substitutes for human tissues, as humans cannot regenerate. When we have diseases, trauma, or injury, our organs or tissues get damaged and they cannot be replaced by the body itself. So we have been looking for decades, I think, for autografts, allografts, and xenografts, but all of them have their own disadvantages. Like autografts create comorbidities and risk of infection into other sites. Allografts have their immune compromising the patient. Also, they pose the risk of transferring diseases from one patient to the other, while xenografts have mostly been successful in a little bit of clinical trials, but mostly unsuccessful, and they have been lethal in many clinical trials. So we have thought about, about tissue engineered grafts in which we can use the patient cells itself to create a tissue or organ that can be finally transplanted into the patient itself. So this will pose minimum immune response or no immune response as we envisage. and there will be full functional compatibility of the tissue with the patient. The paradigm of tissue engineering involves isolating cells from the patient itself, growing them in vitro, then putting them onto the scaffolds, having a functional tissue, engineered tissue or organ, which can be then transplanted back into the patient itself. Um, so there can be many applications of tissue engineering. The first one involves tissue regeneration. The second one involves a more fundamental kind of research in which we can understand the developmental biology of how organs or tissue develop or embryonic development. And the third and very important application of tissue engineered models is that we can create models of diseases in vitro that are very hard to understand in humans or in their animal models, because animal models have a lot of complications and disadvantages, while humans can be very rare to study for some diseases that are actually rare in the population. So why to go for an in vitro model? So to discuss that, a lot of fundamental scientists will differ from me, but we have to accept this, that the animal models that we use right now are a lot different from humans. The animals have a very different genetic makeup than us. The molecular mechanism of a lot of diseases that we study in animals are totally different than in humans. There are a lot of ethical issues people usually demonstrate in front of our hospital here in Cincinnati. Uh, there can be differences in the results because of animal behavior as they are kept in captivity, and there are other factors, infections, and other risks when raising the animals. As well as these animal trials are very expensive. We work on farm pay government guidelines. So across the globe, a lot of nations, including India, European Union, and Israel, have banned the production and import of cosmetics tested on animals. We all know this has been like for 10 years in India, I think. So companies are looking for other models that are more humane to test your products on. European Union is thinking of a complete blanket ban on animal testing for some cosmetics and other products. Other countries around the world are also looking for banning these animal trials. So why and how can these in vitro disease models can help us? So the first most important advantage of in vitro disease model is that it can help us understand the whole pathology of rarely known diseases. There are a lot of diseases which are very common in the human population, whose pathophysiology is very poorly understood because there are so many factors when you study the disease inside a human or animal model. So in vitro, you can pick up one factor and study that in depth. It can also be used for devising new treatment therapies for diseases, as well as for screening drugs in pharmaceutical industry, and specifically for patient-specific drug screening, which can lead to personalized medicine applications of this technology. So I'll be discussing two disease models. Basically, the first model is an in vitro disease model of osteoarthritis, which was published as a cover page article. So osteoarthritis is again a very common disease. 70% of the population around the world is hit by it by some time, especially elderly people gets affected, and the mobility is very poor because of this disease. The pathophysiology is known, but it is very complicated, and there are no drugs that can treat this disease. It's mostly because of aging. So the pathophysiology of osteoarthritis that is known till date is because of an pro-inflammatory response to any mechanical injury or aging problems in the knee. The pro-inflammatory cytokines IL-1 beta and TNF alpha play a very important role in triggering this disease. And if we can manipulate these two pro-inflammatory cytokines in vitro, we can maybe able to develop a disease model of osteoarthritis. So keeping in mind this, the triggering molecules of osteoarthritis, we thought of a system in which we can have a controlled release of TNF alpha and alpha beta in vitro. So we thought of a system that can mimic the biomechanical properties of cartilage as well as give this biochemical trigger for the disease development. So we thought of silk because it's a very biocompatible biomaterial. Plus, it has very good biomechanical properties that mimic the mechanical properties of cartilage, and it can be chemically modified with certain chemistries to introduce molecules, sulfate moieties in the silk chains. So these triggering molecules of TNF alpha and alpha beta are stored in the extracellular matrix, and they are bound to sulfated glycosaminoglycans. So if we can mimic this chemistry in vitro, we would be able to make a model where we can introduce this disease, induce this disease. So we use diazonium coupling chemistry to introduce sulfate groups onto silk fibrin chains. This is a very general chemistry that is used for modifying protein chains. We looked if this will work on silk, and bioinformatically we calculated if TNF alpha and alpha beta will then be binding onto this modified silk chains, and it showed that they were both binding with high binding energies. Then we actually did the diazonium coupling reaction of silk, and we found that it was getting modified as seen through both UV visible spectroscopy and FTIR. Then we also looked at how it was binding to the silk chains, these alpha and beta molecules. So it was binding as a function of time over a very short period of time, just 2 hours. So it was a very strong binding that was seen with alpha and beta. Then we studied the release kinetics of alpha and beta from these silk scaffolds into the media that they were dipped in, and it was a controlled release over multiple days. The data here shows just for some hours, but it was we had a controlled release of these molecules TNF alpha and alpha beta at least for a month from the system. Sorry. Oh, slide is not moving. Can you see it now? Okay. So then we cultured cells from patients. We isolated cells from human patients that we got from AIMS, New Delhi. And we cultured these primary chondrocytes onto our scaffolds that were having TNF alpha and alpha beta, and had a controlled release over 28 days. We actually saw the hallmark of osteoarthritic chondrocytes in vitro, which are extensive expression of cellular philopodias, which gave us the confidence that we were able to mimic the disease cellular pathophysiology in vitro. Then moving on, we did some histological analysis, and we saw that the cellular morphology was changing when the disease was induced in vitro, as well as the GAP production was released, which is a hallmark of osteoarthritis again. Then we tried to compare the gene expression profile of these tissue engineered disease model and the healthy model that we developed in the lab against osteoarthritic patient biopsies. So we first took the biopsies from the patients during their total knee replacement surgeries and characterized them. They were having all the hallmarks of osteoarthritis as seen in histology. And we then run a microarray analysis or whole genome profiling on all of these samples. Interestingly, our disease biopsies were mirroring the gene expression profile of the disease model that we created in vitro, while the healthy models were mirroring the gene expression profile, whole genome expression profile of the healthy tissue. This was a very interesting, as this was the first study and has been a benchmark for the later development of osteoarthritis models in vitro. Then we, because you have to quantify through RT-PCR the gene expression profiles that we have got through microarray analysis. So we just confirmed that it was going correct, and it was validated through RT-PCR, most of the gene expressions. I'll skip the other data that gene analysis that we did, and finally skip to the conclusion of this model. So till date that we did this study, it was thought that the expression of MMPs and collagen molecules play a very important role in the triggering of this disease or the progression of this disease, while our model showed that the tissue inhibitors of matrix metalloproteinases play a major role in in the actual progression of this disease, as they can control how matrix metalloproteinases are degrading the tissue, the cartilage tissue. So this was a very important finding and was published as a cover page article in Tissue Engineering Part A. Moving on to the next disease model, which was of macular corneal dystrophy. So this is again a very different disease from the first one. This is a corneal disease which very heavily affect Indian population. It is an autosomal recessive disorder and is caused due to a mutation in CHST6 gene, which is responsible for the sulfation of glycosaminoglycans, especially keratin sulfate in the corneas. So this is one of the most prevalent corneal dystrophies and very badly hit the Indian population because of the inbred nature of our population, as I think a speaker earlier discussed, because of the caste system and other things. We are highly inbred, and this is a very big problem for this disease. So we have a very high prevalence, and nobody else in the world is studying this disease like in depth, because nobody else is getting affected that much as we are. So the pathophysiology, we know it is caused due to a mutation in CHST6 gene, but the whole pathophysiology of this disease is not known. It's very poorly understood. So that's why we thought of creating a disease model to more probably have more insight into the pathophysiology of this disease. There is no successful treatment of this disease again, as the disease generate abnormal sulfated keratin sulfate molecules that form clumps in the cornea and form haziness in the cornea, and the person that is affected due to this disease goes blind by the time he or she reaches adulthood. So there can be corneal replacements that can help the patients, that can be treated with penetrating keratoplasty and deep anterior lamellar keratoplasty. But again, this corneal transplant developed haziness, and these persons go blind again within the span of 2 to 3 years, as the gene expression, you cannot stop the gene expression. So there is no surgical treatment, no transplant treatment available. There are no chemical molecules or drug molecules that can treat this disease. So in order to, if we can understand the pathophysiology of this disease, that will help treat a lot of patients around India. So to create a disease model of macular corneal dystrophy, we just targeted the enzyme that was getting inhibited in the disease itself. So we took a chemical inhibitor of carbohydrate sulfotransferase, which was PAPS, and looked at how well it is a toxic molecule, PAPS. So we first studied, we took it in low doses, and we studied the toxic effect on primary corneal cells from goat. This was a primary culture, and we saw that this toxic chemical could initially slow down the metabolic activity, but later on the cells could survive easily with this chemical, and this would affect the production of keratin sulfate molecule, which we were trying to achieve. So we cultured these cells over a span of 14 days. In our disease model system, it was a 2D disease model in which we introduced this chemical into primary cells at different concentrations till 50 millimolar. And we saw that as we increase the concentration of this chemical, there were production of abnormally sulfated keratin sulfate molecules, uh, while when there was no concentration of this chemical in the controlled healthy controls, we can get a lot of healthy keratin sulfate molecules, which you can see in these two images. So we were first getting a healthy model, and then we were creating a disease model. It was further studied the formation and abnormal of normal keratin sulfate molecules and abnormally sulfated keratin molecules through Raman spectroscopy, and it confirmed the formation of abnormally sulfated keratin sulfate molecules in the disease model. We further went on to study how the ECM production was getting affected, and we saw that due to the introduction of PAPs, the GAG production was inhibited as we increased the concentration. Then we studied the gene expression profile, and we found that the WNT pathway was actually affecting and was responsible for slowing down the expression of a lot of GAG molecules, as well as integrins and cadaverins, as well as that of CHST6. Then on, we again went on to study protein interactions and gene ontological profiles, which finally led us to the conclusion that inhibition of sulfation in vitro regulates, or in the actual disease, down regulates the WNT pathway, and this WNT pathway can reduce the expression of MMP molecules, which can actually dissolve the abnormally sulfated keratin sulfate molecules. So because the expression of matrix metalloproteinases is hindered in this disease, the abnormally sulfated keratin sulfate molecules cannot be cleared, and the haziness builds up as the person is growing. While the downregulation of integrins and cadaverins result in the disruption of epithelia and Bowman's layer of cornea, and the cornea may get detached actually over as the person is growing. So we could study the whole pathophysiology and give in-depth insights into pathophysiology of the disease with this disease model. This model system work was also published as a cover page article in Nature Laboratory Investigations. So now moving from disease models to tissue engineered systems that can help regenerate tissues. So the first system that I will be talking about is a hybrid injectable hydrogel system for tissue engineering of intervertebral disc. So this is again related to orthopedics. Lower back pain is a very big problem, socioeconomic problem throughout the world. It affects, I think, the whole population. We all get affected with the lower back pain problem at some point of time in our lives. It is caused due to deorganization of extracellular matrix and loss of water in the intervertebral disc in the lower vertebra. Again, this disease is poorly understood, though it is very common. The pathophysiology is very poorly understood. Doctors give you, symptomatic treatments of painkillers. If the disease gets chronic, they can surgically remove your disc and fuse the adjacent vertebra, which further hinders your mobility and the quality of your life. It is also a very dangerous surgery because there is risk of partial to full paralysis to the patient. So there is no, fully functional surgical treatment available. There are no drugs available. Doctors refer you some over-the-counter drugs that might help or might not help because most of the clinical studies are not conducted properly for this disease. So there is a lacuna in the treatment strategy for this disease. So we would like to remove the surgical part of this treatment strategy, and that's why we thought of an injectable system that can be developed to treat this disease. So what kind of drug molecules that we can use, as some targeted therapy is not available? So we looked at over-the-counter drug medications available for osteoarthritis to lower back pain, and we saw a drug with a molecule that is very commonly prescribed by doctors called N-acetyl-D-glucosamine. So this glucosamine molecule is recommended by doctors very often to osteoarthritis patients. They give you a powder. The very important thing to note down here is that if the concentration of N-acetyl-D-glucosamine, the systemic concentration increases, it can actually increase the disease. So there is a very important take-home message here that the concentration should be controlled. So we thought of a controlled release system that can deliver this molecule to the affected IVDs. And as this is a precursor of GAG molecules, it can increase the GAG production and thus also increase the water imbibed in the disc and retain the biomechanical properties of the disc again. So we thought of a silk and silk system again. So I worked on silk a lot as biomaterial. So we created hollow microspheres of silk, and we introduced this drug in these hollow microspheres, embedded then into again a silk hydrogel, and studied and optimized the rheology of this silk hydrogel so that it can be injected through a minimal invasive surgery into the defect sites in the intervertebral disc. So how did we create these silk microspheres? So we took polystyrene beads, micro beads. We coated them with polyol lysine, the same image. We then coated it with silk and then dissolved the polystyrene coat so that we can get a hollow silk microsphere. So why we were looking at silk hollow microspheres? Because the payload capacity of these hollow spheres is way better than solid spheres. So that's why we developed these silk hollow microspheres. Then we started the stability and the removal of tetrahydrofuran, which was used to dissolve polystyrene. These microspheres were stable, and also the tetrahydrofuran was removed after washing. So during the disease of lower back pain, the pathophysiology develops so that the pH and ionic strength of the intervertebral disc changes over time during the whole span of the disease. The ionic strength of the fluid that is inside intervertebral disc exceeds the ionic concentration that is anywhere in the rest of the body. So any strategy that is being developed for treating this disease should be stable on these two chemical profiles. So we studied the stability of these silk microspheres, and they were found to be stable. Then we studied the release profile of these of N-acetyl-D-glucosamine from silk microspheres, and we found that there was a controlled release that was happening from these silk microspheres. Then we studied the cytocompatibility, which was a very important study as we were using many chemicals during the synthesis of these silk microspheres. And these microspheres were found to be biocompatible towards human ADSCs as well as bovine nucleus pulposus cells. Then we studied the injectability and rheological properties of this material system. So as you can imagine, we are introducing microspheres that can choke the needle while they are being injected into the patient's body. So we studied the rheology of the whole hydrogel system, and we devised a rheological test that can mimic the injection process. So we provided a shear rate which mimicked the filling of the syringe, then provided a very high shear rate that mimicked the injection process when the material is actually going through the needle, and then let the system sit for 5 minutes, and then understood the rheological properties again. And we found that our silk and silk microsphere system could heal 80% of its rheological properties within 5 minutes, which is very important if you're looking for a system that can biomechanically stabilize your intervertebral disc. So the two important goals of this project were to biomechanically stabilize the intervertebral disc and also induce regeneration in the disc. So the first goal was started with an ex vivo bovine intervertebral disc model. So we took up bovine intervertebral disc and treated them with collagenase, injected collagenase into their nucleus pulposus, degenerated it, and then filled it with our silk and cell system. As you can see in the mechanical testing results here, our 1% silk hydrogel system and 2% silk hydrogen system both could recover the biomechanical properties to the native levels within 5 to 10 minutes of the injection. So this was a really big deal because nobody has shown this earlier. Secondly, we cultured these ADACs, human ADACs, onto our system, silk and silk hydrogel system, which was carrying an acetylglucosamine. And we saw that as we uh, delivered this molecule, the ECM production was getting enhanced, especially the GAG molecules. So the system was giving us both biomechanical stability and regeneration in the tissue, which was our goal. So it was studied with gene expression profile and further through histological imaging with safranin O and alshenbu, and the uh, model system showed us increased regeneration in all the cases. So finally, we came to the conclusion that when we are delivering N-acetyl-glucosamine, it increases the production of TGF-beta, which is the triggering molecule for the initiation of production of glycosaminoglycans. And if we can regenerate glycosaminoglycans in the intervertebral disc, the water imbibing properties of the disc will increase. That will result in reduced pain and basically treatment of the disease itself. So this study was also selected as for the poster prize in biomaterials, which is an LTVL journal. The final study that I'm going to discuss is what we are currently pursuing in my current lab. It's a system that involves in-solution mineralization of alginates DCM bioink for 3D bioprinting and injectable purposes. So the disease that we are looking at here is called avascular necrosis. It has resulted due to the interruption of blood supply to the femoral head, which results in tissue necrosis. It finally leads to the collapse of the femoral head, that is the hip joint. It can be caused due to a lot of etiologies, starting from trauma to steroid abuse, radiation therapy, and excessive alcohol use. The diagnosis of the disease is very hard because this cannot be diagnosed through X-ray. Even at chronic levels, it can only be shown through MRI, diagnosed through MRI. And there is no successful surgical or drug therapy that is available to slow down the progression of this disease. Most of the times you don't know, even the doctors don't know that you have this disease because the diagnosis is so poor. Especially in pediatric patients, it is very hard to manage. As in elderly patients, if your joint is destroyed, you can go for a hip replacement surgery, total hip replacement surgery, and get a metallic implant placed. That will ease your mobility as well as increase your quality of life very much. But in the case of pediatric patients, this surgery is not recommended as children are growing. They will need multiple revision hip replacement surgeries throughout their childhood that will significantly decrease their mobility as well as reduce their quality of life during their childhood. So surgeons do try to avoid total hip replacement surgeries for this disease in pediatric patients. So a potential solution can again be an injectable therapy that is minimally invasive, that helps in regeneration and also supports biomechanics of the tissue. So when we talk about any orthopedic tissue, the biomechanics plays a very important role because the whole system actually supports our body to move. So whenever we are talking about any orthopedic regeneration, we also talk about biomechanics, the regeneration or restoration of biomechanics. So there has been a lot of research going on in bone tissue engineering, but none of the treatments that are being researched are biomechanically stable. So that component of any tissue engineering strategy is still lacking, though there are many hydrogels available in the market commercially that are used to heal bones nowadays during surgeries. So a guided bone regeneration system should, for this disease, should have osteogenic signals, angiogenic signals, biomechanical stability, and also it should create a mineralized composite when the bone is getting regenerated. So in a lot of systems, there is a disadvantage that they cannot be mineralized over time. So if the system, your biomechanical biomaterial system is not being mineralized over time, its biomechanical properties are not going to be sufficient to support the body weight basically. So in this system, we are focusing on regeneration of mineralization as well as regeneration of other tissue components as well. So we have designed a chemical strategy with which we synthesize hydroxyapatite molecules chemically in situ onto decellularized extracellular matrix that is isolated from bone, and we make a injectable system that can be bioprinted as well as injected into the patient with a minimal invasive surgery. So this is the first report where anybody has tried to print cells and organoids within a mineralized bioink. So there have been commercially available products in which you put hydroxyapatite, which is the mineral component of bone, in the form of bone cement and other commercial products that are available. But these are put just without cells. The cells are never embedded in the cells. But our strategy is to embed live cells and organoids into this mineralized bioink. So our system works is created through bone decellularization. We decellularize the bone through a patented protocol that works both for cartilage and bone in our lab. The protocol also decalcifies the tissue because that is a very important component, because if the tissue is mineralized, it cannot be injected. So our strategy is to partially mineralize the tissue so that it is biomechanically stable, as well as it can help the biomaterial system to be injected. When we decellularize the tissue, the GAG content, the collagen content, and small protein content of the matrix was preserved. We also introduced angiogenic decellularized extracellular matrix into this system in the form of aorta tissue, which is a very good reservoir uh, of angiogenic factors. So we decellularized aorta tissue, and during decellularization, both the matrix and elastic network was preserved. Then we studied how our chemical reaction is able to make these hydroxyapatite crystals onto the decellularized matrix, and the controls was alginate, which was the second part of our system. Uh, This system clearly showed when we mixed a decellularized matrix, the protein molecules could act as a template uh, for the nucleus for the hydroxyapatite crystal growth and its nucleation, and you can clearly see that in DCM molecules there were clear hydroxyapatite crystals that were created on top of these DCM templates, while in alginate there was formation of calcium phosphate that was amorphous in nature. This was confirmed by, again, by a lot of chemical studies, chemical characterization studies. The elemental analysis showed the formation of calcium phosphate in both the samples, but the presence of hydroxyapatite was confirmed through FTIR studies shown here only in the DCM samples. And there was no hydroxyapatite formation in the on the alginate samples. As you can see, in the DCM samples, the 211 plane was very clearly visible, while in the alginate samples, there was no formation of hydroxyapatite crystals, which confirmed that DCM molecules were acting as a template for the nuclear formation and crystal growth of hydroxyapatite. Then we visualize this hydroxyapatite formation through atomic force microscopy, and we could see the hydroxyapatite crystals growing onto the DCM molecular chains. So this was a confirmation that the chemical system was working towards the formation of a mineralized biomechanical ink that can support the biomechanics of the degenerating femoral head. Then we studied the printability and injectability of these biomechanical inks that were found to be very good for both 3D bioprinting purposes as well as injection purposes. We adopted the same rheological injectability test in this case too, and we found that within 5 minutes of injection, our system could regain its biomechanical properties, which is very important when we are looking for stabilizing the hip joint, because the hip joint is the most important joint in the body that takes up the whole body weight. Then we are going ahead with the bioprinting of this biomechanical ink system with human mesenchymal stem cells and iPSC-derived human endothelial organoids, and we have found that the hydroxyapatite content is not hindering the viability of the cells when we are printing it, and the cells are surviving till day 28, which is one of the only studies, very few studies that have shown the survival of live cells and organoids in any mineralized ink systems. So we hope that our ink system will also help in regeneration through angiogenesis and osteogenesis. This study is currently undergoing, and we look forward to some good results through this study. I would like to acknowledge all my institutions, all my mentors, and all my students who have helped me throughout all these studies. And I would also like to thank the organizers of the conference and my friend Kali Charan, who kindly invited me to this conference. Thank you so much, and I'm open to questions now.
Dr. Murab, Dr. Murab, it was an excellent presentation. Uh, Apart from orthopedic tissue, can some other tissues like the soft tissues, for example, the beta cells of the pancreas, are you also looking into, or the field is looking into some tissue framework where we can put the beta cells and they survive. They are not attacked by the immune cells of the host.
So yeah, there can be different applications of these injectable systems. We are not looking into soft tissues because I am in orthopedic surgery department, but there are studies that are being carried out for tackling those kind of problems. But we are not studying, unfortunately.
Thank you, Dr. Mora. Thank you. So, thank you, Dr. Murab, for speaking at length about the tissue engineering part. We'll start with the next session, and I've asked all the oral speakers to be ready. So in this session, we'll have oral presentations, and we are having altogether 5 speakers. First, Dr. Ankita Alexander. Second, Dr. Vanita Kamath. Dr. Ankita is from Harvard Med School. Dr. Vanita Kamath is from Mumbai. Dr. Sushobhan Banik is from RK College, Madhubani. Dr. Putul Banerjee from St. Joseph's College, Bangalore. And Mr. Pranavesh Mandal from Central University of South Bihar. All of you are requested to keep your presentations ready. Since we are running short of time, you will just have 7 minutes for presentation plus 3 minutes for discussion. Please keep time, keep your presentations ready, and we start with Dr. Ankita Alexander. If Ankita is ready, you may start.
Yeah. Am I audible to all?
Yes.
Share. Good morning, everyone, and thank you so much, Dr. Parul Banerjee, for introducing me. Today I am going to talk on host microbe interaction lead to changes in the physio-biochemical activity of plants to adopt nitrogen starvation condition. Oh, I need the... So you all can see my screen?
Yes.
Yeah. Thank you. Good evening, everyone. Myself, Ankita Alexander, and today I am going to talk about host microbe interaction lead to changes in physio-biochemical activity of plants to adopt nitrogen starvation condition. So my talk is mainly concerned about how the microorganism help the plants to adapt for the abiotic stress condition. So first of all, the microorganisms are of the 2 types, as we know that are some are good bacteria and another are the bad bacteria. As animals, in plants, there are also some good bacteria which are present at the rhizospheric region of the plants. They are called as a rhizobacteria. Due to their plant growth promoting activities, they are called as a plant growth promoting rhizobacteria. Some of these qualities include ACC deaminase activity, osmolyte production, nutrient accessibility via the production of siderophores, and phosphate solubility. They also include in the ion exclusion mechanism, nitrogen fixation, and some phytohormone productions like indole acetic acid and gibberellins. They also act as a biocontrol agent. Now, for my study, I choose the peanut plant for the study because it is one of the most important edible oil crop in India, and India is rated as the second largest producer of the groundnut in the world. But the various abiotic stresses like heat, drought, salinity, and poor soil quality affect the production of groundnut in the India, specifically in the Gujarat region, because there is a high salt concentration present in the soil. This is the cartoon which is showing the effect of biotic and abiotic stress on the plant health and growth in the absence of PGPR and in the presence of PGPR. So in the absence of PGPR, there is enhancement in the chlorophyll loss, the transpiration decrement in transpiration rate and photosynthesis, the increment in generation of reactive oxygen species, and the excessive ethylene production take place. So all these factors cumulatively degrade the health and the growth of the plant, while in the presence of PGPR, as I discussed in the previous slide, the all factor cumulatively enhance the growth and overall production of the plant. So for my study, I use the halotolerant rhizobacteria named as Bacillobacterium sorastriensis JG06, which is isolated from the rhizosphere of a halotolerant plant, Salicornia brachiata, in the Saurashtra region of the Gujarat, and it was previously described for our lab members for their plant growth promoting traits like the nitrogen fixation, ACCD aminase activity, etc. So I inoculated this bacteria to peanut plant in the hydroponics condition, in the normal condition, which I termed as a control plant, and in the stress condition where the nitrogen in the media is absent. And the morphological parameters like root length, shoot length, fresh weight, and dry weight were measured at each 7 day up to 21 days. And this is the graphs which are showing the changes in the shoot length, root length, fresh weight, and dry weight up to 24 days at each 7 days interval. And we can clearly see from the graphs and for this photograph that after 21 days, the plant which were inoculated with the bacteria have a higher shoot length, root length, fresh weight, and dry weight in compared to uninoculated plant in the control as well as stressed condition where the nitrogen deficit is in the media. To check the effect of this bacteria on the photosynthetic pigments of the leaves of the peanut plant, the total chlorophyll, chlorophyll A, chlorophyll B, and carotenoids were measured. And we can see that the significant enhancement present in the total chlorophyll, chlorophyll B, and also the increment in the chlorophyll A and slight increment in the carotenoid is present in the plants which are having the bacteria in compared to uninoculated plant, both in control condition, which is nitrogen supplemented, and the stress condition, which is in the nitrogen deficit case. To check its effect on the physiological and biochemical status of the plant, the electrolyte leakage, membrane stability index, MDA content, proline content, total amino acid, and total soluble sugar were measured, and we can clearly see that there is a significant decrement in the electrolyte leakage, MDA contents, proline content, and the TSS were observed in the plants having Bacillobacterium sorastriensis in the control and stress condition, while the membrane stability index and the total amino acid concentration were enhanced in the plants having bacteria. Resulting by these experiments, we can say that the bacteria support the plant to fight against the abiotic stress condition compared to control condition. Now, to check the effect of this bacteria on the metabolic level, the total phenolic compound, total flavonoid contents, total antioxidant activity, and DPPH scavenging activity recorded. And you can easily see that the total phenolic compound, total flavonoids, and antioxidant activities were significantly higher in the plants having Bacillobacterium zorastriensis, because the plant secretes some secondary metabolites in the stress condition to scavenge the reactive oxygen species, which हाँ हाँ yes, but yeah, I have just 2 to 3 slides more. So the metabolic activity is also increased. Uh, these are the three graphs which are the multivariant data analysis by the PCA, heat map, and the partial least square discriminant analysis, which is showing the correlation between different parameters in control and stress condition in presence of bacteria and absence of bacteria. To check the effect on the molecular level, I isolate the RNA and convert it into the cDNA in control and treated plant, and go for the suppression subtractive hybridization and create the clone library. And when this clone library were divided, we get the maximum gene related to hypothetical or uncharacterized group, followed by the metabolism and photosynthesis related group. And these genes are correlated by the using CLC analysis. So conclusion of the study is that in this study, we checked the effect of Bacillobacterium zorastriensis on peanut plant under nitrogen stress condition. We measured the physiology, metabolic parameters, and we can see that plant has changes in the physiology and metabolic parameters to adapt in the stress condition. We also prove that host has a differential expression of gene after interaction with plant. So the use of this PGPR for field condition and agriculture needs the extensive research on the molecular and metabolic level. All this work is published in Plant Physiology and Biochemistry Journal. And thank you so much for CSIR, CSMCRI, ACSIR for all my academics and research. And thank you so much, the University Department of Zoology, to invite me and present my work here. Thank you so much.
Yeah, madam. Yes, ma'am. Yeah, can you see my screen? One moment. I'll start sharing the screen. Kindly check. Do you see the screen? Okay. Yeah. Shall I begin, madam? Hello. Shall I begin? Thank you. Am I audible? I'm audible, no? Yeah. Thank you so much. Yeah. Good evening, everybody. I'm Vanita Kamath. I worked under Dr. Bhaskar Saha in the National Center for Cell Science, Pune. And this is the work of small work I'm presenting here. The topic is RSK inhibitor SL0101, which enhances the anti-inflammatory but not pro-leishmanial influence of CD40 in susceptible mice, bulbsy mice. So our laboratory has previously established CD40 induced P38 kinase, MAP kinase, and ERK1 and 2 kinase are reciprocally regulated. We have previously shown in Leishmania major infection CD40 induced P38 MAP kinase activation or the phosphorylation increases, and IL-12 synthesis expression also increases, while as in the ERK1 and 2 phosphorylation and IL-10 synthesis decreases. So this reciprocal regulation of CD4 is because of the CD40 ligand interaction. So I'm moving further in the work directly. So we all know that CD40 is a transmembrane protein present on the macrophages. CD40 signaling is initiated due to the CD40 ligand stimulation. Now, in this case, we are mimicking the CD40 ligand by use of anti-CD40, which is derived from the cell line, or even it is available in the pure form. So, but it has been observed that whenever there's a high anti-CD40 dose stimulation is being given, P38 MAP kinase gets activated, and low anti-CD40 dose ERK MAP kinase get activated. I'm looking out at the MAP cap kinases, which are downstream of MAP kinases. This is MAP cap kinase 2, which is under the P38 MAP kinase, and ribosomal S6 kinase under the ERK1 and 2. I'm going to focus only on RSK1 and 2 at the moment. So moving further, when we did the check MAPCAP expressions in general in L major infection, this is uninfected macrophages and this is infected macrophages, we found the expression remain unaltered. Further, we did the time kinetic studies on macrophages and at different 3 doses of anti-CD40. It has been observed that the activation is strong enough at around 5th minute, and it lasts till 30th minute. So we further considered 15 minutes as a for our stimulation. All the stimulations were given at the 15th minute. So if you can just check here how there is a differential phosphorylation happens because of the ERK docking. What has been observed that this 359, serine 359, threonine 359 and serine 363 residue phosphorylated. These are the Western blots you can see. In infected macrophages, the phosphorylation is increased in compared to the uninfected macrophages with the anti-CD40 doses, while as serine 380 and serine 227. The serine 227 is the residue, the final residue for the phosphorylation. We found the phosphorylation and the activation decrease in infected macrophages when we compared it with the uninfected macrophages. So we also used this particular inhibitor available out there in the market. There's SL0101, and we checked it using various different concentration, and checked the 3 different, even 4 different residues of this RSK, and we found the inhibition. Then we utilized this particular concentration further. We also checked the upstream kinases, that is P38. If you can just check here, RSK inhibition decreases the phosphorylation of P38. At the same time, it decreases the IL-12 cytokine as well as IL-12 as well as TNF alpha. Pro-inflammatory cytokine synthesis decreases, while as opposite it happens, its own upstream kinase ERK1 and 2 phosphorylation increases, and at the same time the IL-10 synthesis also increases. So this was going with what we had previously checked. So we again checked it along with the ELISA. In ELISA, though the IL-10 ELISA was not much conclusive, IL-12 we found a significant decrease in IL-12 synthesis using the RSK inhibition. Further, we also checked this parasite burden and did the GINSA staining of macrophage cells, and we found similar to our previous results, like using RSK inhibition, still you can see the parasites persistence was there because RSK inhibition has anti-inflammatory effect. Then this is the distribution curve and total parasite load that we can check. So what we observed, in vitro parasites remained there even after the inhibition. So further, we did the in vivo experimentation. So you can see here the Cutaneous leishmaniasis L major infection disease progression was studied by checking the foot pad thickness measurement. This is untreated mouse, we can see, and this is the one which has been treated with the RSK inhibitor SL010. These are the bulbsy mice. So this is progression was done, checked for almost 5 weeks, and what has been observed that in compared to the infected projects, yes, sure, yes. So these are the last 2 slides, and these are the conclusion. What I found out that CD40 stimulation, for the first time we have checked the MAPCAP kinase are activated by the CD40 stimulation. RSK inhibition decreases both the P38 as well as IL-12 cytokine synthesis in vitro, but it increases ERK1 and 2 and IL-10 cytokine synthesis. In vivo also, it has shown the decrease in IFN gamma and increase in IL-4 Thus, RSK inhibition has shown anti-inflammatory function, but in vivo, we couldn't find such a thing. It was not pro-leishmanial, being a anti-inflammatory, as RSK inhibition has and CD40 treatment together has shown less foot pad thickness, lesser parasite load, and lymph node weight. Thus, more studies are needed in this regard of ribosomal S6 kinase and its effect on leishmaniasis. I'm grateful to Dr. Bhaskar Saha, NCCS, DBT for funding this particular work. I'm thankful to the organizers to allow me to present my work here. Thanks a lot, Department of Zoology, Mithila University. Thanks a lot. Thank you so much.
Sure. A very good afternoon, good evening to the senior dignitaries. A very good evening to senior dignitaries on the dais, off the dais, respected senior faculty members, administrative personnel, dear colleagues, and my dear upcoming colleagues. Let me present my screen first. Hope my screen is visible to all of you. Please respond. Today I'll deal with cancerous nutraceutical allicin from the clove of life as my topic of lecture today. Now, as a prelude, these are the topics which I'll discuss about. The modern science and technological advances have not only influenced people, paving the way of enjoyment of so-called modern civilization, but it has also caused serious sufferings with new diseases and severe forms of age-old ailments. The concepts of industrialization, urbanization, globalization, and economic and social developments have led to rapid changes in diets, lifestyles, and have exposed life to pollutants, microbes, parasites, and other agents that have brought about severe forms of diseases and ailments. As a consequence of this, there has been reflected by incidence of disorders and diseases such as obesity, diabetes, cardiovascular diseases, stroke, hypertension, and cancers arising from inappropriate, inadequate, and imbalanced diet, lack of physical activity, and other behaviors detrimental to health. Now, in this era of rapid growth and industrialization, the increase in the economy has by several ways invited several diseases and ailments, as I told before. These are caused due to multiple factors ranging from pollution, lifestyle, microbial, etc., besides therapeutic measures taken in the case of such element has to be brought about several other health and wealth related complications. In contrast to the synthetic therapeutic procedures, there has been extensive research regarding the curative and preventive potencies of various targeted molecules of dietary plant origin, like genistein, diastein, lycopene, imodin, allicin, etc., which have shown great actions to cure dreaded diseases that either caused death or led to several dreaded side effects when treated with those synthetic drugs. Now, cancer is a multifactorial disease, as you all know, characterized by changes in the group of normally dividing cells of the body, gain the ability of uncontrolled growth, causing lump called tumor. Now, this is true for all cancers except leukemia, cancer of the blood. If untreated, tumor cells can grow and invade surrounding normal tissue or to distant parts of the body via circulation and cause tumorigenic growths in those parts. Cancer diagnosis includes physical hematological examinations, biochemical examinations, histopathological examinations, immunological examinations, scanning procedures like MRI, PET, SPECT, etc. Now, cancer therapy includes surgical procedures, radiotherapy, chemotherapy, immunotherapy, endocrine therapy, and gene therapy. The side effects
of cancer therapy are risk of infection, anemia, bruising, bleeding, hair loss, fatigue, sore, sore mouth and ulcers, nausea, loss of appetite, taste changes, and many to name. Now, what happens is that along with the modern system of traditional plant-based therapeutic and preventive procedures have maintained their popularity, demand, and practice because of their easy availability and cost effectiveness. The practice involves various methods of collection, processing of plants and their various products. Now I present you a glimpse of garlic and allicin. Now, allium species of plants are now well known for their therapeutic potential against innumerable number of diseases. These are experimentally proved. Now, this particular family, as you can see, garlic, onion, leek, scallion, shallot, etc. This is the chemistry of allicin. Now, how allicin is being synthesized in the plants. I shall not deal in detail with this. This is the therapeutic potential of allicin. Now you can see that allicin has antibacterial, anti-parasitic, antifungal, immunomodulatory, anti-neoplastic, anti-toxic, antioxidant, anti-hypertensive effects, anti-allergic effects, and these are experimentally determined. These have been experimentally determined. Now, to name a few, allicin against cardiovascular diseases, it has been reported that allicin inhibits key enzymes that regulate cholesterol and fatty acid metabolism. These experiments have been performed in various rat and human model system, human hepatic cells like human breast cancer cells, etc. Now, garlic has also shown to be efficient in reducing cholesterol level. It has also been shown that dietary supplementation of garlic extract causes fibrinolysis. Allicin has been also found to be a very good alternative, herbal alternative to synthetic ones that can fight against microborne diseases. The organosulfur compound allicin, it has very strong antimicrobial activity, which has been shown to work against bacterial, fungal, protozoan as well as viral infections. Owing to the high volatility of allicin, it is supposed to be efficiently targeted into the lungs by incineration as a strategy to cure aspergillosis. Now, allicin against cancer, this is the main part which I am needed. Now, many workers have shown that allicin has multiple roles and multiple strategies by which it can lead against cancer cell propagation. Many scientists have shown that tumor growth could be inhibited by allicin.
Sir, your 10 minutes are over. Please try to wind up.
Oh, sure. Now, I deal with the methodology. What I have performed is that I work on some cell lines. These are the cell lines, and this is the cell culture procedure which I had undertaken. Now, I analyze enzymic antioxidants, non-enzymic antioxidants, analysis of cytotoxicity, and cell cycle analysis by fluorescence protein procedures. Now, these are something which I have found that allicin-treated cancer cells have shown increase in the enzymatic as well as significant increase in NP. Now, as an analysis of cytotoxicity, this is the result I have found that allicin induced cytotoxicity in cancer cell line and reduced the cancer cell propagation. Now, this is my analysis for fat and fat, and it has shown that cancer cell propagation could be arrested at pre-T1 and late T2 phase. Now, as a discussion, allicin, what I found in allicin is that it has shown my study correlated with the studies conducted throughout the globe, and more studies are required to utilize allicin in cancer treatment. More experiments have to be done, advanced techniques have to be followed. Now, one thing I sum up as a conclusion is that this garlic is being considered
sir. The next speaker is Dr. Putul Banerjee from St. Joseph's College, Bangalore, who is my twin sister also. So let us hear from Putul. Putul, please keep time, just 7 minutes, all right?
Thank you, Parul. Yeah, I'll try to keep time and I'll share with you first my screen. Is my screen visible?
No.
Just 1 minute. Is it visible now?
Yes.
Yeah. So today I would like to talk a little bit on the comparative endocrinology of glucose metabolism in vertebrates. But let us first put a disclaimer that this idea and this work is still in its infancy, and we three, me, my sister Parul Banerjee and Kalicharan sir, we are trying to collaborate and start the work. Okay? So we will just try to show some preliminary things that we got to, some exciting, not results per se, but some exciting knowledge about the glucose metabolism, which we got from the comparative study, and this was exclusively in silico. No wet lab experiment was performed. And just when we thought that there was an international, we knew that there was an international conference happening, we started looking into, because we were looking into collaborating, and we started working on this. So in the, is my slide changing? No?
No.
Yeah, now it is. So when we were, the email informing me that this abstract is selected for oral presentation mentioned that we should focus more on the result. But since the origin of what to work and the approach towards this problem, that is quite interesting. I would like to tell more on that, because obviously we have very few things to show as of now. Okay? So the origin of the approach, that is the comparative biology approach. We look at all the different vertebrates, and we, by comparing the system in different vertebrates, we come to certain conclusions, which eventually help us to know more about human physiology. So this approach of comparative biology, I got really interested when I was a PhD student in Banaras Hindu University under Professor K.P. Joy. And I was working with nona peptides, and I was trying to find out the role of nona peptides in the reproduction of a teleos, tetramucil fossils. So my approach at that time was to clone, sequence, and then study the expression profile. While cloning, we cloned a new gene, okay? New nonapeptide gene, which was not reported anywhere else. The new gene was not reported in any vertebrate before. So this is the paper that was published in 2018. This was followed up after the nonapeptide gene was discovered. So once that gene was discovered, we failed to explain the origin for a long time. This gene was discovered just in the first or second year of my PhD. It was 2011 or 12. But we failed to put across the origin of this gene. Then a lot of searches and a lot of efforts went into, and finally we found, as we looked into the very famous zebrafish, which is a genome-enabled teleos, and it is a go-to species whenever you are trying to understand the molecular physiology in a fish. There's a very good fish model. When we looked into zebrafish, again we found the zebrafish has just 2 nonapeptides. So this nonapeptide was not there even in the zebrafish. So while we were trying to decipher the origin, I came across this, although these, the genome duplication and duplication events are well known across vertebrates, and these are actually the reason of diversification and speciation in vertebrates. But for me, it was a little bit new. Although I had heard of it, I appreciated it more. So therefore, I am very interested in understanding biology by taking the comparative biology approach. And as zoologists, we all should do that. So the significance of the comparative biology approach is that it gives us the idea about the expansion and diversification of gene families. So whenever we look into genes, they belong to some family and superfamily, and these families originate due to the events of duplication. Okay? Also, apart from the understanding of the expansion of the gene families, it gives a better understanding of the biology of the diverse vertebrate species and help us to use them as model organism for disease or for some intriguing phenomena like aging and development. So when I was talking about gene family, one gene family that I would like to take as an example is the secretin superfamily. The secretin superfamily includes the pituitary adenylate cyclase activating peptide, the glucagon, the growth hormone releasing hormone, the glucose dependent insulinotropic peptide, secretin and vasoactive intestinal peptide. So you appreciate this is a diverse set of peptides which have a common origin. So this cartoon here taken from a recent publication, Skeptic Stat. So you have there was a single glucagon or pecab-like gene that duplicated to give a glucagon-like family and a pecab-like family, and the glucagon-like family duplicated again to give rise to this GIP, which is glucose-dependent insulinotropic peptide, the glucagon, and another one which is known as lexanate, which I will come to later on. And the pecab-like gene again duplicated multiple times to give rise to the secretin, vasoactive intestinal peptide. Yeah, is the time over?
Yeah, Putul, you will have to wind up. It is 10 minutes are over.
Okay, I'll just quickly show you the last exciting thing that I came across. So, the basic tools that we use are in silico blast and conserved synthetic analysis. So, we were interested in nucleotides. So, we have the glucagon-like peptide and GIP, which I was talking about, and another molecule which is known as the exendin. Now, exendin is not present in humans. Humans, during the process of evolution, have lost this molecule exendin, and it is found very intriguingly in the saliva of Gila monster. It was the place where it was first discovered, the saliva of Gila monster. It's a part of the poison cocktail of the Gila monster. So this exendin is extensively used as a therapeutic in treating type 2 diabetes, where exendin is an insulin secreta bomb. It stimulates the beta cells to secrete insulin. Okay, so this is my take-home message that I am trying to give. And the road ahead, we will try to understand the comparative biology of the pro-hormone converting enzymes, because these are the enzymes which cleave the pro-hormones and make the active hormone, and these hormones are very important, so we will try to look into these. Thank you, and if there are questions and if there is time, I would like to take it. Otherwise, my email ID and my WhatsApp number is provided in the abstract book. You can contact me there.
Thank you, Putul. That was very interesting, and we look forward to a very fruitful collaboration. Next speaker is Mr. Pranavesh Mandal from Central University of South Bihar. Pranavesh, if you are ready, please start.
Yes, ma'am, I am ready.
Okay, Pranavesh, start.
7 minutes.
Pranavesh, please keep time, okay? 7 minutes.
Can you see my screen?
Yes, yes.
So, good evening all of you. I am Pranavesh Mandal and the partial is from Central University of South Bihar, Department of Mathematics. And can you hear my voice?
Yes, yes. It is audible.
Can you hear my voice?
Yes.
Hello?
Yes, yes.
Okay. So, good evening all of you. I am Pranavesh Mandal, currently marking PhD from Central University of South Bihar, Department of Bioinformatics under the supervision of Dr. Zoology. And today I am going to talk about catalytic transition mechanism of ABC transporters by targeted pharmacodynamic study. So, we all have heard of ABC transporters, right? So, human genome encodes 48 ABC transporters and the excess medical unit of intestine, liver, blood vessels, kidney, etc. etc. And they are basically multi-scanning medium transporters, and they can be importers or exporters. Generally, they are exporters, and they transport molecules via additive hydrolysis. And those substrates are like vitamins, amino acids, lipids. In general, they export toxic materials and they metabolize out of the cell in normal conditions. But why should we explore all these ABC transporters? Because according to CDC 2015, 22% of total all around the world cancer, they are 22% of total base. And they are highly excessive in cancerous condition. And they are directly associated with multi-drug resistance and related issues. Like out of the 48 AC transporters, elements are directly associated with multi-drug resistance, and out of these, they are predominantly responsible for multi-drug resistance. Like ABCD1, ABCC1, and ABCD2, also known as breast cancer resistant protein. Our work is focused on two glycoprotein, ABCD1. So the problem starts when it expresses in cancerous condition. Generally, in normal cell, it expresses normal, but in cancerous condition, the P-glycoprotein expresses extensively. And what they do, they can activate drug molecule directly from membrane or from intercellular cells and can catch it and put it out of the cell via additive hydrolysis. As a result, there is reduced bioavailability in cancer patients. So literally, they have to take drug molecule. Not only human, but also different parasites, different and disease called students also produce PGP, and PGP used by the acid defense mechanism. Like in case of plasmodium falciparum, they become fluorocarbon resistant, and we are the and other diseases like dysphagia, cytomegalosis, they also have developed and they use agents. So there are lots of work done by various scientists, and these are the important work done by different scientists over here. In 1990s, there are past what I mentioned, like in 1995, they started the R1 gene expression in cancer and in humans, in normal diseases, and they find out that cancer exists, and among them, the transporters are the most. So in our study, what we are going to do is, we are going to use targeted pharmacodynamic stimulation to find out the way going from inward organism to outward organism catalytic transition. So, like in our work, what we have done a targeted image. So you may be thinking what is targeted image and how it works. It is slightly different from conventional therapy that is based on nutrient and nutrition. In targeted image, what we do, we add an extra force function and try to find out and try to overcome the energy barriers that may present in large quantum of state change. Like here, we are inward to outward to large scale, and surely they are very high energy barriers that cannot be passed by this normal molecular dynamics. So So what we use to allocate, and for that we require some requirements are there, like there should be two states, like from inward state and there should be an outward state. And we are going to go from inward to outward here. And for that we model the both state of transformation by using Zoology modeling and adaptation method. After that we validate the model in to check save Ramachandran's plot, and after the validation to make it more realistic, we put the protein in the membrane because it is membrane protein. So we embedded the protein in the membrane by using inward rotation to outward rotation transformation. By embedding the protein in the membrane by using charge GY to replace the method. So now see the molecular model. So we use Zoology modeling in case of inward rotation transformation, and we use ITSR, that is physically and hybrid approach of both modeling and training approach. So we use both the techniques to model our required structure, and after that to validate the model particularly, we have used Ramachandran's plot by using Adaptations. So we submitted our model and we see that they are energetically good or not, like the backbones are energetically relevant or not. So we plotted the residues against 5 and here you can see the residues that 97% residues are in the third region, and in outward region and inward region also around 96% there, and generally about regions having 2.2% residues and 2.4% in inward region, and there are only few residues that are in outward region, but that can be calculated to in this situation. So after that we generated the model, we with error, that is also an online error, we put our structure here, and you can see here is the sequences and what the analysis that we did cover with higher analysis, and we corrected. So there are only few analysis residues that are actually having higher analysis. Okay, you can see the overall quality that the error is calculated greater than 90, that is very good. And after that we have validated the model with this is it comparatively showing the packing quality of the structure, like it compare it with other experimental structures like for NMR and for X-ray structure. You can see the overall quality of the model, that is -0.25 and -40%. The quality of the model is very good, so we go forward for R. Like after we
Pranavesh, your time is already over. Please try to wind up.
Okay, give me 2 minutes.
Okay.
After the model structure, we can see that we actually noted the distribution of the rotation and how the structure has been. And inward outward of picture and here inward of state, they have 2 transmitted domain and 2 supercardiac binding domain, and they have repetitions are there. They are playing their role in this translation mechanism. Now after doing the model, we incorporate the model in a membrane. For that, we use request on the liquid like sugar around the protein. Now after that, we request the sugar atom with lipid molecule. And then for our purpose in this solution, we must have to forward the protein. And for that, we use if it is a triangle, forward the protein. And then there are some pre-requirements for targeted hypertonic molecules. Like the atom should be equal to both the concentrations, and there should be RNA deletion peak calculated in that, and we will put that input file so that it can carry the structure from outward to inward transformation. After that, we pass on the targeted hypertonic simulation, and you can see that here the RNA is converged at 166, and where our structure reaches from its inward transformation to its outward transformation, and you can clearly see the catalytic transition mechanism there, that the transition already occurred. And I made a video on this, very short video. You can see here. I'm doing it like it can go.
Madam.
Thank you, Pranavesh. If anybody is having any question, please. Otherwise, you can contact Pranavesh directly. If you have the e-copy of the abstract book, you can contact him. So this was a great session. The last session was great. We had wonderful speakers, and we are very sorry that we had to stick to time, and you could not talk for more than 10 minutes. Although all your works were great, and we would like to hear more from you. This is the end of day 1. Tomorrow, that is 27th, we'll have the first technical session from 10 a.m. So looking forward to meet all of you tomorrow. Be there on time, sharp 10 a.m.
10 a.m. Whom you are telling?
Okay.
Thank you.
Thank you everyone for organizing committee of
10 a.m. we are beginning at
First technical session from 10 a.m.
We are here by 9:30.
We'll be here and others would join by 10 a.m.
Okay, if anyone has any
नहीं तो फिर कल भी ऐसे ही प्रॉब्लम होगा है ना. We will start at 10.